Good afternoon, everybody, from research vessel Falkor. Welcome to the start of ROV SuBastian Divestream 916. Once again, we are heading down into the midwater. Our initial depth this time is 1,200 meters. It's about as deep as we're going to go during this cruise. Once again, we'll be imaging, sampling, and obviously scanning a lot of midwater organisms using some of the equipment that's been specially loaded onto the ROV for this cruise, the deep PIV, IRIS, and there's another couple of other devices as well, like the lunchbox camera and the shadow camera. Anyway, enjoy the dive. I'm going to pass you over to Heather Judkins. Okay, Heather.
Thanks. Hey, everybody. This is Heather Judkins reporting again from Mission Control at the Falkor 2. We are diving today with the SuBastian ROV, and the science team and the ROV team are all here ready to go. I am from the University of South Florida, St. Petersburg in Florida, US. And we are going down to 1,200 meters today. Fast is the goal, so we can spend lots of time slowly going up towards the surface. See, we see nothing. We see nothing right now. We are at 400 meters. We see nothing. You know what this is, Dougal. We're good. We good? For reals?
Looks like a Mars, maybe. Dramatic. That's a pretty long one.
That was a dramatic entrance. As I said, we were never going to stop until we got to 1,200 meters. So, as you can see, we are going to make every attempt to get to 1,200 meters and then slowly explore on the way up. So, thanks for joining us today.
Of doom.
Good plan.
There's a little.
See a little thing.
That one. The one on the right. Yeah, that'd be great. Just a little bit.
Past the iris of Mordor to the laser of doom.
And finally,
Into the
through
cone of
shadow world.
Sardaukar.
Finally, through the shadow world.
Excited.
Swag day. Swag day inside.
This is my travel pouch of stickers.
I only bring 3 stuck stickers.
3 sucks to get.
They're on stuff. Oh, a lot of people showing up here now that the swag's come out. Yeah, all the merch.
What's the merch, dude?
We have Niskins
Free
today.
t-shirts.
1,200, 600, 2 at 50, and 2 at 0.
How many 1,200?
A 1, and then 1 at 6, 2 at 50, and 2 at 0. Or as close to 0 as possible. Thanks so much.
Holy sorrow, Mr. Duncan.
You know, let's do some postcards, you guys.
Hi, how are you?
Hi. Our wish list for today.
I think everything's fair game.
Cephalopods
I think everything's fair game. As long as it fits in a container, correct?
de 8 cephalopods.
Reduces Siphonophorus.
Joel,
Yes,
¿qué querés que aparezca hoy? O sea, que es tu cumpleaños.
el pez que no pudimos comprar ayer.
Looks like you've already collected some jelly on your cable.
Sí, el negro que andaba por
Oh,
acá,
really?
que estuvimos persiguiendo.
Last time we had it
¿Cómo
on
se llama?
auxiliary camera. That one's kind of annoying.
That we saw yesterday, the medical thing.
The one that inflated?
The gulper eel. Yep.
All right, so
Wait, you-
I don't know if there's another common name, but I know
You're
it's
gonna catch that
both.
little
Pelican eel.
Pelican eel, right?
Yeah,
Because
some
of the
people feel-
jaw.
Yeah, some people feel strongly that it should only be pelican eel, but there's actually two different species that have the common name gulper,
Right.
gulper eel,
Yeah.
to make it less
do you
confusing.
want something?
One's pelican and one's gulper, but gulper is okay for both.
Both, yeah,
Yeah.
kind of overarches.
No rules for common names, you know?
Right.
It's
Yeah,
like
yeah.
there's two leopard sharks.
Great
Right.
Southern Narwhal.
Really?
Really? Here in Brazil.
Yeah.
Okay,
Okay, we'll try it.
we will make that happen.
You can find that. I'll be over the moon.
Okay.
Yeah, we will make our best. Narwhal.
The southern, the great southern narwhal.
Oh, we got all sorts of. That's a personal question.
It's a work question.
No, we've got some the other AUVs that are about to flying to this table. And then we've got the Dorado
So on. Oh.
Saw nothing, my friend. Just a little bit of water. Couple little tissue layers. Don't you worry about it.
It's mostly just water.
It's 95% water. We're good.
Pelican medusa.
We'll take your pick or let other people know. This is my travel stash. Because sometimes I'll just kind of give talks. Something. Yeah.
Sometimes you get recognized at the airport, and then you gotta give the groupie something.
Cereboma.
What are you talking about?
Bony fish.
Phony fish?
Bony fish.
Telling her bony fish, Sarah Bomer.
Cerebomiridae,
Keto fish.
I probably should say.
Keto fish is a phony fish.
Keto fish. I'll be yelling at, instead of yelling at Siphonophore, I'll be yelling at Cyphosome. We'll get into the ML
Or
labeling.
squid.
No.
Wait. Gosh, I love.
Oh, it's transparent white.
So, many of us in the room
way
right
better.
now have been doing midwater exploration for a while. I don't think I've talked about one of the projects that I've been doing lately since 2010. I work in the Gulf of Mexico, and I'm a part of the Deep End Consortium. This is a consortium of various taxonomy folks and lots of different projects. And we've been investigating the Gulf of Mexico since the Deepwater Horizon in 2010. We've been very fortunate with the through different programs. So we have been able to midwater column, just like we're doing today. We've been doing it with net systems. We have a multiple opening and closing net system. So we can see who the migrators are, the non-migrators, etc., as we've been talking about vertical migration this week. So we've been able to do lots of projects looking at abundance and distribution of these organisms. We looked at the impacts of contaminants, possibly from oil spill or other sources. We've been looking at a lot of trophic ecology and stable isotope work. So lots of different projects that we've been doing focused around the midwater fishes, crustaceans, cephalopods, and the gelatinous organisms. So it's really fun for me to come to the South Atlantic just to see if there's similar or different species and to use an ROV. Again, in the Gulf, we're not using ROV. We are using a net system for that. So restore.deependconsortium.org is our website. You can see what's going on in the news, see different conferences and events. We have lots of publications that have come out from our efforts over time. And by the end of our current grant cycle, we'll have 20 years of data in the same midwater column. So same sampling method, same everything, which is super unique. There's no data set like it out in the world's oceans today. So we're super fortunate to continue to get funding to conduct our cruises. We have a couple of cruises coming up this summer. The first cruise starts May 7th, which is just a few days after I get off of this cruise. I get to go on to another cruise. So it'll be exciting. I get a lot of time out at sea right now. And then in August again, we'll be out again. So the dates and times, and we have a live blog that we do during our cruises and things like that if you want to follow along with Deep End. I'll pass it over to other folks in the room to kind of share some of their deep sea projects. Nice try, Dougal. I'm watching.
No, it was a squid.
I don't think so.
You see the
I'm going to pass it over to some of my colleagues who are currently harassing me
Yeah.
to share their stories about what they've been doing with the midwater
It
column.
was big too.
Dougal, would you like to share what you do in the midwater column or have done?
It was a big squid.
Yeah. Okay.
I can turn back. I don't know.
I'm not listening to him anymore.
Hi, g'day, and konnichiwa. Yeah, it is a squid. Actually. Trust me, I'm a doctor.
Yeah.
Teaching me wrong.
Don't listen to him, Julieta.
So my name's Dougal Lindsay. I'm an Australian-born scientist who is living and working in Japan at the Japan Agency for Marine Earth Science and Technology, JAMSTEC. We run Japan's research and submersible fleet based in Yokosuka, south of Tokyo. I'm here on this cruise as part of a ocean shot project, a collaboration between various groups in Japan and the US, mainly for my part of the project. And we are developing new technologies to try and describe species in situ, because a lot of the animals I work on are gelatinous and extremely fragile. Just looking at them sometimes, it seems, will make them fall apart. They can be up to 99% water. And so we try and image these animals in situ, get some tissue or some DNA in some way to get molecular data as well, and combine the image-based morphology with the DNA data to try and characterize what kinds of organisms they are. I mostly do taxonomy of different gelatinous groups, including the ctenophores and the many cnidarian groups as well, and do a lot of technology development as well, because while I have been studying the jellies, there are many species that you cannot catch by nets or are very damaged in nets, so we had to develop sampling tools for ROVs, different kinds of cameras that could see different kinds of organisms. And so it's, if a new technology comes online where it seems it could be adapted to be used in the deep sea for sampling or imaging or other characterization of these gelatinous organisms, then we try and develop that in a way that it can be useful for doing those kinds of surveys in situ. Lately, I've also been involved in environmental impact assessment types of work for deep sea mining, or methane hydrates, manganese nodules, other things like that, hydrothermal vent, seafloor massive sulfides. So trying to see what is there in the water column in terms of the entire community, which can be from very small organisms such as protists, although we've been seeing very large protists on these dives. Oh, there's a viperfish, Coleotis, and up to animals as long as 60 meters in length, like some of the largest siphonophores like Apolemia and Preya. So we need a variety of different imaging devices, just as you would need a variety of different kinds of nets or fishing lines or trawls or whatever to catch fish of different sizes. You also need a variety of imaging devices, cameras, to collect information on these fragile organisms over a range of different scales. So that's one of the things we have here. I've brought along a 3D 4K camera setup, a stereo camera setup that we deploy on the front of the ROV to try and capture these very large organisms and be able to measure their sizes. I've also brought a shadowgraph camera system, which is for imaging animals that when we suck them through the suction sampler, the slurp gun, to try and take a sample, in many cases, because they're so fragile, little bits get stuck on the mesh in the sampler, but we don't come back with the whole animal. So by passing the water through this shadowgraph camera system on its way to the suction sampler, there's a huge siphonophore. I think it's Stephanomia amphitridis.
There you go, just put it down. Keep going. He already knows what
We're
it is.
going straight past. Oh, I didn't click my button. Where's my... I'm annotating here in Squiddle, so when we go back through the video later on and try and identify these animals,
I didn't stop
look
for this.
for
Waiting for the
nematocyst
wind.
patches on the ends of the bracts. and how much orange it has.
at some point, but
No, Feisenect. Yeah.
See,
I'm
there you go,
pretty
Dougal.
sure that's it. I didn't press capture at the right moment.
don't go back. Nope, don't feel
Stephanomia fortuitous. Don't go back.
Don't go back. Don't feel bad. You guys are doing great, pilot.
I'm kind of multitasking here. Just a second. I need to type.
You can't.
I can't multitask. I'm terrible.
We can
at it.
have Connie and Yost talk about what they've been doing with the midwater projects that they're part of.
Take it away, Dr. Yeast.
All right. Well, hello, chat. My name is Joost Daniels. I'm a research engineer at the Monterey Bay Aquarium Research Institute. That's based in Moss Landing, California, which is actually very close to a deep sea canyon that starts right off the coast there. So a lot of our work actually is off vessels and with remotely operated vehicles right there in the Monterey Canyon, where we have the very fortunate. We're very fortunate that we can go just a short commute to get out to pretty deep water. However, you know, we do a lot of observations right there in that little section of the Pacific. So it's been really great to come out here and be doing some new observations in the Atlantic. What we've brought, our team has brought a few things onto this ROV. This is actually the third time we're on an expedition with the ROV SuBastian. Once in 2019 and once in 2021. You can find out all about it for Designing the Future, Expedition 1, 2, and this is number 3. And our focus is mostly on non-invasive imaging techniques to try and figure out what the morphology and behavior is of these animals in the deep sea through some 3D imaging techniques. And for that, we've brought on board 3 major instruments, or 2.5 if you will. One of them is DPIV. It's sticking out in the science camera right now. The part you can see is a laser. The laser light's actually generated in a large housing that's on the core of the ROV and then brought through a fiber cable. You can actually see it. It's red to that little housing at the end of the stick. And from there, the laser light's collimated into kind of like a laser pointer type beam. And then there's a special optic called a Powell lens that spreads it out in a fan-like pattern. And part of this setup is also a camera that looks at that laser light. So what happens when we shine the laser, we essentially illuminate just a tiny slice of water, creating a laser sheet that we look at with our DPIV camera. And what that allows us to do when we turn off all the other lights on the ROV to only be observing that single slice. So what we can do then is we can actually look at flow in the water by tracking the particles, the little bits of marine snow that might be moving around. And by scanning the whole thing back and forth, we can essentially scan semi-transparent animals. And that's a way that we can create 3D reconstructions with some processing after the fact, or sometimes a lot of processing after the fact.
Thanks, Ben.
So what we'll do is we'll move the entire vehicle back and forth to do a scan like that. And we're definitely going to do some more of that today. We've done a whole bunch of it this week. And you'll know it when you see it, because then the red laser light will be on, and we'll probably be turning off all the ROV lights, and we'll be requesting some very precise movements from our ROV pilots, who have been absolutely stellar at trying to get these tiny, tiny millimeter scale movements with this 8,000-pound vehicle. So that's DPIV. Just off the corner of the screen, we can also see IRIS, which is a light field camera. So with that, we can actually capture 4D data, so 3D data over time. And this works especially well for more opaque surfaces. So we can observe exactly how things move, like the arms of an octopus or the tentacles of a jellyfish, to help us figure out just exactly how that locomotion is performed, which is something that's a big interest of the Bioinspiration Lab that I'm a part of, and that I'm sure Kakani will tell you a little bit more about. Now, I mentioned 2.5 instruments. Both DPIV and IRIS are instruments we've developed at Mbari and integrated into our own ROVs that we operate there. And ROVs, you might be surprised to hear, are not really standardized. There aren't that many research ROVs across the world, so the plugs aren't the same. So similarly to how you might go to a different country and figure out that your regular USB adapter plugs don't fit into the wall outlets, it's kind of that case here. So we have an adapter housing that adapts both DPIV and IRIS to the standards used on SuBastian. This thing has been interesting to work with. We spent some time at the beginning of this expedition to address some issues with it. It was getting a little bit high humidity inside the housing, which is not desirable. And with a little bit high humidity, means there was actual water coming in. We addressed this issue pretty quickly. About a third of a liter, which is more than our target amount of 0. And fortunately we discovered this very quickly, so we actually were able to recover right away. Some of the electronics in there is pretty hardy, so with minimal replacements, but definitely a sleepless night. We were able to, and critically a new O-ring, we were able to address that issue. So that's just kind of demonstrates this is real, real engineering. These things are not so straightforward to do. There's a lot of things that potentially can go wrong, but we've brought the tools and the people that can help address those issues as well. So we're pretty prepared to deal with these types of situations because, it's not an easy thing to do.
Elio is currently fixing the shadow cam for the next time.
Yeah, so these are actually pretty common occurrences, but that's one of the benefits of having all these different tools, that there's always, we can continue doing our ROV dives and rely on the tools that we have, and every now and then we cycle out certain experiments or certain tools to focus on others, giving us a little bit of time to make improvements on deck. So I think that is kind of the instrumentation that we brought on our ROV.
Rosarian.
At the same time, we're also playing with ingesting data in Fathomverse. I'm sure Kakani will tell you more about that. But any data questions, I am very happy to answer about that. I think that's about enough for me here.
Yeah, let's hope Connie's still got something she can talk about.
Julieta's gonna share what she does in our world here, and in your world.
On the Sea Log Station.
No?
Julieta is not here.
Yeah, you are. Hi.
Hi, but I will not speak in English.
Oh, you don't have to.
I will speak en español, más fácil, me hace trabajar menos el cerebro. Buenas tardes a todos. Quien habla acá es Julieta de la FURGI, de la Universidad Federal de Río Grande en el sur de Brasil. Yo soy postdoctoranda en el Laboratorio de Ecología y Conservación de la Megafauna Marina. Nada que ver con este crucero, pero lo que estamos haciendo acá, tanto Silvina como yo, nosotras trabajamos básicamente en el laboratorio con isótopos estables, que es una forma de estudiar las cadenas tróficas y la forma de alimentación de los organismos. Entonces, lo que hacemos básicamente es, una vez que llega todo al laboratorio y una vez que pasan los animales, las medusas, los sifonóforos y todos los bichos que estamos juntando por los diferentes especialistas que hacen experimentos y sacan diferentes fotos, una vez que terminan, nos dan un pedacito de cada animal, una muestra. Puede ser un individuo entero, puede ser un pedacito de músculo, puede ser lo que tengan para darnos. Esas muestras son guardadas, acondicionadas en diferentes Ziploc o viales y se mantienen congeladas hasta llegar al laboratorio. Una vez ahí, lo que vamos a hacer es analizar estos isótopos estables, que son diferentes formas tanto del carbono 13 como del carbono 15, del nitrógeno 15.
Oh,
y lo que nos permite...
that was so dark.
Lo que nos permite ver es
I don't know. We'll never know.
los tipos de la fuente de energía, cómo se van transmitiendo y en qué nivel de la cadena trófica ellos están. Por ejemplo, no vamos a comparar, comparando en estos casos de los individuos que estamos viendo, un copépodo no va a estar en el mismo nivel trófico que, por ejemplo, un cefalópodo, como estábamos viendo anoche. El cefalópodo va a tener unos niveles de nitrógeno bastante más elevados que un copépodo que se alimenta de krill. En cambio, el cefalópodo se alimenta de tal vez peces y otros crustáceos, entonces va a acabar estando más, entre comillas, más arriba en la cadena trófica. Otra cosa interesante es que, como estamos acá en esta región que no se conoce mucho por eso estamos todos los días descubriendo especies nuevas. Me perdí. Bueno, es una región oceánica bastante pobre, que no se tiene mucho conocimiento, entonces es indispensable no solo conocer las especies, sino también cómo se van relacionando y cómo van interactuando entre ellas. Bueno, eso por mi lado. I have just finished.
Okay. Thank you.
Now we can pass to Kakani.
Did you tell them how wonderful siphonophores are?
Los sifonóforos son maravillosos. Son maravillosos. Estamos muy felices de todos los días encontrar sifonóforos. Todos los días. También medusas. Somos fan de las medusas, de los sifonóforos en este estudio. Pero estamos esperando cefalópodos, gente. Pulpos y calamares.
And octopus are kind of a little bit okay as well.
Yo particularmente estoy esperando una ballena, pero no está para... Nuestros favoritos también son los protistas. Que estuvimos viendo bastante y juntando también para Manu.
Someone's connected to our...
Right, sorry about that. Someone started putting something onto the ceiling speakers, and we just had to deal with that. I'm just re-enabling all the mics in the control room again.
Go to 1200. Let's just get to 1200.
Yeah, we're almost there.
We
you
can
guys.
spend the rest of the day at 11
Someone was
to
asking
25.
We don't
about
cover enough ground
cruises
there.
from
It's either a
Argentina. I
big
believe we
bang
are
back
coming
in
back
that
There's
to
another
Argentina.
Agreed.
Betty
I hear
Laggett.
you.
Can't remember exactly when it is,
I think we should put a time.
but
Like
I
1,000 meters, we hang out for
So-
half an
might
hour.
be
45 minutes.
Daniel
And then
Loretta
800, we hang out for
who
45
is
minutes.
the
Whatever
PM
that
We
time
can do
is.
that.
of
Something like that.
the cruise
I understand
where
the time thing, because then we'll hang
the
down
stream
here the
just
whole
went
time. and
crazy.
won't do anything.
Right.
Mhm.
It's
But also we will
we
go
did
forward
want to
in that.
spend a little bit of time at like
Shallower,
50.
right, exactly. So we want to leave time for that at the end.
Yeah.
Yep, yep. Stick with me, kids. We are getting close.
Are we back
Are we
on?
back on, Jimbo?
Yeah, Mom. Yes, we are.
Okay, sorry for that slight interruption. We're back now. We have about 150 meters to go before we stop and we start exploring slowly on the way up. And I think we have just enough time for Kakani to share a couple other things they're doing at Mbari for midwater work.
Yeah, hi everyone. I've had more sleep than last night, so it's nice to chat with the whole group here. My name's Kakani. I'm a principal engineer at the Monterey Bay Aquarium Research Institute, where I lead the Bio-Inspiration Lab. We've done a number of different projects over the years that's involving midwater animals. Yost talked about 2 instruments that we've developed and used and is actually here with us on the vehicle. That's DPAV and IRIS. We also do a lot of work with autonomous vehicles, and hopefully we'll be able to come back in the future on a cruise with Falkor 2 and do some deployments there. But I think what we're really interested in, particularly in the midwater, is the process of ocean life discovery. How do you do that? How do you do that quickly? And then how do you bring along everyone else to also enjoy the life that we see and experience? And so while, you know, these wonderful imaging instruments provide really fantastic detail, they are really just the first step to a number of steps that are required for understanding and describing these animals that we come across for science. So that's something I'm really excited about, this particular expedition, series of expedition, but also this collaboration with lots of biologists and taxonomists and of course engineers, is like, how do you throw a number of approaches all together to, you know, seamlessly come from, you know, that first look or that first discovery into something that is widely known and accepted in science and broader. So one of the things we think a lot about is data. How does this data get shared? How does this information get out to the world, not just to science? And so we've been trying to do that through the FathomNet program. And as part of the FathomNet program, we've created more recently Fathomverse, the app. Now, some of you, I believe, at least Lisa, you're on the chat, several years ago, I think it was during Designing the Future 2, I briefly mentioned FathomNet and the FathomNet database, and I may have even alluded to gaming as an approach to engage broad audiences. And so what I want to do is talk a little bit about Fathomverse, but then also the next step, because why not? And so some of you, you all will be some of the first people to hear about what we're going to be doing the next phase for Fathomverse. So,
spoiler alert.
I know.
Yeah.
I figure why not?
Yeah. Do it.
So, Fathomverse, really quickly, is an app where research institutions all over the world are sharing their data so that we, as users and players of the app, can look at imagery that almost no one has ever seen before and actually label it, provide a name or data that can convert that beautiful imagery into something that can be useful for education, but also training algorithms that can help us accelerate processing of all that visual data. So Fathomverse has been an incredible project, at least for me, something I'm really, really excited about. Probably my favorite project that I've ever worked on in my career. And we've had more than 50,000 people download the app and come along with us. I think we may be very close to 400 people that have reached the highest level where you become a denizen of the deep. And the user base is represented by more than 200 countries. So this is something I'm incredibly proud of, and I think, you know, the Fathomverse community should absolutely be proud of as well. So we wouldn't be able to do this without you all, and we're just so excited to see where this can go. And while Fathomverse has largely focused on data contributions from research organizations like Schmidt Ocean Institute, and very, very soon we're going to start to see contributions coming in from National Geographic Society, so stay tuned for some of that. The other thing we've been thinking a lot about is, how do we make these models, these algorithms, more available to everyone? But also, how do we take in data contributions from anyone, anywhere, who might have a camera and might be interested in helping us document ocean life? And so that is the point of Fathomverse ID. So that's a new app that...
Woo! Fathomverse ID?
Yes.
What?
We're so stoked that we've been developing for several months, year? I can't remember. Time flies.
It's been more than a year.
Yeah, it started out as... We had kind of a test version that we worked on with volunteers at the Monterey Bay Aquarium. And then we kind of took a step back and thought about, okay, how do we want to scale this? How do we want to make this available more broadly? And so Fathomverse ID, we're hoping to launch it in the next couple of months. I can't say exactly when. But the idea is that anyone, anywhere with underwater imagery or imagery of marine animals can share it with us and we can have it be part of Fathomverse and then eventually the FathomNet database, so we can train better models that can be used to identify all ocean life.
Larvation house.
And then also in the app, you'll have the ability to share an image. We'll run inference on that image and then share back the data or the outputs with you, so you can see in near real time what is in your imagery. So for a lot of us, particularly me, I go to the ocean a lot, and I often come across something I've never seen before. I have no idea what it is, and you know what? It'd be great to have that capability. And so that's something that we're working on. Keep in mind, because we're trying to make these models better and better. They only
We
get
have
better.
made a target depth today, my friend.
With
Sorry to interrupt, Kakani.
no worries.
1200. Ladies and gentlemen, feel free to scope and search.
Yay, we're at depth.
Nice job, team. Thank you for allowing us to get there.
But our models only get better and better
So
with
slow up
diverse
forward,
contributions
slow
from
scan. Let's see what we
potential
see.
users like you.
Drop
So
in.
stay tuned. We're stoked and excited about it. So that's an update, or at least that's a big unveiling of Fathomverse ID, and we are in the process of developing the next version of Fathomverse, which we're anticipating to go live. What was the date you guys decided on in my absence? June-ish. And that will involve a number of improvements, including a new mini game and
Middle Street.
a denizen mode or an expert mode that a lot of users have been dying for. So we've got a crazy, crazy year ahead of us for Fathomverse, and I can't wait for everybody to see it.
I think there's going to be a lot of questions about this, Kakani. You want to say
take
how
a look.
INAT is complementary?
Yeah.
You think so?
Yeah, and I wanted to say, we've also worked a lot, chatted a lot. We continue to be really excited by and inspired by the work of iNaturalist. And how this is different is a lot of the data in iNaturalist, it has what was called classifications, right? So names of objects and imagery. And that works really well.
I think, yeah. Caught my eye.
That works really well when you have
take a look at the
imagery
spectrum
that
to
is
see the zoom.
zoomed in on a single object, which is a lot of the imagery that is in iNaturalist. But for those of you that have been watching these dive streams, know that at least in these underwater scenes, we have a lot of other things. We tend to not be zoomed in on a single object. And so an additional data type is required, and that's called localization. So the ability to indicate what is in the image and also where it is when you might have more than one object in it. And so what we're doing is we're adding additional data type called localization to those images. And so then when you send an image to Fathomverse ID, what we return is not just the name of the object, but also where it is in that object as well. And so the idea is that over time, we'll be able to collaborate with INAT and potentially push those localizations to them and share that data. And this is an ongoing conversation that we're having between our groups, right, to ensure that we're only growing our data sets and knowledge together. Yeah, so I should add that these dives, Schmidt Ocean Institute has been incredibly generous with all of their data. And so all of their dives and their dive data, while it hasn't all made it into Fathomverse yet, there is plans for that already. This is more like, data, let's say you go on a scuba diving vacation, or you might go on a beach walk and you might see an animal. Like it's that kind of data. We're trying to expand data sources beyond, research institutions that are creating this kind of content. Because, let's face it, if we want algorithms to process data, underwater video or underwater visual data from anywhere in the ocean, that's going to require a lot of different scenes, a lot of different approaches, a lot of diversity in data, not just data that's collected from remotely operated vehicles. So it's for me anyway, I think it's a really exciting opportunity, and we'll see where it takes us.
tomorrow, but I don't. They're hard to get the species without collecting it. No, not that guy. There's a couple of larger ones. There's one called Macrotriticus de Filippi. It's got the long arms on this octopus. We haven't seen him in our traps yet, but they could be down the south. They're in the boat. So we haven't seen.
With a little octopus, don't you have to dissect it?
Yeah, that's the problem. That first guy, we have to open it out. So the problem with identifying octopus sometimes is because you have to take a look at their gills and you have to count the number of gill lamellae or the sections of gill they have. So that's what makes it difficult, right? We don't have a chance to collect every octopus out there. And many of these octopus look exactly the same. So there's a lot of cryptic species in the world of octopus. So
Really good
there are some midwater ones, and then there are some one with fins and things like that that you'd find deeper in deeper zones as well.
Look at that.
So there's a bunch.
Oh,
There's a bunch.
A crab versus a squid. Oh, fat on that versus fat on
Keep
Sneaky,
them.
sneaky, all with your swag.
Mm-hmm.
Swag.
Excellent.
SOI doesn't have swag.
We have some little stickers.
Usually give y'all shirts.
You guys have
Yeah. Yeah, we did.
Good shirts. I'm waiting for a hat.
most of my swag comes from SOI.
Good thing it's all the same color too, so at least we have navy shirts finally.
Yeah, because I need more navy shirts.
Nice one.
Where'd that guy go? Oh, stop.
Oh, no.
It looks like.
No?
Whoa.
Julieta.
That was not me. I said stop immediately.
The next cephalopod goes through.
That wasn't me. That wasn't me.
Oh my goodness.
Julieta, don't ruin it for me.
It came from my soul.
Yeah, it really was impulsive. Like,
It was. It was just a natural reaction.
There's carbon.
Oh, yeah.
There's carbon just with more of a shape.
He just fell apart, my friends.
The bell's right
The bell's this red. If we could zoom in on the red just to see what's going on there. If that's possible.
No, that one.
It's this guy. Yeah, we're in the same spot.
You can make it modest, yeah. The very mucoidal
There's part of
feelings
him.
of the cytosol is similar to Mars orthicana, which if we were in the North Pacific, I would label it as the most.
I think there's lots of different parts. I don't see the whole. Yeah, here he
There's
is.
another. So try again.
Maybe this is the part that didn't come apart. I think part of the feeding tentacles came apart, maybe not the whole
They
head.
broke all that stuff from the
Yeah.
first time,
Great.
might be different models.
Yep. So we're gonna zoom in on the siphonophore, see if Dougal can get an ID on it.
Bright orange patches on either side.
Yeah, definitely some different color on this one.
Didn't we see one of these earlier?
This bright orange,
Yeah, just on the
maybe.
The video there's not on the regular canal. Yeah. And it's not a ring, it's patches on either side.
Patches, right?
Yeah. I don't know this one.
If we could zoom in a bit, that would be great. He wants the head and then he wants the tail. If that's possible to zoom in on either point. Yep, we can get a little more zoom if possible. Is that possible? He's not too crazy today. He's not moving too bad. Oh, we might be able to get the tail at the same time. Yep, that'd be great. Oh, great zoom, guys. Great zoom.
But they're very, like, they fill the entire nectaphore. Yeah, let's have a look at that nectaphore if we can.
The top?
Look
that fell
just
off the
below.
bottom.
Oh, the one that just fell off. There's one that just fell off. We could take a look at that, please.
Yeah, I think just like almost fills the entire.
Decoy. Throw a part of your body.
Ooh, so pretty, so pretty.
Yep. Are you guys going to
So pretty, so pretty.
PIV or anything? What do
Yeah.
you need to do, my friends?
We can do whatever you guys want.
Yeah, let's do it. Are you going to use all three? Iris? too?
Yes, we should.
Sure.
Yeah.
Yeah. All right. DPI. Do you want DPIV first?
Yost, do you want to do Iris first or?
I'd rather do DPIV first since it's correlation.
Okay.
Very smooth.
So DPIV, Iris, collect. And are we going to collect in a D sampler? Or a suction sampler? Suction? Okay.
It shouldn't be. It's not like an expert believe me.
Okay. Well, are you sure?
I need to see this on.
Yeah, no, it's great.
You're ready, just tell me when lights up.
Yeah.
Oh,
it's already
yeah, there he is.
Okay.
And it's Hoel's birthday today, so this is a birthday scan.
We're going to give him everything larger than one cell to collect today. Oh, that's a gift.
It is a gift. This is a gift to
This is a
Hoel.
gift.
You want the stuff, right? Here it is.
Here it is, my friends.
Such a good gift giver.
Oh.
It's very pretty.
Ooh,
Okay.
that was nice.
Yep, working on it. Nice.
Are you zooming in?
Outside. Oh no, you're good. Oh.
No.
Huh?
So what are we calling this siphonophore, Dougal?
Greg.
Yeah, Fisonec.
We've stumped Dougal.
Patrick.
I put Maris SP.
Now Google's rethinking
But he's
Paris.
thinking, he's rethinking. Go back to your comment, my love.
Could be. But it could be
Comment.
about money.
I would go back to this.
Not swimming.
I think it's up there somewhere. Yeah.
Oh, a little outside.
Not Maris.
So what should I call it then?
You can call it Mars like with a hyphen.
Not Maris, but Mars like.
So it's Fisonecta viriciformis.
Yes. Yeah. Maroides.
You guys want another scanner too on DPIV? Yeah, perfect.
Iris.
Mm-hmm.
There was some good stuff in there. One more, as they say.
Ooh.
I couldn't tell if there was like a little happening in the center.
Okay, Iris next. Let's do it.
And so you're probably wondering why each of these things are, like what kind of information each
I
of these
think it's
imaging
usually
systems are
straight
giving
in
us.
and out a little bit.
So
Like it's in line with,
DPIV, which is the
DPIV.
first instrument that was the laser sheet that you saw, what it does, at least with animals or structures that are semi-transparent, made out of gelatinous tissue or mucus, which is my
Exactly.
favorite. It gives us these slices of information. And we can use that very much like a CT or an MRI scan to create fully three-dimensional models of those structures. And so we just completed a number of laser scans with DPIV so that we can then
Yeah.
use that data
Fabulous.
to create 3D reconstructions. Now, the next thing that you're about to see is Iris. IRIS is another imaging system. It provides three-dimensional data, but slightly different.
Then you can open it up.
Unlike the laser capability where we can see inside interior structures, this one is more focused on exterior surfaces. So
I don't think they're open yet.
You know, being able to measure the exterior area, surface area of, let's say, the head of this animal, or give us really detailed size information.
We're ready for IRIS when you guys are, team.
Of the length of the head. And so we're able to do that or collect three-dimensional information with just a single frame from this particular camera. And the approach is called light field or planoptic imaging.
Agreed.
And so the idea then is by combining these two imaging systems, we're not only getting these beautiful three-dimensional models that are used to describe the animal, but we're also getting really quantitative measurements to give details about animal size, specific features, etc. And so then the idea is we start with these imaging systems, we couple that with the 4K data on the science camera, and then plus collections will give us the DNA or the genomic information of this animal, and that all together will give us what we are calling a digital voucher, or a potential collection of information or data that can be used to describe a new species. So all of these things are working together, and now, cross our fingers, we'll get some IRIS data.
Lower right corner on that camera.
Dougal is mulling over the ID.
Inside this camera, oh, there he is.
The guidance right now is that it is Maris-like.
Back him up.
Oh, we don't have the colors.
We absolutely do publish our scans and make them available to the public. In fact, you can go and look at them now. If you do a web search for deep PIV, then Embari and Sketchpad, you'll see a library of all of the scans that we've already created using this instrument.
Like that.
Like that is a great IRIS measurement. Well done.
Nicely done, team.
Should have said that sooner.
Wow.
The pilot's like, well, if you just said that 6 days ago.
So feel free to take a look. One of my favorite models there is of a giant larvation that's been animated inside of its mucus house, and you can zoom in and around that mucus structure.
Can we do one more
video?
Close zoom on the tail?
On the tail part, sure. Here he is. Yep, coming in.
Sadly, no.
Oh, that's just pretty.
Did you know we have Zoom on Iris?
I'm sure the pilots are excited about that. The pirates and the pilots are excited.
I love it.
Still right in, yeah. He's still right there.
Wow, so dialed in.
I love that he's very calm.
Coel?
Yep,
Or the...
here he comes.
Yeah. Everybody's very calm.
Wow.
Oh, that is great.
Oh, I can't wait to see this data.
Yay.
Lovely.
Nicely done.
Lovely.
Good.
So those colors that you see on that feed
So
is
then
a
if we
depth
can go
map.
back to video, not quickly, but next to get to the tail zoomed in for Dougal, then we're going to collect in suction sampler and go into container 1.
Shot it through really slowly with microbursts.
No.
So we want
Suction sample.
more video first.
Yeah, just of the tail. We got the head really well, but I don't know that he got enough of the tail that he wanted.
And so you saw colors in the feed on the live stream, and those colors are depth information. So like the distance of that particular object from the like the camera sensor. And so the colors are from red to blue. Red is close and blue is furthest away, and so like the sweet spot is kind of the greens and the yellows.
Yeah,
so we definitely got a lot of great looks of that animal in that depth range.
You want middle and end then?
Yeah,
Second half?
closer to the middle or the front where the stomachs are still attached.
Okay.
Those big orange things.
Yep.
Look, one of them has fallen off there, but
Yep.
where they're still attached.
Yep.
And I thought I saw a couple of vents if they
Yeah,
got
if we could
shrink
get
inside
some zoom, that would be great.
Uh-oh, are they losing?
There, he's dropping the stomach parts, yeah? That's great. That's great. Let's see if we can get a little more zoomed if possible. Nicely done. Into focus. Nice. Dougal. Good. Getting there. Yeah, we need a little focus on that. He'll be good. Yeah, he's moving away a little bit. There it is.
Wow, look at that view.
It'll come back. It'll come back.
So pretty.
Here it comes.
How can you hate St. Forest? So pretty.
Yeah.
So pretty.
You need any more close up?
So more close up if possible.
Okay. If we could get close up again on the middle of the stomach region or the tail region.
So if we're not sure what it is, does that mean it's a possible newbie?
Yes, exactly.
Yeah.
We could get a little more in on this middle section.
Long and
That's the
very
goal.
diamond.
If it agrees with you. If it cooperates.
If he's getting close, maybe we should suction him up.
One more time.
Yep, we're gonna give it one more shot and then we'll suction. Do your things, pilot. There you go. Nice. Focus it up. There you go. Dougal, helpful. Okay, if we can get it towards the front a little more, if that's possible.
Yeah, it's worth mentioning that the 3D scans that we put up
By
on
Google?
Sketchfab,
Yeah,
they are
Yep.
publicly available.
There you go.
Specifically,
You good?
it's, with
Okay.
attribution,
Let's collect this,
non-commercial,
baby.
share alike, so
Nicely
you can
done.
use
Yes,
them for all
suction
sorts of
sampler,
things
looks
like
container one.
presentations. But yeah, if you have
You got-
interest
do
in
we have
using them
security
for other
camera?
things,
Oh, you got it.
definitely
Sorry.
feel free to reach out to our team and we can chat about it. It's really important to us that these models get out and are used
Something larger
to inspire
than a
people about
single
ocean
cell.
life.
Thank you, Ocean.
Mhm. You got it.
And then the pump to go off as soon as it's been sucked in. Yep.
The suction is on already, so if you're ready to turn it off, you want to, when you think. Oh, he's in.
Yep. He went through the suction.
Forward.
Bring him through slowly.
No sign of him yet.
One microblast at a time.
Nothing yet.
Give it
Your
some-
shadow cam's
Yeah,
off,
give a little
so it
micro,
shouldn't
little
take
micro
very long.
burst would be great.
Like getting strained in the mesh and it's not getting caught to it. Suction is not too strong. Just a bit of microblast them so they get without the
Is your shadow cam in there?
No, it's just so they don't hit the mesh and get strained.
Very tight. 2 dots. Like this.
Let's try another microburst. Thanks.
We saw them in
And he definitely went in, right?
Yeah.
There he is.
Okay.
That's the top.
Yep, sectioned off.
Very small.
Do you see the
Yep. We can
That's it,
turn the
and turn
canister.
canister.
Thank you. To number 2 would be perfect.
That's great.
Nice job, team.
That's fine.
Nicely done.
Nicely done.
animal collected.
One.
Yep. Sampler 1, Sapphire 4.
Maroides.
Greg.
Maru.
Maroides.
I think it's in front.
Oh, sinker? Is
16.
that what you're looking at? Yeah. Yep. I don't see it in the side cam yet.
Yes.
Mm-hmm. There it is. You guys want to take a look at this mucus house?
Yes.
Figured that if you guys could zoom on said house, that would be great.
Keep some distance.
Yeah.
You might wanna
Yeah, I
see
don't want to
it
get it
too.
too close because the wash. This is the whole, it'll fall apart very easy. I don't know if anybody's home. Anybody see anybody in there? The larvation house.
It's a sinking house.
Definitely
But
sinking.
can we just scan it?
Of course.
Just like do a one scan through?
Yeah.
Because I'm,
See how it
I'm
goes.
just curious.
Yeah, of course.
And if we can do it.
Yep.
The bottom part is like very opaque and there's
He might be in there.
transparent part that will be up there as well.
Somebody in the middle.
Yeah, so that's what I'm wondering. So you see.
That's fine.
Maybe not. Okay, you guys.
I guess the target would be putting
Yep.
the minimum in the center of the field of view. Because I'm not sure what's there.
Right. So that you want the like the V this way, right in the middle. Okay, so the bottom of the V towards the bottom of the laser field. Right?
Just when you go to auto. Just when you
Okay. Are you guys ready? We're ready? You good? Yeah, they're good when you are. Can you guys
Maybe they can have the science camera so that we
Zach, can you get the lights in a second?
don't see. Maybe too big.
Yep. He'll tell you. I'm not telling you.
So how they that down.
yeah, of course. He's in here. Yep. He's on the outside, yeah. I think he's already broken. Yep.
Which part you want to?
Do this part because it's right here.
The one that's in front.
Yep. Do the one right in front of the scan. Because I think if we tried for that, it'll just be broken. Yeah. Try that for that.
I want to actually look at it too.
Yep. He's not there yet.
Yeah, no.
Not this time of day.
Oh, you got it. Did you fix the light sources?
Yeah.
Oh, you fixed it.
For some
Okay.
reason. We're starting to get the first bit.
Well, it's big then.
Yeah.
Yeah, exactly.
Well, it's changing shape as I get there. It's coming.
Still don't see it on my.
No. Not yet.
Eyes coming.
I see like
Which
pieces
part?
of
Which
stuff.
part you want?
Just try straight through. I mean, the whole thing would be great, but I don't think we're gonna get the whole thing.
Oh, it's coming now.
There it is. And we can go back through that again. Yep. If you want to go for the, you want to go back through this guy? There you go.
No, yes. I mean, there's definitely structure in the sheet.
We do it
but
one
I
more
don't see-
time.
Let's just start with all that.
It's just this, yeah. If you could do that. Yeah. Yep. Here it comes.
This is the larvation.
No.
Okay.
Pushing forward. There's some other little bits out there. See if anything reveals itself.
Mm-hmm. This guy? Yep. Closer to the source? Yeah, that guy. You got it. Yeah. All right. Okay. You good?
Yeah.
All right. Nicely done. Thank you, guys. He's like, I'm out. And there's some of that over here. Oh no, wait. There is just a little more of this one over here. This came yesterday, I think. Uh-huh.
Okay.
Put something one over here.
Oh, you good?
One. Number one is used.
Number one.
Yep.
Okay.
Number one, right?
Yeah.
And this is.
Yep, we already did one Niskin. The next will be at 600. Yep. And Bari. Yep. Swag items, if you like. Merch.
Just so you know, we're just doing a watch change here.
Hold on. Awesome. Thank you.
Mm-hmm.
All right.
Hi.
Hi.
We made it to 1200. So we are moving up slow and forward, exploring away.
At what cost?
Yeah, we went straight Mossad seal count, didn't we?
Yeah.
They're calling out coelacanths and all kinds of things to try to make me stop, and I did not. But now we can.
Oh.
We can now. We're slow in searching.
Keep it at squid level, right?
No,
She wasn't
I'm not even
following-
turning it up for that anymore.
You will start going.
The boys cried seal count.
Yep, slow scan would be great. We'll call stuff out.
I mean, squid, there was a possibility.
Yeah, it was. I think it was a big Matthew Laggard. It could have been a Lamponictus.
It was
perhaps.
not that
It
shape. I
was.
saw the shape.
And it had a parasite hanging off the
Uh-huh.
fin.
Yeah, you
I got
saw
a frame
that.
grab.
And you saw all the photo for us too, right?
No, but the frame grab was good enough that when I put it on 3x zoom,
Uh-huh.
I could get a pretty good look at it.
Yep.
That you couldn't see in the video, obviously.
Sure.
Uh-huh.
Going. It keeps going. There's so much belief happening here.
Yeah, exactly. The trust circle is starting to break in the room.
Is that something we want?
So when you do the laser scan, how much spatial resolution?
Yeah, I was gonna say it's a little slower. That was a Rhizarian.
It's about
Yep, yep. Slow scan. I know we haven't been this deep in a few days.
Actually, get slightly better
We're not sure what to do with
spatial
ourselves.
I want to see
because
on
of the
these
scanning,
deals.
you can
Oh yeah,
better spatial
that would be
line.
fun.
Tends to be around
To the right.
0.4
To the right.
Right there, got the little guy.
Yep, I'm assuming this guy right. Yeah.
Might only be a key, but just in case.
Definitely
It's
something.
looking a lot like a key now.
Because it's a
Huh. I like it. You got
There's
Keating
something else in front of us, though. Like
Yep,
Yeah.
this guy back here.
the depth because
Keating Nath is lovely, but we're gonna move on from that.
that's what you're doing the scan.
Zoomed out by default, the XY resolution is about 1/4 millimeter per pixel.
Yeah.
But we can zoom in 10 times. We don't really.
A lot of mucusy things. Yeah, lots of mucus houses and parts we're seeing.
Yeah.
It just looks like mucus.
Mucus.
You're good? Okay. So, yeah, I think we're good. We can keep exploring. That'd be great.
Threads or individual.
Yeah,
Far
we
right.
broke up a larvation
Far
house just a
right,
minute.
just below center.
This guy?
No, oh, it could be a filter.
We can get like pre-filters. I'm trying to think about the diameter of those. So we can get their presence.
Could it be another siphonophore?
Exactly.
And
what
it looks
this
like it might be
size
further
is,
down,
right? Because there would be some
Cruising in on that guy when you guys are good to go.
That is the actual structure itself. Yeah. So there's a limitation, especially with
It's
we're
no,
not
we have
getting really.
Looks good.
Paisonecte.
I'm just here
Yes.
for the Nomias on this.
Paisonecte,
Paisonecte.
frill agalma.
Could you run
That's really deep.
Oh yeah.
What are we at now?
We're at 1174.
Holy
They don't care. It doesn't seem like there's too much there.
Yep. Zoom just like this is great. Yep.
Separate.
No, I'm telling you, he's happy though.
Signal out.
Look at that.
Is this Zoom good, Dougal? Do you need
Yeah, that's great. Yeah,
Okay.
That's awesome.
Perfect.
Great for machine learning model development.
Yep. No worries.
Religion, PPIs.
Can we play with lighting a little bit? Because now we're looking a little noisy. Is there a lighting setting where you can still
Well,
see
right,
well?
and at some point, just like you said, it depends on the visual.
Because you changed, you turned off some lights,
Going
right?
up. I gotta bump in a little more. Trying to be very careful with getting too close to this guy. Messing with it too much. Go.
Great. Yeah. Dougal, good. Little focus. He's moving now, of course. He was perfecto for a hot second.
We were really talking yesterday about.
Yeah, of course. Starts moving. Are we scanning or doing anything with this? No, we're good? You got enough? Okay, we're good. We've got enough. Oh, here's the perfect zoom, too. Oh.
Man, I should have kept recording on.
That's great. Perfect. Okay, thank you, pilot.
That would run midwater transit.
We are continuing our dive slowly and scanning back towards the surface. We have a 7.5-hour dive or 7.5-hour dive today. And we're about an hour and a half in. We're gonna slowly scan this deep bathypelagic zone for a little bit until we get up over 1,000 meters where we enter the mesopelagic zone. That would be a cyclothony down low, left. Perfecto.
What the length of this?
Works for me. I'm hearing no complaints, so I'm gonna say good.
So.
I think that's just a mucus house or a mucus strand of some sort.
We should bring at least two. If we bring, I know we're gonna be off brainers, we wanna bring more.
Looks like another mucus situation. Oh, you think that's a siphon? Wait, on the right? Where are you looking, my friend?
Down.
Oh, you went down. Sorry, Dougal, I missed that. I was looking on the other side.
At that time. And then once it comes back, we can.
You're ensure that you don't, you have to drive away.
Mess him up. Okay.
Hopefully it's not fired up.
This guy,
That would
yeah.
be bad. We would wanna
Yep. We're getting zoom on him, Dougal. Yep.
That's just a tentacle that's come off the main.
Yeah. Okay.
People know.
Good to keep going? Okay.
There's.
Don't catch the tentacle of the camera lens. You did that before.
What, you don't want something constantly just
Waving around.
taking your attention away.
Added challenge.
Straight ahead.
I don't know what's happened. Something's happened here. There you go. Yep. Center. Yep. Center.
For the goal of the judge.
Definitely not moving.
Keep doing that.
And then whatever's down to the right.
Yep. I got you. Is this a Ketignath?
Keeping that.
Did you get Ketignath again? No, it's a siphonophore.
Oh, the Medusa. The Medusa.
You want the Medusa?
Medusa Filigalma.
Yep. Bye-bye siphonophore. We need Medusa focus.
It's very small.
Yep. He's tiny. We'll come on in when they get ready.
It's really hard to scan too.
Oh,
Yeah.
down.
Yep. Take your time.
And the method. Captain Medusa.
Ray.
Come across. Things.
Probably do it just.
Yep. Here it comes, Dougal.
Oh, and then another.
Uh-uh.
We wanna bring out is.
Possible smooth here? Is that red? Oh, the red's just the iridescence.
I think so.
That combines with
It looks like a kind of Smithia. We don't have one of these yet.
What are the gonads? What's halfway down the bell?
Yeah.
That seems low.
That is low, yeah. We don't have one of these yet.
Okay.
So, if we're gonna, get some good video first.
It was 50-50, right?
Working on it. Working on it.
It's very similar to one we see in the Arctic, Smithia arctica.
Oh wow.
But
And remember, this is the. Didn't we see this or somebody similar the other day because the tentacles were kind of curled up at the ends again?
Yeah, that would be a Colovenema, but they have long linear gonads.
Yeah.
These ones, as you say, are further down towards the Bellegram and oval.
Yep.
They're very different. I wonder what the area around the duck
Yeah, that's great. There you go, Dougal. Oh, hold on one sec.
I think this one.
It's
so
tiny.
small.
Yeah,
It's
it's
not really
tiny.
musky.
Yeah.
All right.
We're suction sampling. Yep. This is going into canister 2 if successful.
Wow. So the only other Smithia that's outside the
Smithia.
Arctic right now is Urigasta, which has a red gut. And
I've heard of
it's not
Smithia
a
in the
red gut.
Antarctic. Right. Okay. Smithia.
My master's student, Ricky, is currently
Start over. Yeah. Like giving Darren a new tomato species right now. We're like, we're about to submit. Right? People have already encountered Talifron and Fableverse. Already getting boxes on there.
Oh, yeah. Yeah.
No, I didn't.
Yeah.
Did it go in?
Didn't?
Yeah.
I thought it did. I think it did.
Very slow.
All right.
Right.
It got in. It just went in.
One forward.
Yep.
Shadow
Yep. You can switch off 2. Thank you. Nice job. Woohoo.
Awesome.
All right. 2.
So many tiny jellies, dude.
Tiny jelly.
Really cool.
Tiny Medusa. Tiny Medusa. Ni-
Since we started-
Just throwing it out there.
Calculating that rate and you'll be up by Christmas.
We did discuss, do we have a strategy of X amount of time at a depth and then go up a couple hundred? I'm happy to whatever you guys want to do. It does whatever you want.
I think the actual problem is that we don't cover that much grass.
That's the
Right.
problem. Right.
So it's not so much like, oh, we need less time in the steps. You can still be stopping on
Something
tiny jelly
somewhere.
for 10 minutes,
Right.
go 6 meters, and do another 10-minute
Right.
tiny jelly session.
Right.
So if we want more coverage, we just
Or
do
maybe
that.
we make an X time to go up to 500 no matter where we are, and then up to 250. You wanted shallower later on,
We've
right?
had depths with almost nothing that we spent a heap of time in because we wanted to go to that depth and
Right.
look around, right? So,
Right.
And if we're seeing lots of stuff at a
You
certain
should
depth,
hang.
it's almost, you should hang. Because
Yeah.
even if they're small, there'll be bigger things that are eating them.
Yep.
We can just go from slow mode to fast mode instead.
Sure. Sure.
We do a little bit of fast mode and something like.
Well, the real solution is we should just have AUVs.
Fast mode.
Noted.
And then Dougal's gonna hold on to one of the AUVs and just dismount whenever he sees
Yeah.
tiny jelly.
Let's be a Dougal in a pressure house.
It's not a dive, it's jump, but like, I don't know.
In the middle on the left a little bit, there's a
Oh,
house.
yeah.
Yep. This guy right here.
Another one
Thank
of those
you.
hot air balloons.
Looks like another house.
Put your pews on.
See if somebody's home. Do you guys want a DPIV?
Just run the laser through it. Yeah.
Yeah.
I don't have high expectations. But let's,
Yeah. Gotta test. Gotta
so
test
just-
it.
We run the laser through it without ever going backwards at all. No reversing.
Down a little. This is a UMO, an unidentified UV object.
Yeah, it's most likely a sinking larvation house, perhaps. There's no larvation home at this point, and the team wants to kind of take a look to see what kind of structure possibly could be in these mucus structures. So that's why we're practicing pretty much.
Yeah, it's just faint structure. I don't see another.
I don't see it.
That's right. In a house. It has two lobed in a house. Yeah.
Wait, I think we're doing it. Are we going to try again?
I think it's.
Okay, we're trying this again, even though I think it started to break. I see it, but I think it started to break.
Should we reduce ROV lights to get better?
Oh, it broke. Yeah, it broke up, you guys.
That was good. That was good to see.
Yeah,
Good to see.
keep going.
There's a little blob sticky there. Definitely didn't see anything swimming away.
5.
Large house-like structure.
They're supposed to be.
Right. Find another
Stand by. I can tell you. Yeah, I should look. It's right here.
But here's the actual house.
delicate. Those are
Got
the
it.
one
That's just a
scan wonders.
It is 2:30, 4:30, 4:30.
Yeah.
Yeah, 15 minutes. And then Kakani or Yost are after you, and then Russ and Emily have the second dive. Because the second dive was only 4 hours still, right?
Yeah, okay.
Mm-hmm. There you go. Yep.
Thought I was almost caught up, but I thought. What's that?
As an option. Doesn't matter to me. Yep, 15.
Alrighty.
Sweet. Okay. Scanning away. Mm-hmm. Well, and I can stay a little longer. I don't mind staying a little longer. I don't want to stay the whole, but I can stay a little longer. Right, Connie's here too. We got options. We could figure it in house. Shrimp.
Rise area.
Oh, that was a fish.
How do you feel about this depth? Feel okay?
There's not much here, is it?
What do you mean? There's lots of snow falling down. We're moving up. We're at 11:44. Who's this little guy? Little Hyperid? That was a little amphipod. Yep. I think this is another sinking house. All mucus, adventure. Yeah. Yeah. Mucus. Yep. We can keep moving.
Yeah. I could probably step out though, or.
Golel and nurse without the. It only has the skull on, none of the.
Where are you?
So it's very small and transparent. Don't worry. Sorry.
I was gonna go to the middle. I was gonna go to the middle organism.
Okay.
Is that a Kevignat though? Am I gonna get stuck on this
That's
worm?
a key name. Yeah.
Okay, so we can keep moving.
Yeah, I just saw that little worm.
Yeah. Yep.
Try and follow, but
I don't. Yeah. Kevignat.
Is that a fish on the right? We're gonna take a look at the fish for Marcella.
Where are you?
Bottom right. Bottom right. Then just off the screen now.
Didn't see it. You got him? Oh, she's got him on
Yeah, I
the
see him
side
on the
cam.
crossing
Yep.
camera now.
Good call, Yost.
might get a little.
Sorry, the phone name.
Just on it.
Is it a cyclophone? It's okay. Yep.
It's facing away from us.
Yep.
Where is
He's not hearing
There he is.
me. This little guy? Yep, that'd be great.
Show his face.
Yep.
You
classic, as always, get fish
Oh,
body.
I think
He's thinking where the paparazzi and trying to hide his face so we don't see who he is.
I think it was when you first
yeah, get the auto.
This is
We need the
something
auto
that kids are
not
very
on.
familiar
Auto
with.
never works.
Fish
right?
butts. Anytime you try to take a picture.
There he is. It's focus. Isn't this a cyclothony?
Yeah.
As I say, we're very again
It's a different
cyclothony.
species to the one we have.
It is. I think it's in the Gulf. We have Cyclothony obscura is under 1,000 meters. Yeah, he's great.
Very robust, isn't it?
Yeah. So cyclothony are one of the most abundant fish in the world's oceans. They are commonly called the bristlemouths. They are everywhere. They are a small-sized fish, but they are huge in terms of abundance. They are by far the most common fish species that we collect in the nets in those deep end projects I mentioned earlier. Yeah, he's staying very agreeable right now. Here we go. Don't do it, dude. Little bit, little bit. Good focus. Nice. It might not. Is it alive?
Yeah.
It was moving a second ago.
He's definitely
It's
not.
not acting very alive.
I want to be that relaxed.
It's
I
doing
say,
that-
is it the possum cyclothony?
Yeah, yeah, it's doing the playing dead so that nothing tries to eat it thing.
Kind of
Oh,
weird
okay.
because
There
it's
was a
still
movement.
just fish
Okay. Yeah, I was gonna say he's.
flapping about.
Nope.
in the
I think we're good. Good with video. Thank you.
Oh, there's no photophores.
I don't think it's a
It's
cyclothony.
Cyclothony obscura.
That's what I said. They're found below 1,000
I
meters.
think you might be right.
Once in a while. Once in a while. Once in a while. I'm not a fish girl, but
I was just because I've never seen one.
yeah, we
Like,
collect them in the Gulf all the
ever,
time.
even in the net. Really?
No, yeah, all the time. So we
Alright.
know we're below 1,000 when we collect the obscura.
Yeah.
It's pretty cool. And again, there are a bunch of other cyclothony species. So I say obscura with about 75% confidence.
Right.
I'll give myself a C on that. Well done. All right. Thank you, guys. We can move ahead. Great zooms. Thank you.
Isn't it so cute?
Sideways,
Isn't it a
buddy.
cute fish?
He is important. A very important fish.
You wouldn't get much of a fillet from him though.
You
Because
wouldn't
it's
guess
pretty
it's
small.
misbehaving.
All right, moving on.
Man, it's just-
Pretty good size. From what we have seen. Okay.
Yeah, exactly.
Going for it.
Thank you.
Like good food for something
Its
else.
career as an actor is going to be limited, but maybe as a model. Oh, there you go.
There's a house.
Oik.
Stop house. Oh, it's an oik. Never
It was
mind.
an oikoplurid, yeah?
I thought it was bigger.
Blue eggs are Yukita. Russ is talking copepods.
Yep. I was hearing obscure conversations going in here.
Oh, there you go.
You bring your Coke Pod shirt? Oh no.
I don't have a copepod shirt on today, no.
No.
Rosaria.
Middle. That's just a erase area, yeah?
I think it's about time we zoomed in on some Rosaria
Yep,
as
yep.
well.
I feel we should. Next
we at the
Rhisarian, we'll
bottom?
go
We're
for
going
it.
for bigger stuff.
Yeah, we made it to 1,200. We're on our way back up.
Maybe if we go a little faster, then we don't see him so well.
We've gone 64.
I-
Bottom of our.
I think it's wonderful.
This is progress.
We probably
Not
were in there. I
perfect.
could hold the blue eggs for Yukita.
I didn't see anything.
I know those were in there.
Heather just wants to get us in and out. All the in-between stuff is kind of a distraction.
I was looking for the small one.
She has a mission.
Is it a small one?
I don't know. I just heard somebody say, blue eggs.
And you want to go fast, and I want to go slow. And then some people want to stay deep, and some people want to go shallow. So it's like,
Yeah.
Way early this morning. Like,
We're a great group.
yeah.
You could slow it a smidge.
I left.
Yep, left
Yeah,
center.
Oh, it's a
Galma.
Frillagalma, I think.
You should be excited about
It
that.
would be, because I'd only ever caught a few of them in Japan before coming here, but they're everywhere.
Yeah.
Off to the right.
Yep.
Where am I going?
Going over here.
I always enjoyed
We don't need
Are
to
they common in
study this
Monterey?
one in detail, do we?
No. Okay, we can go on. We can go on.
You weren't gonna go on? Okay.
Yeah, there's
We're moving
limited
ahead.
time.
Okay. Thank you.
I think all the Phyllogamas together.
Oh, something black. Down.
Where?
Just past where the Phylogama was.
Stop.
Go down. Go down with the ROV.
Down.
Down with the ROV. Slowly forward.
Down, slowly.
Very slowly
Is
forward.
it very small or
Go
very
low. There it
far?
is. Just came back on the screen.
Here it is.
A red cuneiform.
Black object.
Black object. You got him?
I see him.
Oh, it's a red object.
But he's
perhaps.
out there.
When
He
we
might
get
be
closer,
red.
We'll find out.
Yeah, I was gonna say. Oh, it's gonna be a shrimp.
Little ruby sedipid, maybe something else.
We keep coming forward. Here it comes,
Red
Dougal.
Shrimpy.
It's a head-on shrimp.
No,
No, a tail on
a
shrimp.
tail on shrimp.
Shrimp butt.
Oh, well,
We have a
there you go.
shrimp,
Shrimp
tail in the view
We got a
right
fish
now.
butt. We have a shrimp butt.
We're
doing well.
All right, sorry about that.
Next up, the squid.
I'm
No,
not
that's
used
okay.
to identifying shrimp by their butts.
How about
Here he
this
is.
then?
Let's get a zoom on him while he's hanging out. Thank you. Beautiful.
I think it's a mysid.
It's small. And bye-bye. Okay.
And he's
Thanks, you
off.
guys. Good eye, Dougal. Good eye.
Did you go to the Marine Imaging Workshop in Victoria?
No,
I don't remember if you were there.
Julietta, how much chocolate are you eating today?
We need what we need. No shame. There's no judgment this one. Not yet.
Okay.
Later.
Not yet.
I saw 2 to 3 bars.
Did you get some of the cake earlier?
Yes.
No. That's why she needs. Yeah.
I did.
We had our fill of chocolate.
Mm-hmm.
There's some cake earlier.
Uh-huh.
You eat as much as you want.
I was not judging. It was pretty funny. I was like, wow, you've got a lot over there, my friend.
I have one, and then you just give me another one. We're all rich in different ways.
Exactly.
I can't say anything.
Oh, I already had a
Fire
piece. I had a small piece of chocolate and a piece of cake.
Part of my mental health.
Oh, let's get. Nope. You want them? Too late now. But
Big things.
this is not a cyclothony in front of us. Is it a Keatignath? It's a Keatignath.
All right, we're going.
Yep. Bye bye Keatignath. But what's this guy? Keatignath.
Okay.
That.
Whole phylum where people just know they exist and ignore them.
No, Russ has been identifying them.
I feel like haddock is not somebody who Nessa worked with.
Would you like to talk about Keatignath
Eric Toothson.
to the crowd?
Bill Murray.
Eric Toothson is a Keatignath
All
person.
we say is Keatignath.
Keatignath.
Well, yeah.
Keatignath, spine jaws.
Okay. Great.
Hairy jaws.
Why do we
Just
see so
like
many in
Russ.
Why do we see? Because they are incredibly effective copepod predators, and you can't see all the copepods that are out here, whose numbers are absolutely staggering. In a square kilometer of ocean, there are more copepods than there are in the Milky Way.
Wow. Okay.
There aren't any copepods in the Milky Way, as far as I know.
That's the exact same number. This is the total number that's in the Milky Way.
Oh, you knew that was going to get called out.
Except on the planet Earth, which is in
In the Milky Way. Ah, there you go.
Which contains all the copepods.
That was good. There's all the copepods in the Milky
Cyclothony,
Way.
we're okay. Or
We're
Keatignath.
actually really
It's
close
actually a
to
Keatignath.
all those cool fish in the Milky Way.
Does that answer the question?
Yes, we just see a lot of them, and we don't say much about them. We just say it's a Keatignath.
Yeah, we're
crustaceans.
over there.
We don't see as
We're
many
seeing very
copepods,
few of
that's
those
what I heard.
crustaceans in the video
So
because
see,
of
come on.
And over this cruise, we've seen various species. So there's more than one species we're seeing out
Yeah,
here.
yeah, we're seeing the ones that we pick up on the most, that the orange ones, which are heterochronia, sorry, euchronia, fowleri, most of them, and then there's some more transparent ones. We peed out in a fairly rushed way, about half a dozen different species
Okay,
that
cool.
are here,
Great.
and that's not been an extensive hunt.
Right, exactly. Yeah, we haven't focused
being
100%
replaced.
on these guys. Right, yeah.
does
Great.
that feel?
Okay, thank you, Russ.
You okay? You okay? Yeah. You might
It's
miss
this
the
comment you made
siphonophore.
about
What do you mean?
siphonophores.
That was pretty funny.
Shrimp.
Shrimp. Yep, let's zoom for a second. Nope, nope, no, we're not. Only if they're staying nice and still to get a quick zoom. There's just happens to be no crustacean.
A lot of people
Right.
probably think
Should be. But there's no crusty
really
fish
tiny
on board.
jellies now that all look kind
They're
of
getting
But I'm happy.
Oh yes, feel free. There's lots of presents in here. There's some in here.
And Heather, I can relieve you whenever you're ready.
Yeah, okay. Yeah, I didn't know what that plan turned into. Yeah. Maybe like 15 minutes?
I'm just looking at data.
Yeah.
In the meantime.
You're like, I want to change, so I'll look at anything else. All right.
Please.
I'm going to turn it over to Kakani for a little bit. We'll be switching in and out throughout the next few hours. Let's see what we find. Oh, that's a
And
dark
if you hear
chocolate
any
right there.
crinkling in the background, that's absolutely not at all the chocolate we're eating.
That would not be near it eating a
Vegetables.
chocolate
No
bar.
chocolate. And it-
Definitely
if and
near,
when we make
on
it to
the sea log
600.
station eating dark chocolate.
Definitely not doing that. Don't worry, guys.
Yeah. Our guesstimate is that there's about 60 pounds of chocolate was loaded on right before this expedition. I don't know how we're doing, but it feels like we've made a dent.
Yeah, the ship seems lighter, don't you think?
Yeah, I know. They had to do crane maintenance earlier. To get
In the middle, in the center, heading down towards the bottom now. I think it's a larvation house, a big one.
It is so
Sort of big. Sort of big.
You can see legs.
You're just playing Kakani.
Yeah.
This
This guy,
actually a jelly.
I think
No,
that is
there
now
I
it's,
see it, but
it's the boy who cried
Kind of
jelly
scannable, but we'll see.
or squid. Is that
Oh, I think it's a
a
fritillarid.
fritillarid?
It's a fritillarid.
Oh,
Yoo-hoo!
well,
Oh, but we
was.
kind of.
But I don't know where the-
It
Do
was
not
a
see it
We
fritillarid.
were pretty
It
far
looks like a
off. I'm
butterfly in the shot.
surprised that we were seeing that.
Fritillaridae. So that's what one of the ones we're after.
Yeah, especially for us.
And they're kind of hard to spot because they're all bunched up. They just look like a piece of marine snow.
oh, is that so?
Down
Ooh,
It's a little juicier than a filigama.
that's a
Oh, yeah.
Lower right.
juicy
Go lower right. We got a bit
siphonophore.
more cyphosome
Levi's
happening
an
here.
X siphonophore. But what kind? Any guesses in the chat?
Let me guess. I reckon the tentilla will be floating up above the cyphosome. What
Okay.
do you reckon?
Okay.
That's a.
I'd say that's a good reckon.
Is that a reckon?
Yeah.
Oh,
I'll reckon that right with you.
you'll reckon that. Oh, and that's a Doliolid nurse down on the right.
Is it just a
Maybe
bigger
not.
filigama? It's just a bigger filigama.
We've seen so many of these.
It's another Physonex down there. Really?
Coming down
Yeah.
a tiny bit and
Look at the pointy shape in the nectavours.
Look at it's the bracts. Yeah, it's a
Ooh, this
Fritillama.
is beautiful.
you're right.
A little bit.
You guys hear that?
I did hear that, yes.
Okay.
and pushing it.
Think
I'll be bringing this up in January.
Used is right again. It
Yeah,
must be
for
that
For your performance review.
Remember that one time Dougal said I was right?
Yes.
Okay.
Bonus points. Ooh. This
it's
is a
got to be
great
because of
shot.
all that training playing Fathomverse.
Yeah.
That must be why, right?
Yeah.
There we go. Thank you very much.
Thank you.
People in the
Next.
chat think you're cute.
Focus.
I think Filigalma's cute.
Oh yeah.
I don't think they think
There's
I'm
some
cute.
ambiguity in the chat about who or what is cute.
Who or what is cute? I don't know if cute
Moving?
would have been. Yes, thank you very much.
Next,
Cute would have been the
please.
first word to describe Filigalma,
Want me to
but
go see what's
definitely
down
beautiful.
Yeah.
in front there.
Elegant. I would
It's
say
probably another siphonophore.
Okay.
Frilligalma.
Yeah, we might as well look at it really quick.
I think the
Frilligalma.
cute thing was the cyclothone, actually.
Oh,
And then there's actually a larvation.
Oh, it's a
What?
Rudgicovia.
Let's go to the larvation first. Sorry, Dougal.
You smell that?
Reggie Coco
Is-
It's a
few of them.
Physonectae.
Okay.
So this is an oikoplora.
RUDJAKOVIA. RUDJAKOVIA.
We should actually. I didn't ask Russ. Does he want more of these?
Oikos?
Yeah.
Regular oikos?
Yes.
I think he wants a Nobi oiko.
Knobby.
I'll go ask him.
Or can you zoom in a little bit more on the house?
But this one does have the.
If they're
bulbous
knob,
shape,
if it
right?
does have knobby,
Yeah, I
a
think so.
knobby house,
Right.
then that is definitely something he would like to collect.
Should we just scan it while we wait on an answer?
We could, yeah. It is knobby. Okay, so if we can get
Yeah, they don't
some
stand
really
out as
good
much. I think,
focus
as the
on
low baits
this.
do within the ctenophores. I think
yeah,
of it
it's
as
right
when you look at the
I think
oikoplorids
we've-
and the Filigalma, they have
We've
kind of this
loosely
horseshoe shape at the
identified
house, and the
these
Filigalma just
with
has more like
kind of the knobs that you see on the
lowy,
front of
yeah.
that mucus structure is meso-oikoplora. You
See
can
the bumpies?
see they almost look like digits to the left.
We will take any of them because they are so poorly known.
So if we can, let's just
So good work.
let's just collect it because it's really small. If we can, I just want to make sure we don't lose the animal.
It was a.
Where's this one going?
D-sampler, please. And we'll want to keep eagle eyes out for the animal because we want to make sure it's still in the house.
Oh.
Did you see the Cyclothymia obscura?
Keeping an eye
No.
on it.
Yep.
Really?
Are we going for collection or are we trying to scan?
Gonna go.
We're going to do a collection, Yost.
Sorry, I can't hear.
Collection.
Okay.
I'm gonna do number 5.
That's right.
Wow.
Oh,
totally black.
To the left with a little bit of it. Don't need to.
Rosaria!
Wow. Go ahead. Go ahead. Wow, totally black.
All four, huh?
You ready?
Where did you see that fish?
I think it's still way
That was
forward.
10 minutes ago.
About half an hour ago.
Oh. I just missed it.
Just
Me too.
before you cycled on, I think.
I'm gonna have to, yeah. I think it's in. There it is.
Oh, it is, yeah. Oh,
Coming down.
it's so small. I don't know if I can see the animal.
Yeah, down.
Coming down. Yep.
This one is different from everything you collect so far.
Yeah.
See it there.
You
Does
get it?
can anyone see the animal?
We got, it had no photophores at all,
Alert? Or
so we think it's
Oikoplora.
Cyclothymia
That
obscura,
thing.
There.
because that's the one without photophores, right?
Uh-huh.
There. Coming over. Coming over.
What do you get so far?
Coming over. There.
So far, we only have,
Coming up.
we have one cyclonophore and one medusa.
There. There. Nope, that's inside.
Is that?
We get one.
Can we back up just
Yep.
Item.
It is.
You sound so surprised, Sarah.
Let's,
Right there, it's right
no, it's out front there. I'm sure of it.
Can I point it out? Do you see it?
You see it in the jar?
No, I see
Or
it
is it
outside the door.
there?
Yeah, it's fine.
Yeah, it's out front.
So I would probably want to look at it really quick just to see if the animal's still in it. This is so small.
No, it came out like you can't even see it up top here once it gets to a certain point.
Yeah.
Anymore though.
Darn it.
Let's put you in, see if we can get a biz first.
Tomorrow is ice cream day.
Tomorrow is?
Okay.
Tomorrow is Sunday, therefore the ice
Sunday.
You have to just set up and scoop it.
Yeah. Yeah. All right. They're hard. That's a Keating-nath. Keating-nath. I know, but no one here is like excited about them.
No, it's like every time I think it's a fish, it's a cute snap.
Right? All right. Back on the search.
Connie, question in the chat. We always mention time constraints. Why are there time constraints? Who decides? What decides? Can't you just stay down?
Where does that? Well, as you can imagine, there's a lot of simultaneous things that are happening on the ship. Not just for the science party, but for everyone else. Like the ROV pilots have their own kind of schedule where 2 pilots at a time are, or how many? 3 pilots at a time are on?
4.
Or 4? 4 pilots at a, okay. They have a watch that's to the right.
Just
6
so we can
hours
also
apiece.
say, oh, a little red one.
That is a shrimp of some kind.
It is a decapod. Type.
Coming up, coming up, coming up. And...
But we're pretty fortunate here in that we have 24-hour ROV ops. So if we wanted to be diving the ROV the whole time, we could. However, that's not the only thing we're doing as part of the science on board. We're doing ROV ops. We're also doing net tows. And then once those net tows and vehicles is back on, we are also doing a lot of lab experiments with the animals that we've collected. So these are things that are happening around the clock. And we've had some challenges with some of our net tows, and so it's been great to have these ROV dives with collection containers so that, you know, the breaks that we take for the vehicle is time for us to, you know, look at the animals, process them, etc. So there's a lot going on besides just the ROV dives. You just want a quick ID on this, right?
Yeah.
It's gonna be Philogama again.
Probably for Legama.
Everyone seems so excited now, but it's for the
Well,
So
it could be an
excited.
empty agama.
That's a good. That's a good.
What did he say?
Empty agama.
But
All right. We want something different. No frills things.
well, it is.
At least this one's
it's
swimming.
a cost-saving effort.
That's a frill.
Frillagalma.
I said
Yes.
Phytonecte frill agalma.
Bisonect siphonophore in fathom verse.
Okay. F-R-I-L-L.
Frillagalma.
AG?
We're still going.
F-R-I-L-L.
All right.
Oh,
Next.
The knife.
Okay,
okay.
Next.
Frill.
Frill.
Yeah, as in frilly. Because when they first described it, the nectaphore was all shrunken in formalin, and the ridges
There's a
looked like they
small
were
jelly.
frills.
I'm not going to find it out.
And it had the same kind of ridge pattern.
Oh,
as
then we have
what
to
they
change the
call
name
agalma.
because it's no longer accurate.
Yeah.
No longer an
If
agalma.
it's well preserved or it's a nice condition, straight after capture, there are no frills.
Okay, I'm gonna get some caffeine and a little bit of sugar just to make me
To the right, what is that?
unbearable.
We look forward to it.
And then hang out on
Yeah.
the bridge?
They used to lock the sugar in my house when I was little.
I mean,
Oh.
Oh, you have the fancy one.
Yeah, there's a
That's
few
why
in
it was empty there.
the drawer upstairs.
I still need the caffeine though.
Is that it? Was that what it was?
Still here?
It looks so
Eating
big.
now.
That was shocking.
That is
I hope
is
you
a
enjoyed that
female,
Keating nap.
Rabid female Keating nap.
oh, what female kidnap?
We thought that was so.
Risaria.
That's hilarious.
What?
It went off the bottom right.
It's just in Russ likes cute. Russ likes it.
Do you want us to collect a Keating nap?
I don't think so. Unless we get like
The
really
silence is
good
deafening.
video and you can see the luminescent organs and everything.
Oh, where are the stickers coming from?
The ones that say Mbari? You got 3 guesses.
The ones that say Amber.
Am I
From
signed out
Jesse.
for them or?
No.
No. Okay.
So I have to get them all right? Or
There's another
just one?
shrimp?
Like
Right in the middle?
Yeah.
Oh, it's
That's
a shrimp.
Yeah.
Is
that a
There's
house?
something down
That's a
below.
glare point right there.
No, it's something.
It's moving.
It looks like a lobate or a house.
There's a couple. Because the
Need
What depth are we at?
There we go. Oh, it's a Sudusa.
1103.
Fill out on the right.
PSEUD.
That's another Frilligama on the right.
Which way am I going?
Just focus on this one. Straight ahead.
You're saying it's not SUDUSA like it could have been?
Yeah.
Well, not if you pay attention to
Have
Latin and Greek.
we also have we collected more than just that one small one? Was there a bigger one of these collected?
It's PSEUD.
Yes. I think we did collect another one yesterday too.
USA.
Did we collect
USA?
Did we
So
collect
like
another
there's
one?
no
Sudusa.
O.
Ah,
What are we saying
The
this
big
is?
one in
The
ethanol.
Sedusa.
Sedusa?
Yeah.
And the other one.
Doliolid.
Sedusa.
A very small clip of tissue off the tiny one they asked.
The
me to do.
doliolid that looks like
So
So there's
there
another
may
a
not be
medusa.
quite easily. So the question is, do we want?
Would you like to catch another?
Oh, for sequencing.
Yeah.
So
Okay, no.
one animal's gone to sequencing.
Yes. I put one in a two.
Cool.
It was
Thank
like
you.
this
That's a
with a
nice
small one
shot.
and a
You
big
can
one,
see
and I
the
took the big
muscle
one.
bands
It was pretty useless.
around its body.
It'd be good for isotope.
And when they
You would like
contract, it looks like the body elongates a little bit.
So we catch it.
We do more good than
Sedusa. Are we wanting to collect this or no?
Well, we seem to have one, at least one that's been probably successfully sequenced. I put a small clip on another animal that may or may not be enough material to sequence.
So
If you aren't finding anything, I guess
We
if
should.
it's
Yeah,
unique
we
enough, would be nice to have a second
we
one to do as a voucher.
I think we should.
Do you want it to be in a section sampler or?
Oh, this can be suction.
Okay, so if we can.
These are pretty robust.
If we can collect that, that would be great. These great shots.
And you've got this as a scan already, right?
I'm pretty sure we did.
Okay.
You got that, hopefully you got that nice big one that we had
I'm
earlier.
pretty
in
sure
the
we
cruise.
did.
I'll be back with a call shortly. And more jokes, but not necessarily any better ones.
Wow. All right. So if we've gotten some really good shots on that. If we could put that in the suction sampler. Yeah.
Got the baby on his head.
Yeah. Any other fun facts about Sedusa we can share? 50%.
Apart from it being carnivorous doliolid and that it's got its little baby sitting on its head there, so there's the adult with the baby on the head.
Like a hat.
Some tunicates can have jellyfish polyps growing in the atrial siphon, so when they suck water in to filter it and take their own food, the jellyfish polyp grabs the particles that are coming in to eat. So it's like one of the few substrates in midwater. We haven't found anything like that in Sudusa yet, but that's not to say that it wouldn't make a good substrate for something else to live on. I'm surprised also I've never seen a hybrid amphipod or anything on it. Yeah, maybe they are carnivorous. Maybe they've got
maybe
some
they
way
eat them
to
too,
get
like-
them from
They
the outside of the bell to the inside and eat it.
secrete some sort of toxin
Are we
that
in?
neutralizes them. I don't know.
So suction sampler 3, sampled suction sampler 3.
Yep. That's in our section sampler.
Here he is. Oh.
Slow that
Shoot up.
down. Alright, but then, so if we can index it, please.
We'll have to try and do more microbursts with the suction.
Yeah,
Once
I
it's in
have
the
it
tube.
then. It's like 15% off. 15-20% off.
Next.
Oh, up high? Hopefully
Oi,
that's
pessoal da
not
Argentina,
another
oi, pessoal do
Filogoma.
Brasil. Quem tá agora aqui também é o Marcelo
It
Melo.
is.
Another Filogoma.
It's another one. All right, let's find
Eu sou professor
another.
de oceanografia na USP. E vou acompanhar vocês aqui
I wouldn't try. Just go around.
junto com a equipe que está a bordo
What is that behind it?
Another Medusa, is it?
navio de pesquisa
Maybe.
Falkor.
It's hard to tell.
Yes. It is hard to tell. Let's take a look.
Ooh.
Oh,
I
no,
think
it's
those
not.
those are tentacles.
It's a little repellonomatid in its feeding position.
Nice.
Trachymedusa. Wow, they don't go out straight. They kind of curve,
I know it looks like a giant
curve
bell.
around.
Trachymedusa.
Yeah, repellonomatid trachymedusa. One of the many, many Octopodema species. Yeah.
Octopodema genus. Wow, that's quite the
It looks like a bear.
posture of all of those tentacles. From
Arctic.
afar,
Arctic.
it looked like a much bigger jelly.
Is that the most specific you're gonna go, or is there trying to be something else?
Octopodema.
Yes.
It's a trachymedusa.
What would you like to do with this?
I think it's way too small
Okay.
to catch. We want to try and get a big one with gonads.
Ooh. If we can get just some nice views of it. It is so small.
Get a good zoom in.
Yeah.
There he goes.
Of course.
Right
Okay.
into the camera.
Yep.
And in the back is another smaller
Yep.
What's
That's another
Octopodema mean?
Filogoma.
Octopodema.
Yeah, what's the arctos? Because arctos is Greek for bear. Like Arctic Circle, the circle with
Right.
the bears.
Yeah, yeah.
I don't know.
Antarctic.
No
circle.
idea where it went.
Circle with the anti-bears.
Oh. Yeah. Yeah, just a nice shot.
Arctopodema.
ARCTA.
Yes.
POD.
Oh.
Yeah, it's the same one. Yeah.
Straight into
Yeah,
the
man.
lens.
Of course.
ARCT.
I mean, I think that was a nice.
A. Okay, got it.
Ooh. Oh, if we can get the focus on that, that would be a great shot.
RCTAPODEMA.
Oh, it's got gonads.
So
This one's got
now,
gonads.
you want to collect
Now
it.
I want it. Yeah, now I want it.
Section sampler, please.
Cut it as soon as it goes in. And
Essa é uma medusa Arctopodema e ela tem gônadas, então a gente vai coletar com o equipamento de sucção. Ela está com as gônadas desenvolvidas, né? Why you need the gonads?
Around the stomach.
Yeah, why?
Because they're species specific. The shape
Oh,
of the
the
gonad
shape.
and arrangement of the
It's too
gonad.
small. I can see it still on dark. So I can't. Move straight ahead. It's a good height.
Renata's telling me that this one might be a new species.
Yes, yes,
I
yes.
was going to post it there, right? I don't post
No,
it.
it's to the right and back. I decided to swim right at that moment. I think it's.
Okay. Hello. Oh, there it is.
Oh, yeah. Good eye.
Oh,
Is this Russ's entry music? Russ, do you want to sit here?
Huh?
Do you want to sit here, Russ?
Oh, yeah, I'm happy to sit there if you want to break. Otherwise,
Are you after us?
no, I think I'm on the next dive, actually.
Are you? Okay.
Sorry, just stumbled across it.
Maybe after we collect
Music
this.
about Saturday night, so.
Oh.
oh, that's actually the Octopodema.
There it goes.
It's in.
All right, in.
Yay.
Off and just
My
moving
first-
forward.
We just collected an Octopodema. It's gonna be time.
Johnny, we actually have pre-roll on this recorder.
We do?
Yeah, so that's 30 seconds.
Do we need a 30-second buffer? That's a lot.
Well, that's the only one. So
Ah.
yeah.
Okay.
Oh, what was that? Which camera was that?
That's not. There you go. Do you think we need it? Or microburst?
Oh, yeah.
I noticed
funny
you put,
that-
I noticed you put that on the list of improvements, so.
We could use it on the way up and on the way down if we see something just shooting by.
Oh, there it is.
Nice.
Turn around.
Well done.
Sample 4, good job.
There's a very small jelly down below.
Tiny
Should I
little
say that out
fast swimming
loud?
jelly that changes direction all the time.
No. Okay.
Did you annotate that it was in substance sample 4?
No. How do you go back here?
Go to press the cross on the top right. On the right hand side, not that cross. Go down a little bit. Down, 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 down.
Oh,
Up.
sorry.
The cross, yeah.
Oh, yeah, okay.
And then
How
over
we
here, maybe.
created.
Where did you capture the Octopodema photo?
There.
Here. So try here. Click, click. A for annotate. And click, click.
Created.
Yeah.
Yep. So that's click, click.
I
And then
think we've
most
taken a look at a
recently
number of these
annotated. Oh,
already.
you'll have to re-click it again.
Holly created.
Yep, that's Conica. Halascara Conica.
Halascara.
And then go
Conica.
to most
Alright.
recent, where it says
I
Octopodema.
think
Yeah.
we should
Yeah,
keep
you got
going.
Okay.
that? And then comment, click, click. And comment. S. Press S.
Husband is
S.
S?
S.
Huh.
And then these will
mean,
come up.
Sample.
weren't
Something
yelling
sample
and
4 it was.
I'm just gonna assume.
You can just click on it down here.
All right.
And then save.
Okay.
Okay.
What does everybody in the chat want to see on this dive? We had some pretty awesome sightings yesterday. I think it's gonna be pretty hard to top.
I'm here to change them.
I know. Do you
Yeah.
want to sit here? Do you want to sit here?
You want me to take the chair? Or you want me to stay here?
No. Do you want to sit at this chair?
Either way.
Yes.
Everyone's trying to give away this.
I'm sure you have some jokes you would like to
Share with the
Share
greater
with-
community.
the live stream.
Oh. No, I'm saving those for my ship tonight.
Are you sure?
Yeah, I'm like doing up the whole routine.
All right. All right.
Because it's going to be live from Falkor. It's Saturday night.
Oh, center left, center right.
He's so ready.
At the right,
Jelly.
going down.
Down,
off
down,
the
yeah.
screen.
That's a jelly,
The
but
orange dude.
Going down.
There he is. Is that a Botronema?
it's got a bit of a.
Oh, it might be our second one of the cruise. Looked like a
Coming up and forward.
the bottle you give milk to babies with. It looks like petite from one of those, perhaps.
It's a very weirdly shaped piece.
Botronema. B-O is a tracking medusa. B-O-T-R-Y.
Botronema.
R-Y-N-E-M-A.
Ooh. That's a nice
Awesome.
shot. Thank you.
Boat tree?
B-O-T-R.
Yeah, why?
Y-N-E-M-A.
Oh, okay, good.
They do look like little suction
Wow.
cups. This is a great
With phenol on the
shot.
top, yeah. And the top of the radial canal is very thin. I think we've written enough papers about Botronema though, right? We should move on to something else.
Yeah.
Thank you. Let's
Okay.
move on to the next
We actually have a
one.
3D model of one already too.
Yeah. Thank you. So we're still at a
That's probably a big species.
Or 1,100 meters ish.
Yes.
How many more hours do we have?
We are supposed to be.
So another 4 hours?
4 more hours.
Rhizarian, nano?
Why don't we start coming up a little bit?
Doesn't say clearly. Let's see. Start 1 to 2. I think we're supposed to be on a 6-hour dive.
That means 7 hours. It's a dive until 9 or 2100 it says.
Dive until you drop.
And then 22 we should back.
Bottom left? Oh, it's
It's just a
okay,
key.
point, yeah.
Yeah.
We're still going.
It's coming back.
Sorry, what's that one down there? To the left? Perhaps a little too late. It's okay. Too late, too late. We can look away.
Rhizaria?
Okay.
Yeah, I don't know what that is. Detritus.
Okay.
Probably of some type.
Yeah, we're on until 2100.
On deck at 9 o'clock.
Yeah. Forward.
So Nira, do you want to answer a few questions for people, or what were you supposed to be doing on this cruise?
Oh, yeah. So I was supposed to be.
You might have to talk a little bit.
Here, Nira.
Okay.
This is you.
We're going to hear from Nira,
Oh,
an
oh,
engineer
okay.
that has joined us on this cruise.
That's a Dolly Olive, right?
The one in the front?
Whoa.
Which way am I going?
Yes,
So
this
this guy,
one,
we're looking
this
at this
one.
guy.
This one. It's too late.
That's just not, the totally not great.
Nothing
Well,
in
I'm
it.
using the technical term. It's actually an acronym. Small,
Nondescript.
nondescript
Object.
object.
Alright.
Transparent.
Keep going.
Yes, we'll keep going.
We both were like, no, wait, no, right there. Something.
So yeah, you'll notice on this cruise we've been largely targeting organisms that are relatively small in terms of sampling physical species. And that is largely because we are limited by the fact that the suction sampler can only suction up whole organisms of certain sizes. And well, the pressure sampler.
Down a little more,
But
but
our initial
depending on
plans
yeah.
involved attaching and bringing along a biopsy sampler that would be able to collect a small, non-invasive piece of tissue from larger organisms so that without hurting or killing them, we'd be able to collect pieces of tissue from these larger organisms and do DNA sequencing on them later on. So I was initially working on that project, and we were hoping to pull it, but due to a few shipping issues, unfortunately, this robot did not make it on. But it'll be something I'd like to deploy in future cruises. I'm really excited to keep working on this project for. Yeah.
And is this your first time on a big
Yes,
research
this is
vessel
my
like
first time on a big research vessel. I am a second-year PhD student, closely from Harvard University, and our biopsy sampler was working in collaboration with a few other researchers, one of whom's on this cruise as well, Katie, from the University of Rhode Island, where she was working on a much larger sampler called the RAD sampler, and fitting it with a really cool laser alignment system that be working in conjunction with our sampling mechanism in order to align organisms within a larger
Straight ahead.
sampler, such
Is
that
that,
when the lasers align properly, we'd be able to
I can't tell.
launch our biopsy tool in order to collect tissue rapid fire in order to do minimal damage to the organisms. Here we have, is that a Floram rhizarium?
Rhizarium.
So zoom in and take a bit of the first picture. I think it's dead and it's just
Detritus?
bits of marines.
That was formerly a rhizarium? All right. Yeah, you can see the spines. All right. Do you want to say some things about the RAD sampler?
Yeah,
And that, what does that mean?
The RAD sampler is a collection device that has been used on previous cruises.
Yeah? Okay.
And it is a very large dodecahedron shaped sampler, which some might be better colloquially known as like a soccer ball shape, or close to soccer ball shape, but just 10 different pentagons lined up, or is it 12? No, 12 pentagons. And the idea is that it's a rotary actuated dodecahedron. In other words, its net folds flat, and attached to one of its faces is a singular motor, and through some very smart linkaging systems, by rotating this one motor, the whole thing closes from a flattened net into an entirely closed dodecahedron near spherical shape, which makes it work very well underwater. And due to the materials used in all of its locking devices, it's very gentle. So it closes very delicately around organisms such as jellyfish and other gelatinous zooplankton in order to enclose them in a smaller environment for sampling, imaging, in our case, taking small biopsy tissue samples without harming the organism during the catch. And then after our images and tissue are all collected, we can release the organism back out without harming it during that enclosing process in order to have it not escape before we can do all of our scientific analysis on it. Yeah.
Yeah, unfortunately, a lot of equipment from University of Rhode Island, Harvard, Bigelow did not make it onto the ship because of some challenges. We'll just leave it at that. But hopefully we'll have another opportunity. Bottom left. I don't know what it.
Yeah, something yellow.
Yeah, it's not that, but let's keep looking. Let's keep
This was
looking.
the collapsed house.
A snot strand. A little more forward. Please. What has been the most surprising thing for you now that you've been out here for over a week?
Let's see. I think I didn't realize I knew that the midwater region was an underexplored area, and as someone who's coming in pretty new to the marine biology field, me as well as a postdoc that's from my lab, Elio, are largely roboticists in bio-inspired systems, but mostly terrestrial. This is our first time doing really big underwater dives and research, but just the actual abundance of organisms and the speed at which you can collect new species and identify them has been really exciting to me. I just didn't know that you could go underwater and in a few hours identify multiple completely new species, and that the encounter rate is so high. And I've been feeling really lucky to have witnessed a lot of this happening and just seeing so many interesting organisms just by sitting in this control room, and surrounded by a lot of people that know a lot about midwater.
I was going to add, I mean, you are surrounded by the world's experts in a lot of this taxonomy, so that makes a huge difference.
Biologists just being able to point at organisms and say, oh, that's a so-and-so. I think I'm learning a lot as well, but definitely really inspirational to see.
I'm just getting closer so I can hear. So I'm not trying to push us.
No, do you, I don't want to take away your thunder, but have you been on a Schmidt boat before? Falkor?
No, I've not been on one of the Schmidt vessels before. So what's the question?
How has it been different than some of the other research expeditions you've been
Oh,
on?
it's bigger.
The boat itself?
Yeah.
To the top left, please.
I mean, most of the university fleet vessels are below 100 meters. They're more like 80, the bigger ocean class. So it's definitely bigger. The only other research vessel of this kind of size that I've been on is Falkor, which is bigger, longer.
Okay.
I'm not sure I'd say it's bigger.
Did you see that?
Yeah,
Is that a larvation?
At first I thought it was something totally different, but yeah.
I thought it was a
must have been.
unicorn at first.
Oh,
Okay.
I thought it was a empty house that was slowly collapsing.
There's a location swimming in the top right.
Top right. And Russ, you've been involved in a lot of ROV operations too, right?
Yep.
What do you normally use those ROVs for?
Since I left Embar, what do I use them for, or what ROVs?
For
do I
your
use?
science. You could do both.
Oh, very similar to what we're doing here, just to use them to kind of survey the macro plankton diversity, because they don't collect well with nets, and you get a better impression because the ROV census is a much bigger
Can you
volume
stop?
of water than what you do with plankton nets.
Oh, it came back.
Oh, cyclonia? Okay. The one time.
Okay, zoom in, please.
Yeah, can we get a zoom on
The
that
chat
fish,
activity is
please?
pretty quiet today.
It is quiet. Ooh.
A bit more active than the last one.
So,
Hey.
where do we want to go, given the remaining time? I was starting to have
Well, we've been
Sarah move up.
Yeah, I mean, we haven't covered this depth zone much on many of the dives. We've been starting shallower, so I think, did we go to 1,000 last night? And then
I
start?
think we did.
So this is the important part to cover is is the zone we're just finishing off, maybe up to 8.
You want to collect it? Yeah. Marcelo would like to collect the fish.
All right, let's try in the suction sampler.
How's the host activity been down here? What?
I've seen a few,
Yeah.
very small ones though. I think we saw a Meso-Oikoplura.
The clubby.
Yeah, the clubby one, but
Yeah.
they're so small.
Yeah, none of these, I mean, you get spoiled at Mbari with Mesocordius and Bathycordius, right? In terms of a big animal.
Yeah, but
Um,
I think we're missing, I don't know, I think it was either Yost or Dougal saw a bigger one.
That doesn't
300
guarantee, yeah,
meters.
that doesn't guarantee they're
Here.
Bathycordius.
No, but
Yeah.
just a bigger one.
Mesocordius is also kind of underappreciated in its distribution.
Mhm.
So we've done at least one other warm water cruise where we saw Mesocordius and collapsed. That seemed more consistent with I think the host we saw the other day,
Yeah.
because there wasn't really much of an outer envelope associated with it. It's hard to tell how it collapsed.
Right. Well, and I think depending on the times of day, they might be easier targets to spot. Bruce has
water,
a,
it shouldn't matter.
but for if the bigger larvaceans are shallower,
If they were shallow, yeah.
Bruce has like the theory that their houses disappear because of vertical migrators.
Yep.
Find something?
Get the arrow.
Oh, you found another?
This ROV approach we're using really depends on you being
Yeah, right.
deep enough that surface light is no longer an issue. The water's so clear here that the upper 300-400 meters of the water really aren't dark enough
Yeah.
to use at midday, which is surprising. Like,
Yeah.
it's not surprising, but it's something I don't think we thought about.
No, not at
The
all.
implication of in terms of the design of trying to do day-night dives. It just, you can't do the day part.
It's there. You see it, right, Sunny? Boom.
Fish,
Same
fish, looks just like the same fish.
Right. It's interesting because we will see, Monterey Bay at 200 meters.
Yeah, that's
Generally
that's
is
the
super
sweet spot.
dark.
Yeah,
And then we would see the larger larvaceans anywhere from 50 meters to 400 meters. But our vehicles don't work a lot above 200 meters. So often we will miss some of the biggest ones, like Bathycordius mcnutti, named after Marcia McNutt, who is currently the president of the National Academy of
she
Sciences.
was, yeah, but she was president of Mbari at the time
At
she
the
was
time.
named.
Yeah, she's
The name
giving
was put for it.
she's giving a talk about the state of the science in a week and a half. I'd be really curious to listen to. But anyway, so it's possible, at least with what some people have said, they saw at least bigger larvaceans at around 320 meters. That might be places where we want to.
Marsh
Wanna
Youngblood
look.
used to get a lot of Bathycordius in the Florida Current, just off of Harbor Branch.
Okay.
And I'm trying to remember, I did a cruise with him back in 2020.
Okay.
No, 2000. Sorry. And I just can't remember. Yeah, it's only 20 years ago. I just put an extra 2 in there.
It happens.
I just can't remember how much deeper or shallower they were than Mbari. I'd have to go back and look for notes. I don't think anything ever quantitative got published from that cruise.
Okay.
But to me, the eye opener on that was that so the Harbor Branch had the big Johnson sea lynx, right? The big
Right.
sphere.
Right.
And I did one dive in the sphere, and for the stuff that we're often interested in, the sphere doesn't really work any better. It gives you a bigger field of view.
Right.
But you can only see super large stuff. And like I was looking at the larvaceans, and well, So what do? We got this big sphere, you're in the water, and then you get close and you have to look in the camera to get the magnification required to tell you anything.
Yeah. I'm not sure. Oh, yeah, it's still there. And that's a submersible. So actually, since you've been using lots of ROVs and you've also been in submersibles, what, maybe explain what those observational differences are as somebody who works on some of these appendicularians.
Well, I mean, yeah, for appendicularians, I don't think the submersible is the right technology. I think, well, you could maybe bathycordius would be the exception. You can find them over a bigger distance.
Yeah.
The details you're interested in require the magnification you get from a zoom lens. So what's the advantage in the extra cost and risk associated with being in a manned submersible?
Right.
If you could do exactly the same thing, letting the ROV do your eyes in the water.
Yeah.
Plus, you know, you've got a bathroom on the ship and you don't have one in the ROV. You've got all the snack food you could want on the ship.
Minor
and
detail.
you don't have, you've got
another
stuff in
side.
your pockets when you go in the submersible. It's a very kind of different comfort level.
That's a good point.
But I think there are some specific things, and particularly benthic work, where that being there makes a difference.
Oh, really? Oh, just to the starter. How so?
I think that there's some things that you kind of need to be able to kind of take in the whole landscape when you're on the benthic environment.
Okay.
Kind of appreciate it, even though it is limited by your light field. You get much more of a 3D impression
Okay.
of the environment. It's a much more heterogeneous environment, whereas here, the blue water here is going to look the same in an ROV as it does from a submersible, right?
Yeah. Look at went down there.
I was amazed though, like diving with a manned submersible, where you're always
crude
focused
submersible.
kind of a little bit away in the sweet spot of the light, looking for things, and you see, you kind of get an idea what the community is like, and then you kind of reset and look really close to the window at the really small transparent things, and there's all these little doliolids and other things that you weren't seeing at all, even with your eyes when they're focused further out. So with a camera, there's all these.
Yeah, the
probably
camera,
all
you're
these
waiting for the
tiny little-
camera optics to catch up, and your eyes are much more instantaneous and adaptable.
Right.
Yeah.
So there is that aspect that gets to it.
Yeah. I mean, I don't see the point of any one technology going away. I think both are really valuable.
We're just trying to catch this fish.
I have never been in a submersible.
I've only done the one.
Yeah.
Google, on the other hand, has spent more time in GemStex. Just 3 times?
I thought I said like 22.
23 times.
23 times Dougal has gone in a submersible. What's the deepest?
I think it's one reason that I can identify things, you know, with a range of different distances away, etc. Because I've seen them, like, so many times at, like, down to various conditions. But the way
Oh.
you see them with different ROV, even just with the different ROVs, they look different
Could you
depending
turn
on the
off
lighting,
the lock?
the camera,
I don't know, I'm not seeing it. I'm
and
just
with
like,
your eye, they look as they are.
before we were seeing
So
everything.
you've been trained on different ambient conditions.
Basically,
Much
yeah.
like you want to do with machine learning.
With machine
Yeah.
learning,
Yeah.
yeah, right. And I've seen them bash up in nets as well, so.
There you go.
Oh.
But there's definitely a loss of image fidelity and resolution with the cameras compared to what you get from your eyes.
Yeah.
What's that? Depending on your eyes.
And depending on your memory.
It's on.
Well, your eyes are these wonderfully constructed,
now.
you know,
Sensing devices.
high resolution, wide depth of field, quickly focusing, refocusing structures that the cameras are trying to emulate. well, they've got more zoom. They do. There are some downsides with the camera.
Yeah.
The good thing about the camera is you can, like, if you're on an ROV, because you're dependent on the camera, everything you're like trying to zoom and pan and tilt, etc. a lot more than when you're in a sub, when you can just pull the animal out, and sometimes you don't even bother facing the camera to it, so you can't go back and check and recheck.
Yeah, you've got no
Yeah.
record.
Just kidding.
Sunny is like, can we please just do DPIV instead?
I know.
Scan a bunch of cephalophores instead of trying to collect this fish that keeps swimming away.
That's what the problem is. You get back here and there's all the white noise from that dam, and it just kind of splits
There we
the
go.
room.
Okay.
Did you get him?
Yeah.
I think so. It looked like it.
Was it that small?
It was.
Oh yeah, it's small.
That was a lot of work.
Oh, it did. Oh, it went back up.
I think it was.
Yep,
Yeah,
it's
he did.
in. Oh, okay, okay. Lock it, lock it, lock it.
Let's index.
Maybe it's section sample
please.
5.
Yes. Section sample 5.
Okay.
Down.
No, you like nobody has written it down here. Okay, you have it.
All right.
Oh, I got you.
Moving
don't
on
worry.
from that cyclophony. Thank you so much, Sunny.
Oh my
that
God, it's been
was
30 minutes.
ridiculous.
It has been 30 minutes.
There is something straight ahead.
Pronounced different.
What's that?
Straight ahead into the
Try and get a.
Japanese translation is it just
Japanese is when you have that.
different enough.
Yeah. Oh, what's that?
I-
Silver reflective.
Yeah.
I'm gonna guess it's just
A fish.
A suggested is a good guess.
These are two parallel lines.
Like
Yeah.
two things together.
Could be a sedipid with bright white tentacles. hanging
Maybe
off the bottom.
that.
Is it the
Maybe
glare
that.
of the camera?
I think it was.
Yeah. Stop
Oh,
moving.
it's a thing. It's moving.
It is moving.
It's far, far away, swimming away.
Keep
from
going.
Maybe it's
Coming
another
closer
giant
again.
octopus.
Go up a bit more. Here we go. Here
Here
we
we
go.
go.
We're
Two
catching up.
fish.
Two cereboma next to each other. Oh, wow.
One fish, two
Hold on,
fish.
hold
Oh, they're
on.
mackerel. They're not cereboma.
Oh, whoa.
What is that? Please
It's,
back off.
cocktails. Oh! Oh my
Oh,
goodness!
Got them, got them.
Oh my goodness!
What?
It
What?
was, oh, gee, what do you call them? With the forward facing arms.
telescopic.
Gigantic.
Gigantosaurus,
Yeah.
Giganta.
Look down a little bit.
Oh, Tracy was right. He was catching them like
We
more
caught him. We
often
caught him in the
pairs
Gulf.
in the nets,
Whoa.
right? They do associate together.
So do they make?
That's
Holy
amazing.
mackerel!
Not mackerel.
Not.
Oh, give
Have I ever seen one of these?
The iris back.
The iris. A little more. A little more.
Reminding you of different
Fish for the
legs.
moment.
These are just
But
bouncing
it's
back.
the one with the telescopic eyes. Giganta.
Yeah.
Giganta.
What is that?
Guys in the chat.
Giganta.
Google it. We tried
I'm
Googling
gonna-
it.
We tried Googling it.
Doogling it isn't working
Yeah,
this
well,
time.
Dougal just failed. The Dougal.
I know.
Where did that?
Google's
go?
offline for
I
a
have a
second.
picture of it.
And they're in
I did.
pairs.
Oh, here
Okay,
it is.
so
I'm
what?
glad we went
Telescope
after
fish.
them.
Telescope fish
fish,
is
yeah.
the common name.
Okay, so what
And
is?
then
But
Gigantura, and
That's
there's
it.
a couple of different
Yeah.
species.
Gigantura.
Yep, I think you're right.
Were they
Gi-
moving like
amazing.
while
Yeah.
remaining vertical? Were they getting
Yeah.
further away from the ROV?
They must
yeah,
have.
yeah,
They
yeah.
must have
Yeah.
been. They must have
It was like they
been.
were just like.
Yeah,
It was
Sunny
like
says.
going like that, right?
Sunny says that they were moving away from the vehicle.
So somehow they managed to
Big
swim
guys.
in that position, and they are in pairs.
Yes,
I don't know if
that
I
to
think it's
me
maybe
was
that's
crazy that they're in pairs.
Yeah, I'm like. There's a paper. I think Tracy Sutton was at least on it,
Right.
if not the first
So, well,
author,
that's his other
where he'd
yeah.
analysed all
But I
the
think because
net
he was
toes
vertical in that
and
scene.
found that
They're in
doing
pairs.
the statistics, it's more likely that they always occur in pairs.
What? Well, there we go. We've
There
got the
we go.
visual observation.
I'd say it's like.
Nicely done.
Wow.
I think it was the forked fin maybe that they're able to do like swim sideways.
That's cool.
Because there's one lobe was very long.
I need to look. I need to look at that again because
They must have been moving away while vertical.
they
Yeah, they must have been, because they never changed. They were like vertical the whole time. To go back and analyze that video.
I can say I'm gonna get the timestamp on the video.
Timestamp
I got it.
is 2027-18.
Thank you.
Wow.
Yes.
GIG and
Yeah, I got it. So isn't the time black?
That was incredible.
Yeah, this is
Yeah,
the time of the video.
yeah.
So you can look it up quickly.
Okay.
Oh my goodness.
It's a
That,
damn-
that's like up there with the halifrog.
It is. It is for the fishy world. Absolutely. Anglerfish, telescope fish,
Yeah, it's a Turidae, it's a whole family.
family. Yeah, there's a couple of species.
I've seen one fleetingly as it went in the distance in an ROV dive like 20 years ago.
And we
so
made-
it's not a
Wow.
range extension, but it's just rare.
Well,
It's just rare. I think rare.
seeing two together like
That is rare.
I don't.
It had been supposed by doing statistics on how often an even number of animals were caught in net catches that maybe they occurred in pairs because you almost never got 1 or 3 or 5 in a net. And but that's only like maybe because the net's towing through such a large volume. Here we see they're right next to each other in coordinated swimming. Like they were swimming away from us at a rapid pace while being vertical, and they were
Together.
still the same distance as they were when
From
we
each
first
other.
came
Right.
across them.
Basically
Right?
maintaining formation.
Yeah.
Right. Because right, we were still getting the sense that it was.
Odd that he noticed they were only found in pairs.
But
I mean, not doesn't quite hit the same, but
no.
we'll get an observation and
Oh,
move.
yeah.
Of course we should.
Foram.
Zoom in.
They all look the
Good
same.
zooms.
This is his little forearm.
Wow.
They're in pairs too.
What is it? Heliglobe.
Alisterella.
Are all forams rhizarians?
Oh, there we go.
All forams are rhizarian, but all rhizarian are not forams.
That's it. That's what I wanted to
Oh,
say.
there you go. Manu popping in quick.
But
Really special.
oh, look at the. This is the secondary spines. Oh, this has the bubbles.
What do you mean?
This has the gas bubbles. How are we doing on the samplers? We've got.
We have two buckets filled and two couplers
and the
are
copper pod
empty.
is being eaten alive right there.
As long as you guys haven't done anything.
Oh,
Oh, did you guys have put things
Another
in?
one right there. You
Well,
can
no
see
one's
the
totally me
tail.
up here. We need it up here too. Stand by, let me close this to make sure I don't spill
Is
it.
it a Keating name?
It's
Could be a Keating
or a
name being
Larvacean.
eaten. Oh,
it
no. It has its own coat. Actually, another rhizarian. You see the circle around it?
Yeah. I could see it being a folded up larvation. Yeah, larvation.
Oh, yeah, you can see the
Larvation.
tail. Yeah. Oh, the larvation is gonna die
So the
today.
and the thing around it.
Bye-bye
We
larvation.
should catch this.
So if we collect it, we get
should
both.
collect it. We
Yeah,
get both.
both. Yeah,
Okay,
there you
good
go.
job,
That's a
Russ.
good job.
Good
Be
job.
very efficient sampling.
Are you wanting it in a
We gotta do it in
in
the
a
sample.
D sampler, right?
In the
Yep.
freeze.
If you guys could go for D1, if that works for you guys, pilots, that'd be great.
But I'm gonna have it. Let
That
it
makes the
eat.
most
We collected a
Not- I
cyclotron?
wanna see it.
Why?
I've been saying.
Excellent.
Of course.
Actually, the larvacean is separating from it right now.
Is this why we haven't been seeing enough of the larvations we want?
Yeah. That would make a lot. Yeah. I mean, I think I've seen
No, I like it
them
even
eat
less.
all kinds of things, but larvation would be the first.
Here it comes, Manu. See how it goes.
Gone now. I think it was
No, the larvation is attached right there.
Yeah.
Yeah, right there.
Yep,
That
at the end.
little.
Whichever one you guys want.
Which one you
Pilots.
want?
Pilots, whichever you want.
Which one you want to?
It's still attached.
so that's
it's
two.
super sticky. Like when I'm handling them, they are very, very sticky. And the snot doesn't help. I guess it probably is the other way
Well,
around.
it actually probably helps the animal not be digested right away, but I'm surprised the animal hasn't bailed out.
Oh, it could leave the house? I had a
Yeah, it could
feeling
leave
that
the
it's
house behind.
actually attached. The animal is attached. Yeah, it's still there.
Out to the right.
Big shame we didn't
Oh,
look at the head of the.
Oh. Yeah.
Hard, so hard.
If we got viewers in
Uh-huh.
here, we could all.
Everybody hold your breath.
You said beyond 4 a.m. What was this
This
Manu?
is Hestigerella
Hestigerella.
flagita.
H-A-S-T-I.
Outside.
H-A-S-T.
Oh, it's
Oh,
in front of
it's
the
in, it's
suction
in.
sampler.
No, it went on the
Oh,
outside.
It's in front of the suction
Sorry.
sampler. Right down to the right here. It's right there.
I thought it was filigama.
Down.
No, not filigama.
This one down. It's more in the center
Oh.
now.
Oh, you're looking at that other
Going
thing.
down.
Oh, yeah.
Yeah, I think those approaches to test it.
Yeah.
There it is right there.
Hold on. I mean, we had.
Oh, is that the fuzzy stuff?
Yeah.
I thought it was just an artifact of the imaging.
Like the octopus eating the jelly.
Yeah.
It's like
You saw the jellyfish too?
I did.
There was something getting in the way repeatedly.
It's getting in the way. You couldn't see the whole jelly.
And what's the Hestigerella eating right now, you said?
It's eating
A larvation.
a larvation, which I cannot tell what
It's
it is.
trying to eat it.
Nope. We're close.
The larvation is
Why can we not tell?
Manu, do you want to explain what's being collected right now?
Yes, we are collecting a foraminifera.
Give me the mic, sorry.
So most of you can see this shimmering star. Right in the center of it is the nucleus. This is a single cell that is eating an animal.
Oh.
These are some of the largest protists. Oh, and we just destroyed it into
No,
pieces.
I think he's in the front still.
I think
He's okay.
the core is there. But these are called forams or
There it is, up
foraminifera.
front. You got him.
And oh, now you can see it's shed its
It did.
secondary
Yep.
tentacles, but the primary tentacles are still there. But what was exciting was it was possibly eating a larvation, which is an animal. So many a times these protists get so large that they're capable of feeding on animals, including copepods. So now you can see the larvation survived. Forams are very exciting because they form the fossil
Nope.
record on the planet, and a lot that we know about different eras come from these fossil records.
Yeah, he is.
So if you ever have some sedimentary rocks around your house, you can actually dissolve and find a lot of foram fossils in them.
Oh.
That was close. That was close.
Just slow. Oh, wait.
It's interesting that there is something still
Oh.
stuck here.
Yeah, there is a little something still stuck on one of those
Yeah.
tentacles. All is not lost, Manu.
Yeah, no. I'm saying even in the front of the sampler.
Oh, yeah.
Yeah.
He's close.
I think the nice thing was that this has the gas bubbles on it. They're not gas, they're lipid. And those lipid bubbles help it stay buoyant.
Oh, there it is.
Over on the left.
The fish wasn't that bad. And fish can
Yeah.
run away.
Oh, there's.
Oh, there's another one.
Yeah, maybe we catch the big one. Maybe we catch
You want the big one or the small one?
No, let's catch the big one. Oh.
Well, no. I don't think you want the big one who's also small.
Yeah, we're both small now.
Either small one is fine at this point.
This is very painful to watch.
Manu, close your eyes, my friend.
No, we will get it. We will get it.
I don't see him. Oh, up over here.
So is this a good fish snack, or are these eating fish?
It would be a good fish snack, except it would be very spiky. Oh.
Wasn't that a large copepod?
As you can see that they don't have any propulsion. So
Just
very
Here
sensitive
it is.
to the water flow.
Getting away, so
Yeah, good at getting
Just
away.
above. No.
And
movement.
you can see this
Oh,
as
come on,
drag
buddy.
lines
Come on, buddy.
that are. Oh.
Nope. Nope. Nope.
Zoom out a little bit. Was in front.
Yeah, 100%. He's behind us. Yeah,
Yeah.
it's back in here. So there he is.
Good practice.
So I don't know how many of you know this, that spiders can fly by shooting what is called a line, and that gets dragged in the air, and this is how spiders fly. You can see the same principle apply here when it has all of these silicaceous tentacles. It's very good in entraining in the fluid. So it behaves like all the advecting particles around it, which makes it very hard to catch.
What's on board?
Nope.
It could go in
You have
the
to
other
figure
one
on.
if it wants.
Do you have to figure on? Nope. He's still behind. Yeah.
Whichever one you want to try. Jump.
This wasn't bad at all.
Come on, buddy.
These speech fans, we got a lot of behavior out of the...
Yeah, and you had good video with that larvation.
Yeah, we know at least now they at least attempt to eat larvation.
Mm-hmm.
But you know, that would be a food for a couple years. If you look at the metabolic costs, there's plenty
So
to
the
go.
larvation will probably decay before this.
Possibly
Totally,
the bacteria.
yeah.
Yeah, the bacteria is gonna get in there as well. But we don't, I think
He's
every
outside.
time I've seen
Just
they
outside.
encapsulated in a membrane. Yeah, it's not clear whether it would let a bacteria get to it or not. So we've been able to keep this alive in the lab for a couple of days now.
Nope. In front.
Oh.
Oh.
It's tricky.
Oh. Oh.
Just so close. Just so close.
It's all right. It's all good.
Another really fascinating thing about rhizarians in general is many of them form this silicaceous silica shell, and that's roughly 20 to 30% of all of the silica cycle in the ocean. So they bring a significant portion of silica, which is critical for the geochemistry of the ocean down.
Yeah.
Yes.
Yes. Thank you for sticking with that. Thank you. Okay, so that was in deep sampler.
Get the flux, and then bring it up.
We got him in there.
Push up.
Yeah, because it's a passive
D2,
particle.
right?
Yes,
We're trying to get
D2.
the volume of fluid. Great job, pilots.
Nicely done.
You see that it was eating. Okay, it was eating the larvation, and it got flushed away. Yeah.
That's cool.
Very cool.
I mean, I don't know if they shut it on purpose, but
See,
it
it's not
What?
like well.
can, right? Like that outer.
I think that's all too much. Like when if I calculate the turbulence numbers here from this is 100 times higher than what it's.
Nicely done.
I don't think it would shed that purposefully.
Yep.
I mean,
Back to scanning, slow
yeah,
up,
it
right?
grows
That's what we were in the middle of.
There is a purpose to it. There is a stiffer
Perfect.
rod in the. I think the stiffer.
Oh yeah, it has a tubular eye.
You want to
That's
get
amazing.
in the first.
For him?
It's one of the few fish that has tubular
They're
eyes.
everywhere.
So anybody that's on
All right. Thank you, guys. We're going to continue on slowly searching around, slowly ascending. We're inside 1,000 meters now, moving our way up. Yay.
It was actually moving pretty fast.
Middle,
Filigama, we can move past it.
then we are going to move past
What's the
it.
simple number?
D2 was the rhizarian. That's okay. He'll be found. He'll find it.
Can we maybe go a little more forwards too, or point the camera further up?
Yeah, forward and zoom out as far as possible would be great. I think they are pretty out. Yeah, they are pretty far. Yep. They're as far as they can go. Middle.
What photo there?
Oh. We can move on. Rosarian.
Rosarian.
It's the same guy we just collected.
They're everywhere.
They are. It's a very rhizarian rich area.
Spiky fish food.
Uh-huh. Stop. Thanks. See if we can get it. I don't know if we can. Just might be a cyclothony or something. Oh, shrimpy. Let's get a zoom on him pretty quick for a sec while he is not moving. He's a little different.
Looks like one we collected.
It does. Russ, are you in here? No, Russ isn't in here, is he? Yeah, that would be great if we could get that zoom for a second. That'd be great. This guy's a little different.
Not particularly active.
No,
Is he okay?
I think so. I think he's taking a nap.
Hang out here in midwater for a little bit.
That's great. Great zoom, you guys. Oh, it's a... Is it a mysid?
Just woken up.
I think the mysids are the ones that have those swimming legs on the back only. Not sure on that, though. Great. Thank you, guys.
About to see something epic again.
You never know.
Yeah, we
You
are.
never know. Could be anything.
Some cool fish.
Mucus or something right there. Super close.
We did dragonfish. I haven't seen any.
What orientation do you expect it to be?
I don't know.
Horizontal? Am I looking for a horizontal stripe?
I have
Vertical
no
stripe?
idea.
Mm-mm. Oh, fishy fish.
I have no idea.
Middle. Oh, and there, yeah. I think that's going to be
Look at some
another.
anglerfish, they swim
Take a look
upside
real
down.
quick.
Ooh.
What do we got? Oh, it's a little oikopleurid, yeah? This little guy?
Seems almost too round.
Yeah. I think he might be looking at us. Not, you know what I mean? Not sideways.
Oh, that's a zoom.
Thanks for the zoom.
Yeah,
It's tiny.
Yeah, I don't see anybody home.
No, I don't either.
Turning mid zone.
Okay. All right.
Should we pass it by?
Yeah, I'm just making sure we get a quick zoom on it just in case someone's excited, and then we'll move on. Great. Thank you, guys.
There should be a button in the mess room.
Right.
Alerts here.
Like
it's
a panic button?
This is the one.
Right, like a panic button or
It doesn't
something.
say anything else, but alert,
Uh-huh.
alert.
You don't have like a group WhatsApp or anything?
We do, but it would take too long. Lift, middle.
Bottom left?
And we've already found that none of us really.
Yeah, we're not
pay
paying
that
enough
much
attention.
to
We are, but it's not
the
enough.
flashing red light in sunny
This
space.
could be another siphonophore. We'll take a quick
Yeah,
zoom.
Ferligama again.
This thing's everywhere. We'll
Yeah,
zoom in
Dougal
for a
was remarking
second.
that it's pretty interesting how we've seen it
Any
in all
light
depths almost.
Yeah,
Same species.
common.
Usually they're much more
Great.
stratified.
Thank you. Yep, just for a
Nice
minute or
shot.
so, not even a minute, 30 seconds or so, and then we can. Oh, come back to us. Focus. There we go. Great. Good deal. Okay. We can keep moving. This guy in the right middle, might not be any, might just be a fish. I think we're okay. Oh, is it a Tomopterus? I think it is. If it's a Tomopterus, we're gonna collect it.
Should we just go to
Or is it at that other worm, Yost? It's one of them. Let's get a quick video on it, but not too long as they attend to escape, as he just tried to do. Is he in the middle back?
As in up there.
Is that a Tomopterus or?
This guy right out here, or that other one, Anopterus. Didn't Karen say there was another one? Oh, thank you. Nicely done. Yeah, we'll zoom in real quick and then we'll collect.
Treading water a little bit.
Tiny, right?
Yeah, he's small.
Really tiny.
Yeah. Do you want to collect him? You good? Yeah.
I'm not sizist.
I figured as much.
All right.
Yeah, let's collect him. He'll go into sampler 6.
Another day, another worm.
No. Come on, buddy. He's in. He's in. No, isn't he in? I think he was in. You think he went outside? I thought he looked inside, but because it looked like he was stuck in the suction and tried to fight it and couldn't fight it.
Yeah, that's what I thought as well. Maybe I was wrong.
Maybe. Let's give it a second. Can you give it a little microburst?
Yeah. Probably missed them. You'd have been on already.
Yeah, I feel like because we don't have the shadow cam, right?
Full capture.
My fault. Sorry,
I
guys.
thought it went in. Okay.
Looked like you caught that little swirl.
Okay.
Outside the
Yeah, but I thought he was inside the funnel and I was pushing ahead.
We could go to that jelly.
Yeah.
Over to the left. While we're here.
Hey.
Hello.
Hi. Of course. You literally log copepods and cyclothonia about every 7 seconds. It would be accurate. It's Medusa. I like Trachymedusa better than the others. It's not Narcomedusa. It's not Narcomedusa.
Big stuff, please. Big stuff.
Yep. We'll get this guy and then we'll go for bigger objects. How's that? Yep. If we could get a zoom and then we can scooch, that'd be great. And hello, close one. All good. Yeah, that's all good. Thank you. Thank you. All right. Scanning up and onward, please. We're gonna have a pilot switch here, so hang on with us for 1 second. Somebody cover it.
You're welcome.
Somebody cover it.
Yeah, give your questions to 8, and then I'll close out.
Perfect.
So cold.
Thanks, you guys, for working all that out. It doesn't matter what it switches all the time. I'm like, pretty much it's name holders at this point. So how do you guys want to work? Hi.
Hi.
How's it
How you going?
I'm doing good. How are you?
Was the
The goal here is up slow. We're actually on the way back up. We made our target test today, believe it or not. So up and slow, search mode, stop on bigger things. That's the goal.
Oh, bigger things.
Well, we're trying for bigger things.
I like bigger.
Yep, we've had a rhizarian collected, so we've got some small stuff. So
3 jars left, and
and then there are some Niskins at 600 and then 50 and 0. See, that guy's too small, so we're gonna keep moving.
What's
Yep.
a teeny houses?
Yeah, everything
Wow.
seems small. If we could go a smidge slower. Thanks. Middle,
What's a focus hunting again?
middle. Right here. Yep. Let's just see. The rhizarian on the top right. That's a Ketignath.
Couldn't be.
It could be. You've been getting me more often than not.
Yep.
I think it is.
But then
We can keep going. Nah, we're good. It's a Ketignath. We love them, arrowworms. Thank you. But we're good. Keep eating the copepods, my friends. Yeah. Ketignath. You get me.
Oh, there's
Would-
left.
Was it big or small? Nope. But thank you. I can't see the left all the time. This is it. Thank you. Yep. Ketignath. Little tiny nothing. Something. Amphipod maybe. Not a lot in here, huh? A lot of small stuff. Ketignath. Hello, kitty. I think these would be too small.
Oh, it's the angel. Stop.
Oh, stop. Sorry. My bad. Nice cub. But Marcelo was like, am I getting stuck again with a Ketignath?
Yeah.
We want this guy. We want some video first, if possible. Oh, there was a fish too. This is a sea angel, one of the pteropod species. It's a gymnosome. It does not have a shell. There's another group of pteropods that has shells.
Oh, you good?
A little cyclothony up there as
Cyclothony,
well. This is the guy, if we can zoom on this. Yep. when you're ready. Good eyes, Marcelo.
Okay, I'm going to zoom in.
What type of pteropod?
It's a gymnosome. Could be a pseudothecosome, but what are my options? Gymnosome.
Thicosome?
Yep, thecosome, gymnosome, or other. Okay. Might be a pseudothecosome, but I don't think so. I think it's one of the true gymnosomes. He has wings that he can use for swimming. It's his modified foot. He is a mollusca. It's one of the midwater snails. We're gonna work to see if we can get some zoom on this and then see if we can figure it out.
Butterflies have shells. Angels do not. Is that kind of a common name for?
Correct. Yep. Sea butterflies and sea angels. Let's see if we can get a zoom on that white spot out there. There's no autofocus, right?
Yep.
Okay.
Both pteropods.
Yeah, I would love to grab some dinner in a
butterflies
second
have
after
the
this
shell,
pteropod.
like a snail.
Yep, the
on land.
Thaliptodon. This is one of the
And
pteropod
the
species. We've collected a few of
angels
these
just
in the
have,
nets. We're collecting lots of pteropods in the
a
net.
soft body, no shell.
We haven't seen many in the ROV, even though they are all over this place.
That's deep.
I was gonna say, should we collect it?
Well, I think there's only one species, isn't there?
I don't know that answer. Maybe.
Collective sequence, it.
Yeah.
You said you've already done it from shallow water?
No. I haven't done it. We don't get many gymnosomes that we use. Our net mesh is too big in the Gulf, so I don't get the smaller animals. Yeah, if we get video for a second while he's relatively calm. That's great.
How come we only get mollusks on your watch?
I know. I think I might be the mollusk whisperer. All right, let's collect this puppy. If we could, suction sampler would be fine.
Do you want to put in the same
He's
sampler
way
as
out
the
there.
fish?
What was that?
You want to put in the same sampler as the fish?
Do you think the cyclothone is going to eat him?
I don't think so.
I think he's going to go in his own bucket.
How did you spot him?
Marcelo did. I thought he was like a Keatignath or something. Marcelo's got the keen eye.
Swimming around.
Hmm?
Swimming around.
That would
It's like swimming.
That's good.
Coming off my screen. Around. Gonna go into 6 is the canister of interest. Oh, that's so gentle. The inside or on the outside? Uh-huh. There.
That's it.
Yeah.
Going down.
Yep.
Nice.
Maybe he'll be with a Tomopterus. Maybe they'll both come in now. We're gonna see like a quick dot. You can give it a microburst if you want for some suction.
Is it?
I haven't seen him. I think he's hanging out with the Tomopterus in the tube somewhere. I think the Tomopterus just ate him.
too?
Well, we were trying for Tomopterus, but he never made it into the canister. So he's either at the top up here somewhere, which I don't. Oh, something's at the edge. So we know the flow is working.
Oh yeah, that's been there for a while though. That's something.
Right. This is canister number 6, so
Yep.
this is the cursed canister on this trip. I have not seen anything go in.
Have we sucked anything big that we kind of jam across the?
Nope, I don't think so. 5 was an archipedema.
I'd say the biggest thing is like the cyclone probably, and that made it in. He's got it. If you bursted it.
Well, what we can do is, if we've bursted it a couple of times, let's just leave it on this canister, and we'll see what comes into it. We may have a Tomopterus, we may have a gymnosome, we may not have anybody.
It may just be all pinned against the screen.
Right. I feel like we would have seen it go down and then up. I'm okay. Let's just leave maybe a gentle suction on, and we'll continue on. And Russ, are you here for me?
Do you want me to relieve your turn, dude?
No, I don't think we need to rotate, because I'm not confident that either animal got in there. So we'll just call it a bonus bucket if there's something in there.
Want me to flush up here?
I would love to have some dinner if that's okay.
Okay, yeah, I wasn't sure what the,
I think
wasn't on ship line
Kakani
up
or
till
Yost
next.
is on next at maybe 6.
No, Kakani was on for
Yeah.
2 hours early. Get a Karen.
Right, so we're kind of
We're just
filling, slipping.
switching? Oh,
Yeah,
we're filling in for Karen.
Correct.
Okay, I was wondering what was happening.
Yep. I'll get it. Thanks. You need
All
something?
right.
I think Kakani will be coming back.
Okay.
$971 meters. Still nothing going on with canister 6, and the suction's on or off?
It's on slow.
I can't believe that there isn't anything in there now. It's not going to help but to have force on it the whole time. Yeah, let's get a little bit more upward velocity. House just went by on the right. But overall, it was kind of barren down here last night till we got up a little ways. Another house. Actually, I think it was a rise area. Sorry. What is that?
Okay. I'm not as good at this one.
Just rise area. Okay.
Don't worry.
Well, hello, John.
Hello,
I feel like I haven't seen you much.
Yeah, I'm in
Opposite
tucked
labs.
away in the lab all the time.
I know.
We got a question in the chat
Not that good at,
about
like,
O2 saturation
talking
and
all
its
the time.
effects by depth. If you feel like talking about oxygen minimum at all.
The O2 saturation is affected by depth. If we're looking at just the overall profile of oxygen concentration, it's affected by the different water masses that oxygen is coming from. Surface layer has in good contact with the air interface. It mixes oxygen down. But as we get into the next layer below that, the decay of the material falling from the surface water draws that oxygen concentration down. And below that, on the profile, we have oxygen that's being replenished from more poleward latitudes that has a higher concentration of oxygen in it. Does that answer the question as to what's causing the profile?
Yeah, I can, I'll put the profile up.
See, so we do have a little screen here that's showing the...
Yeah, so under tropical water like we are right now, there's just very little, that mid-layer depth has very little communication with the surface layer because of the stratification, that warm, floaty water that's on top of it, and then that colder water that's replenishing things underneath, and that mid-layer depth just doesn't get ventilated very well.
Yeah, so that lighter blue, more squiggly line there, that's your
Yeah, and
oxygen.
remember, it's a little bit deceptive because that line doesn't go all the way to zero on that axis. So there's still oxygen left. This area is not renowned for a really pronounced oxygen minimum zone as we get underneath some more productive waters, such as the North Pacific or even the northern Indian Ocean, where there's so much stuff that falls down seasonally into that layer and so much bacterial composition that the oxygen concentration goes down to something that is challenging for many organisms to live in, particularly challenging for things like fish that have very high metabolic needs. Some invertebrates are much more tolerant to that low oxygen than what vertebrates are. Vampire squid is one of the ultimate examples of that, where it's living in a habitat that most squid can't because it's found a way to ratchet its metabolism down ultra low.
What's that? Keeping that. Oh, fish?
Fish of some sort, maybe? We are still...
I don't know how to call this fish in here.
Ooh, that was a big house. Can we, it's too late now, it's gone.
Yeah.
I was looking at the chat and I missed it coming up. It might have been a ctenophore as well if we go back and review it. I just caught it at the last minute. You snooze, you lose.
Yeah, so although we don't have a really crazy oxygen minimum zone in this location, as we're coming up, it will dip down a bit and then it'll start climbing back up after a certain point as we approach the surface. Field of view is going to change because we just zoom a lot on this one. I don't know what the raw numbers are for you, unfortunately.
Jelly time again.
Only,
Gymnast time?
only because you're.
Like a
Yeah,
swimming
to
snail
me,
just before?
to me it looked like Polyptodon. But unusually deep for that. I normally get it in shallow water.
Where'd it go, Mom? Fight next siphonical.
The fish.
Ooh, you're doing the mouse on the leg trick. That's hard.
Oh. Nice.
Ooh, and there's a little jelly off to the left.
No, go ahead.
There is a Lycopleuros
House
straight
at the
ahead.
bottom. Can we drop on it?
Yep. Oh, capture. Forgot it.
That looks knobby.
I'm going to try and track down that field of view for you.
Maybe. Probably it's not really angel winged from this perspective.
Okay.
Yeah, zoom in, please.
Oh, was that the same one?
You got a D-sampler open still?
We do. Is it in there? It's in there.
See if we can get it
Yeah.
before it bails.
The animal's in there for sure.
The animal's there. It's Mesoicoplura. Very, very small head, long tail, thin.
You said like really close, zoom in.
Oh, interesting question here of whether fish will run away from the light or if they'll sort of freeze, like a like a deer in a headlight sort of thing. We see both behaviors. It really depends. And then the ROV itself also puts out a lot of noise, a lot of vibrations in the water. So you'll get critters that'll run from that as well. But there's certainly some critters that, when they're in the light, they really seem to get a bit stunned by how bright it is. They're pretty bright lights, and obviously in this environment, any light is a bright light. But yeah, some fish just, they book it, they run away.
Keep messing way down.
Biological clock.
Unlikely to get it back up with creating turbulence and making it bail.
I want to talk to you later.
Yeah.
When you have time, showing videos and talking about it.
Okay, it's gone.
Yeah.
Yeah, let me see
You
it.
guys want to do it? Yes.
Are you
I saw that search
awake now until whenever?
Until the samples come.
Okay.
And
Yeah,
then.
so we're on like really different schedules.
Yeah.
I haven't seen you a lot.
Actually,
Put the camera back up.
I'm working with the samples that come in the Tucker Tall. Also, sometimes with the ring net and helping Dougal with the samples of the ROV as well. And when we get some nice
Okay,
animals that
continue.
swim, and I pick some of them and record.
Are you getting good stuff?
Sorry?
Are you getting good stuff?
Philogama?
In the room?
Yeah. I kind of mounted a small studio. Has been great because many of these animals we don't have access from the coast. Is open ocean animals, and it's good to have them.
Yeah, I think it's just an old house. Matt, let's go after that.
We've only caught one, actually.
It's just a little. Is that a little red sedipid?
Oh, that's a scan, isn't it? Johanny?
Yeah, we can.
Or a little red jelly? Little red jelly.
That's not what I'm looking
Oh,
at.
is that orange lace cut?
Can
the little
we?
red one.
Oh,
it is.
come on, but
They're in
look
the
at that.
Atlantic
But
as well.
look at the thing behind it.
Yeah. Dougal, I think you make all this stuff up. You just to justify you collecting it.
Look.
Holy mackerel, let me get a photo of it.
Look.
And I think this is what we went by that I missed was a decayed
Can we do both?
structure like this. So
They're too close together.
you want footage of this and then you want a collection, I presume?
What depth is that, 927?
Yeah, but look at that behind it.
That's the first one we've seen all
It's
cruise.
the perfect one you are looking for, like big ones.
Okay, fine. Dougal. Do we want to scan this?
Can't we get both? He's only going to be small.
He's very
Okay,
small,
so
and
you're
he's
just gonna
gonna
collect
he's
it.
gonna be
Yeah.
pretty mobile once he gets excited. Looking at the way
Okay.
it's swimming, first of all.
What is this tracking
Medusa.
medusa?
Yeah. Well, repellent-nemated.
Repellent-nemated. Okay.
My master student now is
He or she.
working on changing a genus to fit one of these and combining them with others.
Close to Atlanta, something.
So, Dougal.
No, it's closest to Sminthia, probably.
Sminthia.
Are you happy with these shots so we can collect
Yeah,
it?
I know what it is.
All right,
That's great.
you're gonna collect it.
If we can collect it, yeah, I'd love to collect it.
Yep. Okay.
These are whole.
Dougal, in midwater,
Where
are
we
these?
collecting it in?
Yeah, we're collecting it in sections. Should be fine, right?
Yep.
In midwater, are the jellies are still all dioecious? There's separate males and females, or not sequential?
West.
Except for the siphonophores.
Yeah, siphonophores have got both. Usually they're separate males and females. You get some that bud.
Looks
and
like a fish
asexually
hanging out
reproduce.
over top of things, or a ketognath. I'm going to go with ketognath.
This guy, once it drops its tentacles, is going to be really fast.
Okay.
And changes direction as
bring the,
well.
Camera down to the nozzle.
Don't tell them what you're doing. And you'll be fine.
Is this still going into 6?
Still going on 6? Down to 6?
Yeah, I guess so,
Yep,
because we're not sure we got anything in there.
Let's try, because we still have nice.
Worst case is he's gonna eat the polyptodon.
Yeah.
Generally, the jellies are so upset about the collection process that they don't eat for a little while.
Samples from the Tuckertal and organize my stuff. I did not record anything today and organize my sequence.
Up behind it. Close.
We have in my place Sminthia aurigaster.
Is it what?
Sminthia aurigaster.
No, it's not.
No, I have.
Ah, you have?
Yeah.
Mm.
But it's not tall at all.
No.
It's like
Yeah, it's very
more
flat.
Flat and
Yeah, Sminthia needs to be spread.
This is also going into 6?
Yeah, we're not sure we've got anything in 6. That's the weird thing. And if we don't get this, then the pipe must be jammed.
Or
Again.
It's the evil 6.
It's the evil 6.
It's the evil 6. You're right. This is the cursed bucket.
Oh, it's
Oh.
There he goes.
Oh, we have tentacles.
Somebody's going in.
Some pieces of tentacle might have, yeah. Just behind it. It goes down.
There he goes. Oh, no.
Oh.
Almost.
Oh,
Just teasing you.
Teasing.
Teasing you.
No.
We'll get it.
Way up. Come down about now. No, I thought I saw him on that camera briefly, but
No.
can a little bit more to.
Is the big lavation house still there somewhere?
It was an old structure of something, but there was no house structure, no filtration structure left to it.
See if there's some tentacle. I know at least one tentacle.
Remember how hard it was to see
Yeah,
the
we
whole
got some
house?
tentacle.
Yeah. You don't think they're decaying and the inner filter decays faster than the outer filter?
I think it's the opposite.
Why would the inner be transparent though?
And maybe all of the big stuff, right, is landing on the outer filter, which is the bigger structure that we're seeing, the signature that we're seeing. It might have a really effective big filter, so that it's only getting really small things.
All right,
It
let's
looked to me
keep
like
on.
they were dead and they were sinking.
911
Still had-
meters. Let's see what's out there.
Acabamos de tentar coletar uma pequena hidromedusa da família Rhopalonematidae, provavelmente uma espécie ainda desconhecida, mas 1 centímetro de tamanho, bastante difícil. Foi possível. Mas alguns tentáculos foram coletados e esses tentáculos de biologia molecular, né, para tentar entender se essa espécie pertence a uma espécie já conhecida com uma... Realmente.
Not, or is it a tomopterid? Antennae on the one end. What? Is some type of a fish with a lure out in front of it?
Wow.
So I think it's still there.
Right.
It is.
Wow.
What I'm trying to make is
That's little
It's
larvae.
kinda-
Looks like
it's
a
a
larva.
fish
larvae, yeah.
Yeah.
But it had those antennae-like things out as like a lure.
Yeah, that's a deep sea fish
That's
larvae with a very long.
Different one.
Look at the tail.
Wow.
It doesn't seem to be very
myself.
fast, though.
They wouldn't need
How
to
far are we out there? Well, it's a good size. I mean, the fish itself's got to be on the order of 8 to 10 centimeters plus the tail.
Would that be under bony
Close
fish?
the iris.
Yeah, be under
Yeah.
bony fish.
Waiting for Marcelo. Yeah, Marcelo. Oh, yeah, right. Ok, agora chegou o professor Marcelo Melo, da USP e ao USP,
Stomach or yolk?
You're right. Could be yolk.
para
The
explicar
way it
melhor
was
o
to
que
be
pode ser
yolk,
esse
It would
peixe.
have to be an absolutely huge egg.
I don't know what is that.
Bizarre
Ah,
fish.
could it be a larval gigantura? The eyes
Peixe
look-
muito interessante
Yeah.
aqui. Uma larva, provavelmente, né? Dá pra ver ali o saco vitelínico ainda presente ali na região ventral do animal. Mas um bicho muito diferente que ainda não tinha sido visto.
We don't know what the larval fish stage in many of these fish actually looks like.
Identificar larvas de peixe é algo muito difícil, mesmo tendo uma morfologia diferente assim.
Just remember,
can I-
if you try to
how
collect
can I
this in the
tag it
suction
in the
sampler,
family?
it
You
will
have-
eat the ctenophore and eat it'll like the food web in the jar. So
Sorry.
everything in the jar will eat something else. So
I can try to put if the
do you want to try and
cyclotron.
Cyclotron?
Yeah, for the cyclotron.
Do you want it?
Yes.
Okay, we got some good
Please.
footage.
Not really. Better to stay on it a little bit more, I think.
Nice.
We don't have a side-on view. You can't see the eye.
Okay, well then we'll go zoom back in on it.
Ou sei.
Não, isso não é cyclotron,
A little
não.
bit more footage. It
Ah, não,
doesn't seem
pixel.
particularly
Isso eu
mobile.
acho que é uma gigantura.
No. And it doesn't even seem to be sinking.
Gigantura.
Really. Look at it in like the other particles.
It
Oh,
looks like
slowly
a
sinking.
dragon.
Giganto.
You know how they mimetize jellyfish, Ethanophorus?
Pretending to be a siphonophore?
Yeah, it's pretending to be something stinky.
Olha só. Explica aí pra galera, Marcelo.
I believe it because I know when I came across it.
This is very interesting behavior.
Né, esses peixes, eles mimetizam
So we're looking at it from the
medusas e
belly
sifonóforos.
side.
Pra parecer algo que não é comestível. Eles têm várias projeções nos raios, às vezes no intestino. E isso confunde um predador.
Bucket we'd like to put them in. I'm just not convinced we've got anything in 6, but we know we think we've sucked them up. I just
You did suck up
Gênero
one tentacle.
gigantura, então? Posso deixar?
Não sei, cara.
Giganturide.
Bota aí.
Giganturide.
We haven't seen anything
Well,
end up
we
in
should go get
6.
it before it runs away.
So if we want the fish and we try and put it in 6, it's haunted.
You could rotate to the.
We could rotate to
cyclophone
7
put it on the
and then come
cyclophone.
back to 6 later in case we get desperate.
Put it on top of cyclophone. Because that won't get beat up.
Cyclotron is also very small. It won't eat anything.
Which one's got the cyclothonia in it?
5.
Okay, rotate back to 5.
You can get a shot in focus.
Vamos tentar fazer a coleta do peixe, mantê-lo dentro do mesmo.
Here we go. This is a chance.
Wow.
Olha só que imagem, cara. Olha só, velho.
There's some of it when the head was facing away. Like
Yeah,
that was
we should-
almost in focus.
I think it's the propeller.
The jets from the propeller.
Yeah. It's creating a current.
It's normal for the fish have these long tail.
Yeah, it's
Mm.
normal for some.
From this
Depends
deep
on what you call
deep water.
normal.
No, in deep water.
Yeah, sometimes they
animals.
mimetize.
He said, we've all had it.
Yeah.
Okay, do we have what we need yet?
One in focus. Close. It's a little bit bright.
Yeah. Love to short line.
Still not in focus.
Yeah, just go and get it.
You just gonna get it? Give it?
Yeah. No, do that. Yeah, let's get it.
It's only gotta have one frame.
Okay,
Just do like Google, just get a little piece of it.
Yeah, it's.
let's go collect.
Collect it.
You just
Yep,
go
we're gonna collect it.
Yes.
Suction.
All right.
Was it in the 5 plus the cyclotron?
Say again?
Cyclotron was in.
Katie's list says it's in 5. You should go check.
Katie, check with her. Are you sure cyclotron was in canister number 5?
Yeah, according to
Why is it still indexed to 5?
Well,
Turned
do you
it
not
back
have
to
it in
5
there?
Because cyclophone, the
I
fish
thought it
is
was
in
number
there.
4.
You can
Yeah.
put
That's
this
the-
on
it
top of
says
it.
that
'cause
number
the
4 is the
hard body
arc
don't-
Oh, but do you
Oh,
see
yeah.
it?
We want 5, right? Okay.
5 is ready.
Is 5 what we
Do
want?
we still see it in there?
5 is labeled as the cycle.
I'm just
I can't-
wham
Okay.
right now.
Can you see it?
No.
If it's swam out, it should get sucked back in.
Yeah.
I can't see it on the mesh unless that's it there.
Yeah.
Maybe something will swim down from 6 so we can see if there's something in there. Right now I'm
Huh?
just seeing the far.
What is that?
6. Thought something for a second. Whoop. Okay.
That definitely was done.
That definitely went in.
Oh, yeah,
Oh, the cyclotron is there. Yeah.
Oh,
wow.
yeah.
Mini aquarium.
We got some swimming around on the top of there right now.
That's cyclophone from before.
Yeah, no, I'm just saying he's still
He's
there.
still in there, yeah.
Yeah.
You want micro sock?
It's not that much pressure because he was able to swim off the screen, so that's good.
Is that like duct tape
Do you want
and
micro pump?
zip zip ties.
Yep, you can't fix it with duct tape and oceanography, you can't fix it. Yay! Whoa, the tail broke off!
Wait.
Okay, did we decide what it was?
Oh. Okay, you can rotate that.
Legal, coleta feita. Agora temos dois peixinhos ali.
You could definitely get an ID.
Um cyclotone e esse que pode ser da família Ganturide, segundo o professor Marcelo Melo.
Did that overshoot by half a turn just then? There we go.
Yeah.
Good job.
Thank you. Thank you, Marco.
Okay.
Try to push it up.
No, can't.
Put it not up.
Thank you.
Good?
I'm good.
All right. That was exciting.
I'm trying something really low.
That's fun.
Doesn't seem to be.
Okay, we definitely need the panic button up in the mess.
Go.
A little, like a little mic situation or a little intercom or something. Marcelo ran down a lot of stairs. He did a very good job.
Yeah, you almost fell there.
That's what I did the other day.
Yeah, it was fast.
I think just a button, it was the sounds of alarm or it turns on the light, it's like wait for the
Let's get ourselves set up and get back into hunt mode.
No, I hadn't seen it before.
Where are we at? 902 meters.
Sometimes they are.
Thanks, Marco.
You're welcome.
So then they will use the yolk and things like that. They use the end of an egg string. Yeah. I don't know. Right. Very cool.
The fish were crazy. Jen, I have something for you.
Siphonophore down. Nope, it's not over here. What is that? It's moving fast. Helicreatid?
Small jelly.
Oh, I don't know about that. This fish, I'm not sure.
Helicreas.
It's awesome. We have a couple good images from deep end.
Halopithectrepes.
Could take that big fish. Yeah, that was a different one we saw tonight.
Oh, sorry, Botryonema. I'll get it right. It was Botryonema.
That was a little scary.
That was my second guess too.
Yeah, I saw the knob, but my head grabbed the wrong word.
What is the Botryonema?
Botryonema is a helicreatid.
Trachy
They're the
medusa.
same as the other one that I'd thrown the name out on.
Yeah, I saw that.
We skipped each other.
Oh, the title.
No.
I think so.
What's that? That's starting to look like a Doliopsis.
Maybe they're just staying in the density layer. Don't have to do anything.
Trying to get a zoom in on it, figure out which of them it is.
Moving away from the ROV rapidly while remaining vertical in formation
detail me.
The dorsal fin was moving like a seahorse.
Right? Maybe.
Yeah, but to generate.
But look at the fins. It's not like they look.
Well, if we watch the video.
Hey Russ, is a doliolid this one? Is a doliolid?
This is, yeah, it's a doliolid. We've had three different things that all look very similar here. A doliopsoides, a doliolula, and a paradoliopsis. So I'm trying to figure out which of them it is.
Okay.
To decide if we need to collect it.
Right.
Okay, I'm guessing this is worth collecting. Let's put it into the cursed suction sampler 6.
No. No, not tonight. Have fun.
But we still got both these samplers open?
Yeah, one.
Actually, let's do a D sample on it, because I think this is one of the one that we only have one of so far. It's swimming different. Let's do a D sample, just to make sure we get it in good shape. And it's also a lot deeper than what we found the others, so. It's going to be about 2 centimeters in size, to put it in perspective for scale.
Preemptively deal with it.
Doliolida.
Mandei a foto do café quando eu passava lá pro
Café com quê? Que loucura esses
E
bichos.
aí ele disse, eu canto marinho. E ele essa é bem. Tá seguindo, ele falou, passa uma foto dele assistindo na TV.
Ah, ele tá vendo aí? Agora um doliolido, um animal pequeno ali de 1 centímetro mais ou menos. Estamos tentando fazer a coleta. O professor Russ Hopcroft vai tentar analisar o material em laboratório para fazer a identificação desse animal, algo que é muito difícil, mesmo trabalhando em laboratório em condições mais controladas, é bastante difícil entender as estruturas do corpo desses organismos gelatinosos, bastante transparentes e pequenos.
Way down. Probably lost at this point then. Unless that's it out there, I think it's gone. No, that's not it.
Deu pra ver, né? Aquele print que ele tirou depois só. Não tinha pra onde escandinavo.
Não.
Só que eu achei estranho porque eles não podem divulgar um resultado assim. Mandando um abraço aqui pro professor Eric Muchagata da FURG, que deve estar nos assistindo aqui. Um abraço aí, acompanhando sempre as transmissões.
Sinker there, I think.
É.
Something here is producing some very big old mucus structures.
Sim. Olha lá. Opa.
No, a penguin. It's a
Pinguim.
Okay.
Olha, acabamos de encontrar um pinguim agora.
Any idea? This is like looking at clouds.
Um pinguim, olha
Yeah.
que
I think it's a
bonito.
penguin with a
Eu
shawl.
vou marcar aqui no Squidle. Which,
Baby chick out of an egg.
which species?
Yeah, could be, could be. But look at how much of an umbrella structure that has. It really.
Do you want to scan this?
É, acho que sim.
It won't hold together.
Just one scan at the top there.
Can we zoom in at that top structure? Here. No, nothing. All right.
Quem que falou isso?
Yeah, I can. I actually think it's a horse head now.
Isso fala, né?
You know, with that scan, it does look much more like a lobate, doesn't it? A carcass of a lobate that's just decaying.
Essas estruturas mucosas podem ser produzidas por vários animais marinhos que secretam muco e usam esse muco para aglutinar partículas que eles se alimentam depois, ou pode ser um próprio animal marinho gelatinoso que morre e deixa essa estrutura, essa carcaça, né, que vai afundando. E essas partículas têm uma importância enorme para o transporte vertical de matéria orgânica, porque elas vão agregando outras partículas, né? Então existe uma coagulação que ocorre e essas partículas que vão afundando acabam sendo povoadas por outros animais, né? Copépodes principalmente, que ficam ali esperando encontrar essas partículas e eles se colonizam, né? E se aproveitam dessa processo de agregação, aglutinação, e usam essas partículas para se alimentar, né? Constroem uma verdadeira casa dentro dessas carcaças de animais mortos.
Okay, let's try and get a little bit of height. Go forward. Yeah, I'm trying to figure out what's above it. We don't need to stop, just drive through it. I think it's just part of the house. I'll
I just
open
think
it.
it's definitely how far
Oi?
Yeah.
Ah, tá costurando?
Okay,
Rasgou, né?
let's try and get vertical
Guilherme falou.
velocity of about 5 or 6 meters a minute till I stop you again.
Maybe it's not.
There's a jelly of some type down below.
Maybe we could add. It's a, right? It could be a bonus bucket for the day. We don't know. It's 6. It's holding up to its name.
It's whatever.
It's whatever could possibly get in there next.
We need to do one of Carol's eating and put it in there. Shake it around.
See how many. See evidence of.
Two things off to the left. Another house off to the right. Let's try that. See what's on it.
Other house? Or is it?
Indicator.
Heating mat behind it. Oh, that's big.
Oh, it's beautiful.
Sedusa?
Sedusa.
Okay, and keep us behind it. We can go along. We did get the other one, right?
I know, this is a treat.
And they're like this.
Yeah, we did.
I saw a ship today. You remember you said you hadn't seen a ship?
You saw a ship?
Yeah, there's a ship.
Whoa, you mean we're not alone in the world?
And I think it's a livestock ship.
Are we getting
Sedusa.
What's that circular thing on the that completely circular?
How do you spell sedusa?
P.
Is that where the bud of the baby
P.
normally appears?
E-S.
On the top?
Ah,
On the
E-S.
bottom, you see like a slightly greenish looking, completely circular object.
Livestock.
Yeah,
Yes,
there's
you.
several of them have had that kind of process off of them.
Wow. They came to resupply us?
The ship.
E-S-E.
Seduce
We now have
PS.
goats.
It's under mollusk.
Seduce?
Different
Goats
menu
and
items for
doliolus.
the next couple of days.
Is it? No, sorry, it's a doliolus, sorry. Yeah.
Doliol. Sedusa.
Oh, I think we can just leave it. We just collected one already this time, so. Yeah. Google.
Yep.
Did the
Sorry.
one we
You want me to hand you a laptop?
go in for whole genome
yeah,
or
one
just
more.
skim?
And then I'll have to-
Just the standard barcodes.
Huh?
You're gonna do the
No. This one.
Couple of jellies out there. $870. A little slow.
Going for big and exciting. I forgot. It's right here.
No, yeah.
It's like, yeah, I could have done it. You could have done it.
Oh, next time. Next time.
Do you want me to come back?
I don't know. I mean,
Because we were covering
Here's
for
your
Karen.
kitty
Right,
cat.
so we were covering for Karen.
Come back
So-
fully prepped.
when is
In theory.
When is Karen's?
Thought I brought
Because
one.
Russ has
Okay.
got an entire plan laid out for what he's gonna say.
Huh?
Fish. You think it was a fish or a Keith? Must be a Keith.
On the left.
Yeah, probably.
Okay.
Yes, 5 minutes.
Russ, you have 5 minutes.
I have 5 minutes. It's enough time to make what? Long grain rice?
I think the rotation.
Okay.
Oh, yeah.
Okay, we got 5 minutes to make the world a better place.
Oh, that's great.
Take some chocolate too.
Great. Yeah.
Or at least to make some organism famous. Euchronia, Falkor. Check this out. Right here. Another Euchronia. Oh, suckered in every time.
Tomatoes?
Where? There?
There.
That's key.
Oh, it's fast.
That actually look like a coelodendron.
Philodendron.
House.
Lololith.
Ooh. Whatever it was.
What was that?
Slow down a little bit. We're going too fast. Let's check on
Small
this.
jelly. Another jelly in the center. Oh.
Jellyfish. Some type.
It looks like six
Oh.
tentacles. Six
Oh, that's the
tentacles.
Atorella.
Yeah.
That's it.
Torella. Oh, yeah.
Get a close zoom in and
Oh,
see if it's got
this-
those green fluorescent
Yeah. But
patches on either
from
side of
yeah,
the
it might be interesting to see. The one we're looking for. Yeah, good. Parece que é uma água-viva que estávamos procurando.
It looks like it might have. It's the
Six
same one.
tentacles, yeah, six tentacles.
Dozen of these now, right?
There were two species.
Count.
We've collected two of one and one of the other.
Exatamente. Oh, beautiful.
Wow.
Very
Agora,
beautiful.
uma água-viva sifosô do gênero Atorella, uma espécie que nós estávamos procurando.
Google, we get a spelling?
Atorella. It's a scyphozoan. It's an undescribed species. And we found some very
Oh,
interesting
look, it's got
organs.
Santa Claus face on it. There's the eyes and the
Can we
and the
see anything?
eyebrows.
I can't see green. Though.
Yeah, it's by the tentacles. It's white. It's white, yeah.
It's white, not green.
Yeah. É uma água-viva que ele é bem diferente pelo fato dela ter uma simetria hexâmera, né? Tem 6
While we have
tentáculos.
it in like nicely positioned in front of the ROV, dim the lights down as dark as you can, so we don't lose it, but so we can see if it's bioluminescent.
Okay, looks like we found it.
Yeah, we found it.
Yes, this is it. This is it.
Encontramos a espécie que estávamos procurando.
I saw the two dots, but of course the green is.
The green is hard to see,
I don't
right?
think you can
Yeah, it
see
still
them.
looks like a face. I can see
Yeah,
the
it
does have the
eyebrows.
two
the
patches
nose.
of-
It's very
It has the
tiny.
two
These.
patches, but
Yeah,
the
it's.
two dots
We
under.
can see the pads. Amazing.
The gonads look pretty round, don't they?
Pretty round, not like sausage
It's
shape.
kind of weird because it has 6 tentacles but 8 gonads.
Yeah.
Normally, 8 and 8 or 6 and 6 or 4 and 4 or whatever.
I
The
see a
gonads or
patch
that
right
actually carry
under
embryos?
the flap.
Well, yeah,
They're
they
gonna
call them
sample this,
gonads.
right?
We're gonna sample it, yeah.
Esse gênero, ele é bem
tipo de uma simetria diferente, né? Simetria hexagonal. Seis tentáculos, 6 estatocistos.
What do you guys want to do with it? You want to do a full sweep?
E geralmente as águas vivas têm uma simetria tetraradial, né? Múltiplos de 4, né? 4 tentáculos, 8, 16. Essa aqui teve ao longo da evolução do gênero uma transição para uma simetria hexagonal. Mas ela interessante o fato de que ela ainda permanece com 8 gônadas, né? Então não é uma transição completa da simetria do corpo desse animal. E nós estamos estudando a bioluminescência que esse animal produz, né? Trazendo para o laboratório, estudando os tentáculos, a base dos tentáculos, que tem um órgão bioluminescente muito interessante, que nós estamos fazendo experimentos em laboratório com esse organismo.
Yes. It does it, yeah. And did you see Gasper's paper, like last year? They finally mapped the
So something could go in.
one of the they mapped it
Or
for,
I'll ask you what he wants.
for the Neopsis, the nerve net, to show how the reorientation couples.
You guys still have them, right?
Yeah. Super
Right there.
nice.
Perfect.
Again, just for one species.
We
So
can't
should
lose
also
this
look at the
guy.
statisticians.
Dougal,
Yo.
when we get to suction sampling down the road, do
Yep.
you want this in canister 6 or do you want this in canister 7?
Six
Six
is
is
cursed.
cursed.
Six is
All right.
So no
I would
6,
like
7.
canister 7, please,
Six,
for this suction sampler. We're currently on 6.
My kids are listening.
No, every day.
And we'll go back to 6.
It's over. It's not over. It's not over.
That's why I was asking.
Trust me.
Healer's choice on that.
Oh my God, everybody.
You have to break the curse, too.
Well, well, actually, we
Not
just
a
need,
problem.
we just
Yeah.
need to
Yeah.
remove a sticker. There's 3 6s taped to it.
Yeah.
Oh, there it is. Looking good.
They're
Yeah, you can
showing
try
up.
to keep him right in that picture if you wanted to. You don't
Oh,
have to go
yes.
back
He's
and forth.
coming to the scan. You
He's
have
coming
to-
back
It's
up.
aligned.
Coming to the scan.
Oh, no.
Don't lose the other
Yeah,
went up,
we're gonna
he's
maybe
going up
get,
and out.
get a twofer.
He's up over here.
2 for 1.
Okay, I can't watch this torture. When you guys do the sample.
Mondo
Is he
can't
saying
handle the
working
pressure.
with us is torture?
Coming back. Yep, close. You're close.
Oh, nice house structure.
There he is. And you want to try to keep him in the frame. He does not have to go back and forth. He's too close.
You do, however, need to back off a little bit. Close.
Yep. Right.
Well, you said he had go next, right? So it's definitely.
He's gone back out. He's going over to
Well,
the side. Going down.
We've been clear which gender are going out.
Okay.
It's pretty hard to tell them apart.
Is it?
Yeah. In some species, there'd be like a difference in hue or a difference in texture of the gonads, but they're really in the same place.
Okay.
There he is, coming in. Got him in the middle. He's too close. You could scooch him backwards a little bit. Just gone down below the frame. Back in. No, I think he's just outside. There you go. Try to keep him there. I don't think that's easy, but. Yep, now he's back close. Oh, just off the screen. Definitely the right pattern. The right pattern for sure. You guys want a couple more of those, right? We haven't gotten really in there, but it's passed through.
I think we had to pass through there. I think
Are
we
you
can
okay
move on
with
to
that?
DPIV probably.
Okay. We'll go to DPIV then.
Então essa medusa foi escaneada pelo iRIS e agora
Everybody keep their eyes on him at all
DPIV.
times. We don't want to lose this guy.
so invested in this jellyfish.
Jenny's like, I need to see this. He's a little too far outside on the right. He needs to come in here-ish when you can settle him. Yeah, getting there. Yep. Slow and steady. Oh, he went low. He went low. He's low below my frame. Yeah, I would definitely go down.
Yeah.
Shit. Okay, here he is. There he is in your frame.
Do you think this is scanning?
Going up, going up, going up, going way up. Oh, now he's settled.
Species description, like morphological description for.
Little
That's so
far up,
important
right?
that we can add this to a species description.
Yeah.
Because line drawings are so nice, so 1800s.
Yeah.
Oh, that would be great. Got him. Did you see that, Yost?
I did see it. That looks pretty good.
Is he a little too far right?
Okay.
He's going up off my screen. Gone. Back.
Today we found another species with a greenish structure inside. We could not see yet, but it's a Siphonophore.
Going low.
I was trying to identify with white light, and with white light you cannot see the green structure. And then I moved to black light and boom! Greenish. Yeah, it seems like it can be more common that we
We
think
just
it is.
haven't been able to observe it.
Yeah, and because
Yeah.
most of these animals
Nothing too
are
far
very
right.
fragile, so it's
Yeah.
the best way. No, it's not possible to see.
There he is. It's in your frame. Are we getting tugged by the surface? There's enough umbilical, right? Yeah. No, it's fine. It's no. I am not suggesting this is easy by any means. No, all is good. All is good. I couldn't do this. This would never work for me. What you guys do, I could. Oh, that'd be perfecto if you could do this. Oh, he's going up again. A little better. A little better. Yeah. It's too far out. Getting there. We're seeing him in view. He's gonna come in tentacles first. Maybe head first.
Push. Oh, it's going high.
Nope, he's right there.
Push the tide high.
Yep. Coming back.
Left.
He's up here. Okay, you want one more pass, Yost? You want one more pass? What you need?
Yeah, I would like another pass. I
Yep.
did a bit.
Sure. We give you one more. That would be great. And then we'll collect them up. Off my screen. Back in, back in.
For you?
Hey, Renata.
Yeah.
Somebody's asking if anybody speaks Spanish. I don't know who's in there.
Spanish.
Yeah. If not, fine.
Not
I just
Spanish,
saw
but
in the.
Português.
I was gonna say Portuguese. I didn't know if you knew both.
Português, sim. Professor Renato Nagata, aqui da Universidade Federal do Rio Grande. Estou tentando falar em português, às vezes portunhol. O que vocês estão vendo é uma espécie do gênero Atorella, uma água-viva de sifozoa, que provavelmente é uma espécie não descrita e que nós temos coletado em alguns dias e que tentaremos trazer para o laboratório para realizar experimentos sobre esse animal, que tem órgãos bioluminescentes na base dos tentáculos. Então, na base do tentáculo tem dois pontinhos que às vezes eles aparecem, quando olhamos no estereomicroscópio eles são verdes, mas aqui eles são brilhantes, né? E esses pontos são órgãos bioluminescentes que esse animal possui.
Right on the edge. You can bring him closer. There he comes. Here it comes. Yep, a little fast, but it was okay, right?
All right.
Yost, you okay?
Let's go to collection.
Okay. Sweet.
Good.
All right, team. Next task. Wait, we said number 7. Perfect. Thanks, you guys. That little thing. Yeah. Right now we're attempting to collect this Medusa, small undescribed jellyfish. We've seen only one other time on this cruise, so we're excited to grab this guy. We are about 360 miles in the South Atlantic off of Brazil, and we're here for another few days doing some fun science. Exploring the midwater column down to about 1,200 meters today. And we have various tools and equipment that we are using to make imaging and sampling of the midwater a little more efficient. Just to start.
Oh, not yet. Oh.
No. Right in here. Yeah.
Oh.
Nope. Too close. He is tiny.
Animal que mede cerca de 3 centímetros, né? Então imagine a precisão na pilotagem do ROV para se fazer essa amostragem. E a bomba de sucção não pode ser muito forte, né? Porque esses animais são frágeis e queremos trazê-los intactos praticamente.
Coming in.
Nós coletamos já alguns exemplares dessa espécie e ela rapidamente, quando começa a ser analisada em laboratório, ela já começa a se desintegrar. Então, tudo que precisamos fazer, né, para testar a emissão de luz, né, desses órgãos bioluminescentes tem que ser muito rápido.
Up off. Back in. Just starboard. Yep.
Oh.
Yeah. Yeah. We got him. Let's take a look to make sure he gets in canister 7, please.
So, this is valve 5 and 6.
Vamos ver se ela entrou realmente ali. Canister 7, canister 7.
Okay, 5 or 6. 5 and 6. Okay.
Those that seat together. One side. The other one moves.
Ainda não apareceu, só se confirma quando vemos ela dentro do canister.
Dougal, do you want to do a microburst?
Yes, please.
If we could do a microburst on the suction. Very quick little pulse.
And like there's whole workshops just focusing on marketing. That's the only way we should do it right.
There he is. Hold on one second. Okay, you can wait 1 sec. You can change canisters
Conseguimos.
to number 8. Yep, we'll leave 6 for the end. Okay, nicely done. Thank you, guys.
Thank you.
Sweet. We definitely have canister. Do we have? That was 7. Okay, so we have 1 canister and a half if we want to go back to 6. And we have 1 sampler, right? We still have 1 sample? Good, we have a D1 open. All right, so I'm going to propose, can I propose that we go down to 600 meters and explore a little bit? And then 400 meters and explore a little bit?
Yeah.
Great.
Sounds good to me.
Yeah,
Awesome,
great.
let's do it. Okay. 600 meters, Niskin sample, and some exploring. We do want to hang up above, yep, we want to hang up above 400 meters at some point for a little bit, so we're going to kind of step our way up a little here.
Vamos começar uma subida aqui agora.
It doesn't have to be super fast, but an okay speed, and if something amazing, then yes, we'll stop. But not as slow as we were doing before. Thank you. If that is as clear as mud. Thanks. Didn't describe that great. There's a question, ROV pilots or Tomer, how deep can Sebastian go? Okay, so Sebastian is rated to go down to 4,500 meters deep. And that's how deep they can do operations safely with enough wire out and things like that. The seafloor here, I think I saw somebody in a chat say 4,000 to 5,000 meters, which is correct. So we're never going to get near the ocean floor on this cruise ever. Our max depth for our cruises or our dives this week and next week are 1,200 meters.
Uma medusa, traquimedusa. Agora
That
estamos.
was a house.
Vamos subir para 600 metros, explorar mais um pouco em 600 metros, depois subir para 400 metros, também explorar mais um pouco ali em 400 metros. Creio que não é possível vocês verem pelo streaming, mas aqui nós conseguimos detectar que tem uma camada de concentração de organismos em 500 metros, mais ou menos, que nós vamos explorar, vamos estar passando por ela, né? Uma camada em que vários animais estão presentes, né? Animais zooplâncton, animais pequenos, e isso atrai outros animais maiores, como peixes e cefalópodes. Então, é uma região bastante interessante para ser explorada.
Thanks, John.
So I see in the chat it says, would it be possible to reach the seafloor? If the seafloor is less than 4,500 meters deep, yes, SuBastian could reach the seafloor and do some benthic surveys. For our cruise and our research projects that we're doing, the Designing the Future 3 is focused on the midwater organisms. This is the largest habitat on Earth between the mesopelagic and the bathypelagic zones across the world's oceans, and they are vastly understudied. So we are excited to be able to highlight some of the amazing organisms that we're finding in these zones.
Professora Heather Judkins respondendo uma pergunta, né, se esse ROV pode chegar ao fundo do oceano, e sim, se o fundo do oceano tiver uma profundidade menor do que 4.500 metros, esse ROV pode explorar o fundo, sim. Mas nesse projeto, Design the Future 3, nós estamos nos concentrando na região da coluna d'água, né, que é o ambiente mesopelágico e o bate-pelágico, que é a região do planeta com maior volume de ambiente habitável por organismos.
We could go up a smidge faster if you wanted, just to make sure we get there so we can spend time maybe to the depths, you know what I mean? Again, if something amazing is out there and large, but it should be good.
Red dot. Station.
Small little Medusa.
Oh, gotcha.
Essas coisas.
Yeah.
We're going to go up about another 130 meters or so, and then we'll hang out there for a little bit of time to do a little exploring. And then we're going to go up above 400 meters to check out that area. We do want to spend some time around 200 meters, 150 meters as well for looking for some of the targets that we still would like to collect. We still have a couple of options for collection on this dive, so we're going to make the most of it. Somebody said it the other day too. I don't know enough about it. Yeah. We start playing.
Someone out here knows how to play song.
Just a musical interlude. Oh. Oh.
It was you. It was me.
Marcelo here.
What'd you find?
Somebody on the chat is suggesting it's a whalefish larva, and not if I didn't just pull up a picture, and it looks very similar.
Really?
Look at the whalefish larva on. I just Googled it. I couldn't Google it.
You couldn't Google it? You
But I
Googled it
could
now?
Google it. But it's not far off. It's got the streamer.
Oh, it's got the same head too.
Yeah, I think
Oh, what are the
nicely
fins?
done.
Fins.
Who is this? TU Explorer 1? Thanks for the heads up on this. We're going to look at that.
Well, why are the fins not good?
I think she got
I don't
from
know.
George.
Could be position.
There they are, but
Jesus.
they're really small.
They're smaller.
This guy is every
right.
day
So I don't know.
in the
But
shed.
close. Maybe whalefish family, different species. There's lots of species of
Whalefish.
whalefish.
Now that is cool.
Mm-hmm. Maybe. It could definitely send Marcelo and Marco into a right direction to look at.
So maybe that thing I yelled out as being a coelacanth was one of those. Whalefish.
Oh, that was it. Yeah. Uh-huh. Coelacanth, whalefish. They're really close.
Really?
Really close.
Good call.
Mm-hmm.
Cool.
Yeah, it was on the chat. Who'd you say it was? Oh, nicely done. Hi, George.
Amy F. Stomaday.
Okay. Nice. Great. Thanks, you guys.
How many of them? Well, if they're on Google,
We've
I
got,
guess.
yeah, we've caught whalefish on deep end, but we haven't caught any larvae, to my knowledge. Nothing that looked like that.
George says that the Smithsonian calls it a tape tail.
Yeah.
George Mascota.
We'll share it with Marcelo for sure.
Livestream oceanographic.
As it metamorphoses, it gets.
Yeah,
shorter,
it's
it
so
gets
different as an adult. I want to know all the steps now. I want to know all the whalefish stages.
I'm not sure if we can get through there with this fish.
Probably not. Probably not. Getting close, team. 600 and about 30 meters.
Oh.
Do a little exploration and then go above 400 to do. We have some chat folks that think it could be a larval whalefish.
What you collected?
Yeah, your
ribbon tail. Tape tail.
tape tail,
Yeah.
duct tape tail, ribbon tail,
Toilet paper tail.
toilet paper tail,
Chicken tail.
streamer tail.
Tapeworm tail.
Philodoma.
You saw a Philodoma?
Yes.
Classified siphonophore. Oh, isn't it beautiful and elegant?
Yep, too bad he's 15 meters too deep.
Close up here. Okay.
We're so close, team. We're close.
You want Niskins at 6?
We do. We need one Niskin at 600, please. Cool. We are arriving. We're gonna.
We can't be. There was a comment someone made on Facebook 6 years ago. A picture of the similar fish made by Black Tide.
Okay.
And Ben Johnson replied to that,
Okay.
saying that it's possible there was a whalefish.
Yeah.
Yeah.
We're getting it from various reliable sources.
Oh, really?
Yeah. The sources on the chat. We know the folks on chat too. Yeah.
Yeah, Ben Johnson was.
Yeah. He's reliable.
Lisa, but in the live stream oceanographic Discord, George Matsumoto.
Vamos disparar uma garrafa, uma abertura de uma garrafa agora
Want to make sure we get
para coleta de água.
Yeah. If you guys are.
It's only one, right?
Yeah, just one. Not on here. I think it's only in here. Yeah.
Hey,
The
Dougal.
crew.
Dougal.
I would just put. I would put that. You know what I mean? Unless it already is. There it goes.
Hey, Dougal.
Yeah.
Do you
That's what I
feel
thought.
the Niskin bottle?
Yes. Yeah. Event. Long event. Is.
What Niskin bottle was that?
2, number 2. So we're gonna go up and forward slowly, stopping on anything
Like
larger
sample.
than small, correct? I think it's larger than small.
Larger than Arctic.
Yes, larger than small. Isn't that thing like a small organism? Right, I was gonna say it. Scannable. Okay, we'll go with scannable. So yeah, up and forward, up into the 500s we go. Does this whole misses count?
Logan. I already see that.
And a doliolid.
We can stop.
Oh, that's a doliolid chain.
We need to stop. We
just
needed
went
that.
straight past
I was saying stop. I was saying stop.
We still want those?
I think we're trying. Yeah.
Why?
Why?
Yeah.
We found polyps.
Here it is. Here it is. Dolio
Oh.
chain. We want that. We want to. Do you want to scan it too? I know we want a little video.
Uma.
Do you want to try to scan it or no?
Look at this, the fish.
Agora
He's not
encontramos
in my view.
doliolos.
Is
Not
that something
sure
the
where they
bigger
are.
large
Oh, I
in the
see. You see
background
them.
there too?
See? Doliolid.
They're coming.
Lose him?
They're coming. I think. Is that guy down here?
Because the doliolid chain we got was
There's
doliolid.
one.
This guy. I think it's back here, isn't it?
Was doliolula equus, which isn't the one that we like the first chain that we're
Yeah,
trying to get.
this guy.
There's the.
It looks
there's
like
your-
a
in the bottom left is your.
There he is.
Oh, yep. You're
But I
right.
agree
That was
bigger
somebody
thing in the
else. Sorry.
distance looks more interesting.
Okay, if we could bring that light back
It's
down.
another
Yeah.
hammerfall.
Could be. Splits up on them, so.
Okay, let's get a good zoom on this and then we'll go. What do you want to, do you want to DPIV it? What would you like to do?
Just
Next to the
just
boats.
suck him up and go to the bigger thing in the.
Oh, okay.
It's a little bit further away for the scanning, I think, right?
Do you want to do a sampler lab for this?
R-
Or do
I-
you just want to suck it up?
Yeah, if we can do it with the
We
desampler,
have a
it'd be better.
desampler
Yeah.
available, I believe.
So hurry.
So no scan is what I'm hearing.
It's
Pula
close.
dar.
I think they might be moving to.
They are moving. But you guys tell me, whatever you want.
Can we see red spots on them or not?
I cannot see red spots. I don't see red spots.
Yeah, so I think this is a different species to the ones we caught.
Okay.
And
So
we do want
then what would you like to do next?
In the desampler, please.
Okay. And no scanning.
No scanning.
Okay. Thank you. So it looks like D1 is the sampler we have
There was a
left, I
bigger
believe,
thing,
right? Correct?
In the
Yeah.
background that we may
D1
still
is
be able
still
to get to.
open. These guys out here into there.
I don't know where it went.
Make the magic happen, pilots. Thank you.
It's big enough, it will probably just come to us.
Down low. Way down. I don't know about way down, but he's down. I don't know if my description is the same perspective of what it really is meaning way down. On his way down. Coming back.
Yeah. Yeah. It's gross. It's a parasite? No, I think it's part of it. Really? Yeah. I thought it was a parasite. Which is a weird fish. Yes. You don't know what it is.
Vamos tentar coletar esse dolíolo agora.
He's in front. Yep, that was a test. That was a test. That was just a test.
He was jumping the gun there.
Nope, just a test.
It's Bathurst Row, you need a slope gun.
I see that.
Yeah, can you do suction and desampler at the same time?
Looking good.
Alright.
Yep, he's too close to us,
Ooh,
but not bad.
almost. Here we go.
Come on, baby.
Here we go.
Oh, I think we're close. Yeah,
Yeah.
nicely done. If we could
Sample.
scroll down just to
Good,
make sure
good.
he's in there.
Yep.
Yeah.
I see it.
Yeah.
Yeah.
What is the?
Thank you.
Thank you.
Thank you.
Okay, we've got him.
Hey,
Doliolid chain.
is that
Collect.
Kenophore that has
Doliolid. Where do you want us to go next?
Samplers.
Yeah, there was a Bathurst
Okay.
Row that was up
Betty,
here.
B-A-T-H.
B-A-T-H.
Let's go back out.
Oh,
Scan mode and look for this big
C-Y,
object.
Betsy-
What is it?
okay.
What?
but 0 is something.
Okay.
Yeah,
So we'll just
Just
go up
forward,
and go
little bit
forward.
left maybe to get back to where we were.
Yep.
Just in case he's still there.
Yeah, absolutely.
It looked a bit like a halifron.
Nice try. Told you. Your tricks are not gonna work with me anymore. It's not working for me anymore. That was a good attempt. I'll give you a good try.
Yeah. 8 for effort.
Yeah. He's tried it all day. Every time he wants to stop on some 1 centimeter Medusa,
It's very
he's
dark
trying
on
to
the
tell me
right.
it's an octopus
Are
or
you-
something.
Are we lit okay?
Coelacanth.
Oh yeah, the coelacanth he tried at one point too.
I did say that, I must admit.
Yep. He went over the line.
Even
Just
for
went
me.
over the line.
Those aren't so special anymore, are they?
Coelacanths.
Are they still special?
They would be from here.
Right.
We got them off Brazil.
No, not big.
Yeah, you started to look right now too. Somebody will be like, but Dougal said he saw one off the coast of Brazil.
It's a little known fact.
This right here.
Siphonophore.
Left, straight ahead.
Calicophron.
This guy.
Calicophron.
Oh.
Don't.
We just want some zoom on this, right?
Some zoom, yeah.
Yep.
Yeah.
If we
Looks
can
like it
zoom on
might be
this
a
gentleman, that'd be
Lancia.
great.
Defeat. Sifonóforo calicóforo aqui.
Drive through the siphonophore to get to the
Pequenininho.
siphonophore.
That's what I was like. That felt like a
Vamos dar
siphonophore.
zoom.
Ooh, is it a
Oh,
multi-striate
dois.
Lentia?
That's what I was gonna say.
Zooming in.
Dois nectóforos.
Before he turns around.
Pode ser uma lência, gênero lência, nectóforo anterior, nectóforo posterior.
You need to see the head and whether it's got 5 ridges, 7 ridges, or many ridges.
Podemos ver ali um ponto brilhante que é o somatocisto, que tem uma gota de óleo. E ajuda o animal a se posicionar, a manter a flutuabilidade.
If we could focus them in and get a little close when
E
you
também é
settle it in,
uma reserva de
that'd be
alimento, né? Aquele ponto branco ali na
Any
parte
do you want head or tail or
central do
all,
animal.
Dougal? So head
Yep,
would be
the
more
head, and we got to
important.
see if it's got 5 ridges, 7 ridges, or many ridges.
Okay.
We'll probably need pretty high contrast to be able to see.
Yeah.
And it to be
O
facing.
animal vê bem transparente,
And it needs to be
difícil
facing
de
the
focar.
opposite way it is.
You probably have to bring it in so close that we'll lose it soon afterwards
Okay.
to be able to get it, the lighting right.
He's down off our screen. Coming back. Nope.
Here we go, here we go, here we go. Other way.
Got it. He's coming off to the right.
Here we go.
Can we make the light a little darker on the side cam by chance? I don't know if that's an option.
So that would be the spot to have him, yeah.
Right.
Tentar enxergar as cristas do sifonóforo,
Okay. Well, that's better.
que são como linhas ao longo do corpo do animal, né? E dependendo do número de linhas, dependendo do tamanho do somatocisto, que é aquela
We're never happy.
a estrutura mais esbranquiçada, bem no meio ali da região que está pulsando.
Nice and slow. Nice and slow. We lost it. He's small.
He's too small. Oh,
Yeah,
might
he
have got a
is
shot
small.
there. Okay, that's good.
Are you sure?
Okay. Yeah, I think we got a shot.
Okay.
Corner there.
Thanks. All right. Up and onward, my friends. Slow scan, moving forward up.
Rather not.
Oik!
Not stopping. But adorable. How's that?
Another one. Oh, safe.
I don't know if I
yeah,
can handle
safe.
that. Want to stop on him?
Safe. It's not a fritter gum, that's something else.
Yep, let's take a look then, if we could. Yeah, that was
no,
my
stop.
bad.
That's okay.
That was my fault. Sorry, Dougal. But I didn't. I was not exactly
Oh, here
clear
it
on
is.
the.
Oh.
You should stop.
It's a lichnigalma,
Mais sifonóforo fisonete.
I think.
That's pretty.
We did catch one, so I guess we've documented it already. but
Yeah,
some
we can get some
would be good.
video. Sure. If we could zoom in towards the head section, that would be great.
Isn't that amazing? That's a polyp colony.
Mm-hmm.
Polyps are just kind of swimming around, not attached to wood or rocks or anything.
Go.
Frilagalma, what you say? No.
Lichnigalma.
Lichnigalma, okay.
Off to the left.
Oh, and a coelodendron.
Yep, it's great. I don't think it is. I think it's one of the ones we collected.
Yeah.
I thought I saw sparklies around it. Thanks, pilot. This is great. You want a little more zoom,
Little
Dougal,
more
or are you
zoom,
okay?
yeah.
A little more would be great
Animal
if you
que
guys
já
could.
conseguimos coletar, então não vamos
Easier
fazer a
said than
coleta.
done.
Fisonete do gênero Lichnigalma. possível ver os nectóforos que estão pulsando ali, todos posicionados no mesmo plano. Pelo formato desses nectóforos, é possível reconhecer pelo menos o gênero, né, de sifonóforos que ele pertence.
Let's drive up really slowly and collect him on the windshield before we say goodbye.
He's definitely not slowing down.
Alrighty.
You
Thank
good?
Yep.
Okay. Thank you.
Good.
Thank you.
Up and onward. All right, everybody, if you want to stop, you have to say the word stop. It's going to help much better. I was getting a little loud.
Use the select the
Nope.
Stop.
Almost.
I don't know what it is. Didn't look like a big house, though.
Down here.
Is it soft?
Does it though?
It's a dead
Looks
tail.
like a dead
Ghost
something.
tail,
Yeah, that wasn't much.
Pedaço de
Okay,
sifonóforo
thanks.
destruído.
We need to, yeah.
Didn't match my
Scanning
image map,
mode.
so I didn't yell out stop.
If we could slow down a smidge, that might help the scanning cause. Yeah.
How many buckets do we have left, Heather?
We have only, well, we have bucket 8 and a potential number 6.
6.
If somebody wants to try 6 at the end.
And the D's are
Exactly.
full too, right?
What was that?
The D's are both full. Right?
Correct. Rhizaria and Doliola chain. So 8's open, 6 is a possibility.
Heat.
Nope, we're good.
Radio.
Yep, that looked like a radiolarian.
Rhizaria. Say. Rhizaria.
After another couple minutes, do you want to go up to 400?
That's
Good.
a good idea.
Yeah. Give it another couple minutes here and then we'll switch
It's
up.
like they've all vacated here to go.
Oh yeah, I think
shallow, haven't they?
everybody's migrated. Yeah. We could go even to 350 if you want. You guys tell me what you want.
It would be great, actually.
Okay.
I think 320 was the
Bottom left,
observation.
that bright white spot.
Your Rhizarian.
Is that a Tuscarora? Oh, it's swimming.
It's swimming. It's a
Praying Calicophora and Siphonophore head only from the look of it.
really. Oh, I see what you're looking at. Yep, yep.
Outro sifonóforo.
Yeah, get a close in zoom and see.
Parece
Yeah.
da família
Close in zoom would be
Praide.
great.
Pode ser do gênero rosácea, como diz o professor Dougal Lindsay, que está aqui do meu lado.
Here it comes. In
Apenas
zoom.
os nectóforos
How's that look for you guys?
Yeah, I can see how
Only
the branch, it's not penetrating the
Nyctophorus.
mesoglea. That's what it is, yeah. Yeah.
Yeah. Okay.
Thank you.
You guys good?
Yep.
All right, so let's scooch up to 350, a little faster like we did before. Would be great, and then we're gonna hang out there for a while.
I'll leave the room for 150 meters.
Goodbye, Dougal. Close your eyes. Thank you.
Oh. Oh.
Nope, we're good.
Sumissos.
Still misses.
Oh.
We have Renata. Renata can tell us what they are.
Aqui uma narcomedusa do gênero Solmissus,
Oh, we're
muito
going.
bonita.
We're not stopping on this guy.
Já vimos várias dessas
Oh, he's
aqui,
floating with
são
us.
muito legais.
Like, you guys
Coletamos
love this guy
algumas
so
já.
much.
Yeah.
E é um gênero que pode ter várias espécies crípticas de algumas espécies de solmissos, né? Então nós coletamos algumas dessas espécies porque elas podem ser diferentes, embora tenham a mesma morfologia. Somente estudos de biologia molecular vão poder definir se realmente são várias espécies diferentes como nós suspeitamos.
Nope. Siphonophore.
Someone just say siphonophore?
Nope. Nope, I said Wahanawo.
Oh.
Yeah, what happened?
Someone say siphonophore.
And you didn't hear me say no, keep going?
I was just standing outside the room with my head in the corner. Waiting to be let back in.
Yep, 350, here we come.
Cheese enchiladas, is that what
Nope.
Someone's life is in Dougal's hands.
Nope.
Did you see those gonads?
Did you see those gonads?
Were they halfway down?
I want a better look.
Someone on the livestream oceanographic is gonna get hammered.
It's gonna get hammered.
It's They're gonna drink every time someone says gonads.
Oh, is that what they say?
Careful, careful. Dougal knows
Their
this
life
now.
is in your hands, Dougal.
Did we find any octopodema with gonads? I think we did catch one with gonads.
Yeah, well, I wasn't sure if I saw the
See the
gonads, but
gonads.
it says something that looked like gonads.
Skull.
What is it?
Yeah, we are going up at this point. We did make it to our target today of 1,200 meters. We hung there for a bit, and we've been moving up into different depth layers to investigate along the way. We're gonna stop next at around 350 meters. We are gonna spend some time in that zone for a while. The deep scattering layer seems to be above 250-ish meters at this point, so we should see some organisms either at the bottom of that deep scattering layer or more.
Today is gigantic.
Oh.
Not yet described species.
I said we can't stop. Nope.
But it can just keep up with us.
If they
Just
keep
come
up with
with.
us, that's fine.
Yeah.
Like last night, we were just staying with
Not
us.
clicked on, right?
Don't know. It's
Iris?
it's probably the iris of the camera.
Oh, just a reflection.
Yeah. Yeah.
Looks warm here.
Yes, it is.
This corner is warm, right?
Yeah, it's this. It's this right here.
It's that monitor literally burning up.
No. This fabulous big box in front of me. You want back in there, Kakani? Take over the seat for the last hour. It's almost 8 o'clock, right? Yeah. Yeah. Time flies. We're having fun. See, look, pilots are switching out. Things are switching out around here.
Going.
Alright, this is your mission, Connie. That's for you. Do it.
Find it, Connie.
Thank you.
Boom. Do it.
I already know what Google's
All the big things.
Where are the fish?
The letter.
Yeah, thank you, sir.
I thought we've.
You did.
No,
we were
it
not close to our
might be a whale fish lunch. Maybe. Still under review.
It's definitely a freaky fish.
Oh my god.
Sure if our ending city was so much cooler.
Any city is, but I don't know. Very diverse.
Temperature gradient.
Doesn't find us.
Whatever climbs right around the field.
All right. We have a little.
We're coming up.
We're supposed to be on deck in an hour.
Let's go to 350.
Haulers are coming out.
Buckle up, everyone. We're going to 350 meters.
Most exciting stuff is from while ascending or descending fast.
It looks like a salt chain. No, let's keep going.
Oh, they're long. Still.
No, I'd like to spend as much time as possible at 350. Like between 350 and 300.
Yes.
And then it's what, 20 minutes to the surface from 300?
Yeah, we'll call it,
Okay, fine. So we're out of D sampler. Are all of our samplers full?
We have an 8 and a 6.
Okay.
The 6 is questionable.
Okay.
But there is a 6 and an 8 available.
All right, so we have potentially two more section samplers. There's an Oikoplura. No. Camera, the science camera feed, which I think everyone's looking at now, will not go 360. It is on a pan and tilt. We won't be able to see behind the mount.
Yeah.
What you're asking for it back.
Oh, no.
3D printing it should be done in an hour.
What's hard?
Looked like a
I'm
fish
3D
straight
printing
ahead. Don't worry about
it.
it.
Should be done in 1 hour.
Don't worry about it.
Oh, yeah, that's
Oh, fish.
I'm not worried about it.
you want to stop
No,
us?
not at all.
No?
I'm just calling it out as I see it.
Okay.
The long
It's
one?
not a hatchet. It might be a hatchet.
I'm still writing hatchet.
I would.
Or,
Close.
or
It's one of the hatchets.
Actinopterygii.
There's a bunch of species.
Why are they called hatchet? Just because they're flat from the side?
And that, yeah, they're super flashy like that all the
Mm.
time. They're the ones that are always flashy like that.
So why are they called hatchet?
Oh, I don't know. That I don't know.
I think the wedge shape is probably
Maybe the
why.
shape.
Hatchets
Flat
also,
from the side.
they normally
Here's the hatchet we
swim
got today.
in a spiral, so they flash.
Right.
Silver, yes.
But hatchets are not flashy.
I don't think it looks like
I've
a
used a
hatchet.
hatchet and they're not flashy at all.
I'm not saying like he's not broken or beat up. It's one of them.
But not very alive.
No, definitely not. There's no water in that
That was a siphonophore.
Nope.
Another siphonophore.
another.
Those are the green ones.
Oh.
Keep going.
Completely
Don't worry about it.
opposite.
Actually, you got a sample.
You haven't?
We sucked one up yesterday, I think, but it was strained through the mesh. So we can actually get it in pieces. So we have to try that one again.
We're calling them headless greentails.
Yes.
Yes.
Okay.
We're going to try putting an
Some Mycopleura.
Yeah.
Yeah.
Or that.
Trying to get over there.
Yeah.
Starting to see more blobbies.
Helpful.
Might be good for the appendicularians.
Tell me when we're in search mode and I can.
Yep.
Stopping at 300.
350. And then like searching between 350 and 300. Solmissus. Probably Keatingnath or shrimp.
You just say that and you don't even have to open your eyes.
Nope. To each their own. That looks like a Solmissus or a siphonophore. Nope, we're starting to see more tiny larvation houses. Big siphonophore. All right, we're at about 350 meters. So then if we can do a little more forward.
Yeah.
Or quite a bit more forward, I guess, and a lot less vertical.
Dialing back from Mach 5.
Yeah.
Or not yet.
A little more forward.
Put the sound barrier there. Which is hard to do in water.
Someone thinks you're hilarious.
Ooh,
Connie
something
said
like.
go up fast. They followed.
Yep, thank you.
Boom.
So can, yeah, now let's.
If I had
we'll
known
get
that,
way more
I would have done that to the 1200 mark today.
Just let me
The
look.
Heather method is just like we just go to 1200, don't nobody look, and then we go back up to the surface, nobody look. That's a successful dive.
No, it is not.
Stop on the squid at the surface.
Nope. I would stop for a siphonophore.
Need a little
one
more forward.
tortoise,
Yeah,
a
yeah,
couple of
yeah.
things you
Yeah,
can-
I'm not getting a lot of looks.
On a pair of protists,
Maybe a new jelly.
That is
still
a
misses, does
still
that count?
misses.
Couple of amphipods I'd stop for.
Feel something coming on soon.
Vampire pods.
Wicked fluid.
Russ probably doesn't want us to suck one of those up.
Dutch word for siphonophore translates as tube jelly.
Tube jelly.
Yeah, I can't decide what
Same
how
as
I feel
Japanese.
about that. Oh yeah, it's a tube jelly.
Kurage, yeah.
So we could have just called it tube jelly instead of siphonophore.
We
Everyone
could
gives
have.
us a weird look when he says it.
Yeah, tube jelly. Tube jelly. Tube jelly. How is the word? Is that?
Buiskool.
Basketball.
Buiskool. And I think in German too. Something the equivalent.
Ah, yeah.
That's,
Because the Japanese word comes from
that looks
German.
like a little amphipod. All right.
Tiny little repellent nematodes around. And rhizarians. There's a rhizarian.
Stop to the left and up.
Heather's still back there to strangle you if you try and stop on it.
Was it that thing? Never mind. I think that was a ctenophore of some type.
You're looking for Bathcordius, right?
Yeah.
It's a low bait ctenophore.
Yeah.
Bathysaroi.
sparkling stuff.
Not
is around
Bathychorus either.
grams, right?
Bathysaroi.
You don't want to stop on that, do you?
You're looking for Bathcordius.
I'm looking for Bathycordius.
Oh, it's not Bathysaroi.
It's Curseblatt.
All right, so what we're looking for is big.
Curseblattiform.
Like big
Low
mucus
bait,
how?
Lobata.
All right, I will. Let's go fast.
It's a
Go
Tenomorph.
fast. Forward with a little bit.
Oh,
of
no, top right, go up. Bathcordius.
Is that a large mucus structure?
Up, top right. Is that, zoom in on him? Is it
I
just
think that's really small.
It is, isn't it?
Yeah.
it was further away.
What's straight ahead?
That's a baby hatchet fish.
I heard the word Bathcordius.
And a krill.
Yeah, in its full sound.
Yeah.
Oh, he was full of optimism.
Yes.
Oh. Oh.
David.
Did we lose it? All right. Let's. No.
Kind of odd.
Let's keep going.
Yeah. And a krill. You know, the very first ctenophore that was ever recorded in the scientific literature was called the snotfish.
The snotfish?
Mm.
Wow.
That's the current name for the old name for Mertensia ovum. The Mertensia ctenophore from the Arctic.
Not seeing anything. 320 yes was where you guys saw it.
Something like that.
It'll be exactly there.
So
And around this
yeah,
time too.
yeah,
Oh no,
we
it was earlier in the day.
slowly.
No, I don't remember.
Yeah, like this is great.
But it was
What's
30
happening to-
nautical miles south.
What happens to the CTD here? We might be a little bit too shallow. No, look at that. Or we might be able to catch him.
Straight ahead. Straight ahead. What is that? That's tiny.
In Portuguese, ctenophore can be sea star fruit.
Ooh.
Yeah, star fruit.
Whoa.
Hold on, let me get closer.
That is a ctenophore.
The salinity
What
and
is this?
oxygen are mirroring
Just
each
above
other.
us.
Okay, you could zoom in now.
Upload.
There's a Calicophrin type molecule underneath on the right side.
Under the elevation.
I think this is an oikopleura. That's okay.
Looks like maybe today 200 meters.
Well, let's just slowly hunt there, but we only have another 18 minutes.
If you expect them to be at the
Yeah.
interface with picnic line.
Mm-hmm.
Hyper.
I changed the iris to 1 hertz. Let's see if it gets a little cooler. That new GPU running at 80%.
That's.
Do you guys like the set camera?
It's 500 watts.
I do like this at counter. But are you asking me if I like the orientation, or?
The Saturn that we changed from. Right there.
Oh, the noise reduction. Yeah, no, it's much better with the noise reduction off. Otherwise, it looks
So
like
Mrs.
Each of these particles has like a long tail or streak. I don't want to solmiss. Let's keep going.
That's okay. You can keep going.
Sorry.
That's that first solstice we got with the high dome and the metasis.
Oh yeah. We got all the data we needed for that, right?
Yep. I think so. I'm going back to look through those scans. Of course. Calicophrin.
A little more forward should help me out a little bit.
Keeps getting me up the corner of my eye.
Right area.
There's another little oik.
A bit more forward, maybe a little less up.
Yeah. That's great. It's much better. Do you think you
So
can
this
actually
is-
tilt the main camera a little bit higher?
Didn't see it.
Kind of match our movement.
What is that?
Maybe a thoracic towers, I'm not sure. Right area.
Azarian.
No. Not sure. What time is the next time?
10.
Same location.
Yeah.
Is this camera known by now?
Yes. I don't think it's 10. I think, well, as soon as we can get Google's camera back on, I think that was scheduled was like 2 hours. Both of them. Yeah.
Rosarian.
There was
Fisenac.
the Fisenac.
Solmissus.
That looks like solmissus.
Yeah.
Yeah, we're looking for a giant larvation.
Ooh,
There's
what is that?
another. There's a, actually, can you stop? On the right. Can I just take a look at that real quick?
Small.
It's really small. Just want to take a look at the house. So that's Nabi, so Mesoequipleura.
Mesoequipleura, yeah.
All right, next please, let's keep going.
Pan to the right a little bit more. Where'd you go? I lost it. That's okay. Oh yeah.
To the right?
Let's look at
To the
that.
right.
Top right. Big sedipid. Is it tetraphalia or something else? It's tetraphalia. Okay, we have data on this, so that's okay.
Okay.
Yeah, rise area. Keep looking through your stuff.
I do like how they swim.
Yeah, they're really great.
But that's good. Good.
How do you spell Halitrophis?
Halitrophis. H-A-L-I. There's another one. Straight forward, a little bit to the right.
What is that?
Or is that a reflection?
I don't think it's a reflection. It looks new to me. It could be.
It's not moving. I think it might just be a reflection.
It might. Yeah, it must be. I didn't notice that before. Okay.
Rise area.
Let's keep going.
Area.
Let's keep going.
Oik.
Yeah, Jamie, you manifested that ctenophore.
Something?
Little knock.
Bit up.
Looks like big Anthopheliscera. Rise area. H84.
A little more forward.
Bump. Slightly higher oxygen. Starting to go down again now.
Thank
Sarah,
you.
do you have any additional controls over the FlexLink cameras? Like the auxiliary camera that we put on
No.
or yours?
No.
I know it takes a bit of work to be able to enable that. We'll bring it next time.
In our spare time.
And Barry is working on it.
That's another tetraphalia.
Is that the same? Do
Yeah,
you want to
same
stop and
one.
look at it or no?
Just zoom past, just go past and collect him on the windshield.
Or, yeah.
Or
We'll
not,
just
like
take-
close enough to see what it is. Okay, that's nice. They've got big keels compared to the one we caught.
Thank you.
What do you call that? Ballistic collections?
But we do have one more section sampler left, Dougal.
That's nice.
How do you spell this name?
You don't want that?
See, we do have one suction sample left.
Yes.
And almost no minutes left.
Yes, yes.
We have
Can,
about
can you suck it up?
15 minutes.
Like, have one try. If you miss, it's okay.
Yeah,
If it
can you
goes
just
in,
do a
it's cool.
section sampler of this?
It's got eggs.
What's that one?
It's a
T.
Mertensidae.
Mertensidae.
Mertensidae. It's quite a strong swimmer, so it will be harder than. It's a sedipid, yes. Mertensidae.
Mertensia or no?
Mertensidae, not Mertensidae.
Tenzide.
Suck. Hey, I suppose.
Oh, just outside.
Outside.
just so impressed with the control of their tentacles.
Mm. Getting them out, pulling them in, kind of thing.
Ooh. 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 Yeah.
It's a-
I know.
Okay, don't worry about it. Let's keep going and get a bathycordia, see if we can find one.
Right up.
Said that sooner.
Did he come
There
in?
you go. Yeah. All right. Here we go.
There's an, he's
There it
in,
is,
stop,
he's in.
whoa!
He's in. Definitely he's in.
Do we have anything that doesn't have anything in it?
Six.
Six.
Potentially.
6, alright, let's.
Could be a
it's just
tomopteris,
six.
a
Let's get a bit of
pteropod,
tissue out of that.
and whatever you want to put in there.
Right, so SS8 has Tetraphalia, Tetraphalia.
All right. We're going to do something amazing in 6 minutes. You can go a lot faster. Solmices.
Solmices, Rizaria.
A pod of some sort.
Solmices.
Really dead Solmis. Yeah,
Rizaria.
Solmices. That looks like
Dipper, Rizaria.
Arnema. Very long
Scythe.
tentacle. Give me a little vertical. Like Alciopid.
What is Alciopid?
So what do we
It's
think
a
is in 6?
polychaete.
Oh, polychaete.
A possible tomopteris and a possible pteropod.
Okay.
And a
But
tentacle
I don't
from
feel,
an orange
I don't
snake,
think either one got in there unless they're stuck at the top
Okay.
to the mesh.
Okay.
Yeah.
Lycopleura.
Did you go back to the image you got, or the video? With the
Solmices.
basso that you saw? Because I saw it, but I was fixed.
Right. No, I didn't get that. Just processing that source.
Kind of a ruse.
A little more vertical. Or sorry, a little more horizontal.
A lot of small giant targets, but not so many big ones.
No, yeah, I see a lot of Oicopleura houses, I think. Oicopleura or Meso-Oicopleura. Oicopleurids. Another one down below.
That's the Rosarians.
Maybe.
Prayer, or Praene at least. Yep.
Yeah.
Calicophyllum. Where are they all? Another 30 meters or so.
That's It's another
Is that one?
small Oikopleura / Meso-Oikopleura.
Dipper.
A little more forward. Thank you.
That's kind of big. Phlomissus.
Phlomissus. Oikopleura.
Oik. Phlomissus.
Phlomissus. Probably another stipe.
Phlomissus. It's gotta be here soon. See them clogged in a depth, they've got to be up shallow
I
somewhere.
know,
when
right?
They're filtering.
So we did see sinkers. Maybe we still need to be shallow.
Hmm?
Oh, stop.
Where's that?
That was kind of big.
Oh no, remember this is like one of those weird circular bands. Or no. No, it doesn't look like a circular band.
I can see the elevation in the bottom there, the white spot in the tail.
Oh, you're
Yeah.
right.
Yeah.
So it's below.
I'm all the way out.
The next one
Yeah.
we need to get.
Okay, let's do that again, but a
Next one we
different
stand.
one.
Nice area.
Here we go. Want.
I'll give you a
Really? Wow. Why is that all of a sudden?
Oh, it's rotating.
We
So we do have the
should
muds
push
in the
through
center
instead of
here.
backing up and blasting it.
Let's check what exact that's in.
Muds in the center.
Yeah, it's on the
Side. Oh, it's going through.
Yeah.
Wait, what is that? Up in the very feet. Something else. No, it came up and over.
Just turn
them all off.
I can't see it yet. I cannot see it there.
It's inside, maybe?
That's inside. It came over the lid.
It is? Oh, yeah.
Yeah.
Careful with that reversing. Oh, there it is. Without ROV lights.
Alright.
Inside.
So we like
Uppers with green reflex. Lowers are still on. It's there.
turn around.
Yep.
I don't think so. I think we're just waiting with bated breath.
Oh, yes.
Trained.
Through.
Not sure that I saw
Are
anything
you
hanging
seeing
on
anything
there.
else? I think I'm seeing
I
a
only see the
mass at the
house.
bottom.
Yeah, the little bright dot, I thought it was, but in the scan it's
It's
just
not
obviously
showing
going
up.
too fast to tell.
You might have to zoom in once if it's all kept. See if you can see it.
No.
You know, get the first passes over.
I mean, it's staying together
Yeah, I
surprisingly.
think it's could still be in there.
Well, now that's a jelly. Is that a Sabisa?
All right.
I just don't understand what I'm looking at.
Are we gonna try a different one?
Yeah, I think you're probably gonna pull us. Yeah, I don't know. Okay. We can either find something or you're gonna pull us.
The whole idea is to keep you guys on track for your next dive. So if we
That's fair.
delay this one, then the
That's
other dive.
totally fair.
So I
I'm
just
not
try
gonna
to
be
keep you
upset.
guys on track. If you want to stay on.
No, it's
I
fine.
don't care to stay in.
No, it's fine. All right. I think they're here. We're just not. I think, Dougal, I think you might be right. They might be shallower than we're expecting.
Keep pushing forward as we go, we'll find out.
That's true.
Just trap roll. Is it you responding?
It's hot here, and I think it's because of that
Okay.
computer.
All right, tell them for 200.
That monitor is also very tall.
That was a pretty good try though, because you see where
You could tell
where we
breakthrough
are
and
here.
start recovery.
We're kind of in the picnic line where they'd be hanging around.
Yeah.
When you hit that
But to
bridge,
200, it's still.
off Japan when we see them, it's like like in a 4 to 6 meter layer
Yeah.
in the picnic line, and almost none
ROV
above
bridge
or none
back.
below, and it's just that one really fine layer.
That's crazy.
We are starting
And that's where
recovery.
The picnic line's going.
There's another house.
Starting recovery. Can you give me roughly a 10-minute heads up for deck crew?
Well, in 10 minutes from now.
Probably 10 minutes from now.
All right, 10 minutes from now. Thank you.
Thank you.
All right, with that, we are going to begin recovery. So mics will be coming down, but we'll leave the cameras rolling for a while. Planning to be back in the water before too long, so keep your eyes peeled a little later tonight, and hopefully we'll see you there. But thanks for joining us on this dive.